Dear colleagues, I'm glad to see you at our conference, the last day of big conference, and we are beginning the morning session. The morning session devoted computational and experimental techniques in biomedicine. I hope the weather in your countries and in your cities is better than in Novosibirsk, because today we have not so warm weather than before yesterday and before yesterday. But I hope the weather doesn't disturb in work of our conference. And I'm glad to introduce the first speaker. It is Olga Saik with presentation Bioinformatics analysis by end system revealed members of interleukin-6 signaling pathway initiated by glucose variability and involved in diabetic retinopathy. Olga, I am glad to see you.
Yes, I'm glad to see you too. Thank you very much. So good morning. Can you hear me?
Okay. Yes, we show you, and your presentation is ready.
Yes, yes.
Okay.
So
all speakers has 15 minutes, including report. Olga, please.
Do you see my screen, the slides?
Yeah.
Okay, super. First of all, I would like to thank the organizers of this beautiful conference to make an opportunity of this talk. And even in this hard time, it's very important that we can make it in the online format. So I'm very glad and pleased. Thank you very much. And now I would like to start my report entitled The Bioinformatics Analysis by a System Revealed Members of the Interleukin-6 Signaling Pathway Initiated by the Glucose Variability and Involved in Diabetic Retinopathy. This work was performed in the Institute of Clinical and Experimental Nephrology in Novosibirsk. So, first of all, what I would like to tell is that during the diabetes, there is a number of complications that are related to the different disruptions of the biological processes. And now there are a growing body of evidence that tell us that not only the high glucose level is causing some damages, but also very important is the high glucose variability. When we see the picture as on the slide, when the level of the blood glucose rises from very high levels to very low levels, and then going again up and then again down, and we see this curve with a very high amplitude. And such pictures in patients, they cause the disruption of biological processes and leading to the pathologic conditions such as oxidative stress, chronic inflammation, and endothelial dysfunctions. All these lead to the diabetic complications. One of it is diabetic retinopathy, which is very widespread, and it causes the damage of vision. And it's a leading cause of blindness in the young and middle-aged population. And what we know is during both the diabetic retinopathy and the high glucose variability, we see that the interleukin-6 is a very important signaling participant. And it could be involved in the pathogenesis of both of these pathologic conditions. And it's important to mention that during the high glucose level, the hyperglycemia, the interleukin-6 level, it rises, it goes up, but also in the conditions of low blood glucose level, the interleukin is also upregulated. And that means when we see in the patient the picture when there is a high glucose variability, in these times, the level of interleukin-6 it goes upper and upper and upper, it rises, and it's very important for us to understand what other players could be influenced by this activation of interleukin-6 signaling pathway. And that was the aim of our work, to understand what genes and proteins are involved in such interleukin-6 signaling pathway, linking together the diabetic retinopathy and the high glucose variability. And for that purposes, we used the technology named N-System, which is developed in the Institute of Cytology and Genetics in Novosibirsk by Professor Ivanisenko and colleagues. And it allows to reconstruct and analyze the huge amount of reconstruct the gene networks based on the analysis of huge amount of scientific publications in the automatic way using the text mining technology. So in my slide, there is a brief scheme that shows how does it work. The N-System receives the publication stored from the PubMed system, then the program converts it and proceeds it, and at the end, the user can reconstruct and analyze the gene networks. So here once again, a little scheme of the N-System. N-System consists of the model of automatic knowledge extraction, the text mining model, all the information which was extracted from the publications about the interactions of the biological objects. It's stored in the N-cell knowledge base, and the user can make the analysis of gene networks by the user interface and video, which is showed in the slide. And now there is more than 4 million of facts about the molecular genetic interactions between different biological objects, which were extracted from the 26 millions of PubMed abstract publications. So what we did the first, we find what were the gene networks of the diabetic retinopathy and the glucose variability. We analyzed this network and see that, in fact, interleukin-6 is a very important player in both of these gene networks. And it is a participant of the gene network with a very high between centrality measure, which means that a lot of signaling pathways and interactions go through the interleukin signaling pathway. And it once again shows the importance of the interleukin in both of these pathologies. But we were interested to find what genes and proteins are influenced by the interleukin during the development of diabetic retinopathy in high glucose variability conditions. For that, we used again a system, and we find that there are 73 genes and proteins which connect the interleukin-6 signaling to the development of diabetic retinopathy. And these genes and proteins are cytokines, growth factors, some enzymes, protein binding proteins, microRNAs, and some others. They're involved mostly in inflammatory responses, cell-cell signaling, cell proliferation, some in-string cell cascades, the aging process and response to hypoxia, and some other biological processes, which was found by the gene ontology enrichment analysis. But when we are talking about these genes and proteins, we were in fact very interested in which of them could be used as further as the potential drug targets. And for that, what we did, we analyzed how specific are the connections of the studied gene to the gene network linking the pathogenesis of diabetic retinopathy to glucose variability. And to understand that, we used the specific measure. It's called crosstalk specificity score, and it's implemented in the N-System. it reflects the number of relation of a particular gene in the studied gene network divided by the number of total interactions of this gene in the global human gene network stored in N system. So what we see is, of course, that there is a number of genes and proteins which have a very huge number of interactions in the global human gene network. These genes are hubs, for example, the insulin, the FOS, MM9, mTOR, and so on. And in fact, these genes, as they have so many connections, they could be considered as potential drug targets, but we always have to remember that if we influence and target these genes, we can disrupt the function of a very big number of protein interactions of biological processes, and we have to understand that it could lead to some adverse reactions in the patient. So it was one group of interesting genes, but the other group, which was very interesting for us, it was the genes with a very high crosstalk specificity values. It means that their connection to the studied gene network of diabetic retinopathy in relation to glucose variability, they are very closely connected to this gene network, and in the same time, they don't have a lot of interconnections with other proteins and genes in the global human gene network. And these genes are listed on the slide. It's fatty acid amide hydrolase, endothelial lipase, aquaporin 4, and NADPH oxidase the first. So these 4 genes, they were very interesting for us because they had not so many connections in the global human network, but they are very closely and tightly connected to the studied network of pathogenesis of diabetic retinopathy. And we consider that these 4 genes have to be studied as there could be potential targets for treatment of the pathology which we are studying. And well, there is a group of 59 genes with the intermediate values of crosstalk specificity, and they require further investigation, which we are going to perform next. So to sum up, what we did is, first of all, we have reconstructed the gene networks of the diabetic retinopathy and glucose variability. We have reconstructed the interleukin interaction pathway, which connects these both networks to each other, and we find that there are 73 genes and proteins which participate in this connection between the diabetic retinopathy and glucose variability involved in the interleukin signaling. These 73 genes and proteins, they're involved mostly in the inflammatory response, the response to hypoxia, aging, some protein interaction signaling pathways, and so on. And the analysis of the reconstructed gene network, it allowed us to understand that there are 4 genes, FAAH, LIPG, aquaporin 4, and NOX the first, which are perhaps very important and prospective candidates for the further research as potential targets for treatment of the diabetic retinopathy during the high glucose variability conditions. And of course, the obtained results can help us to understand the mechanism linking the progression of diabetic retinopathy and some other complications of diabetes during the high glucose variability condition. So that's all. Thank you very much for your attention.
Olga, thank you for your excellent presentation, as usually.
Thank you.
Okay, and unfortunately, we have not so much time for questions, but you, dear colleagues, you can ask your question by chat and any way. Very excellent presentation. Thank you very much, Olga.
Thank you very much.
See you in
Yes,
future.
thank you.
And I with pleasure introduce the next speaker, Irina Ishchenko. Irina? Okay, we see you. Okay, yeah, your presentation on the screen, please.
I welcome you, dear colleagues. I present to your attention a report on the topic of features of the Hyaluronan.
Okay, we are listening to you. Continue, Irina. Okay.
Features of the Hyaluronan receptor 1 expression using 3D modeling technology in liver sinusoidal endothelial cells in C57 black mice under long-term constant lighting. I should be noted that the accelerating pace of life, the development of high technology over the last century has changed the way of human life. Almost every one of us has situations that disrupt the daily light rhythms of the body. There is the professional activity of a person at night, lights across the several time zones, a short light day and its artificial increase by electric lighting in the winter season, etc. As a result of the long existence of a person in such conditions, the circadian rhythms of the production of the pineal gland hormone melatonin is significantly reduced or almost completely blocked. That leads to the development of light-induced functional pinealectomy and desynchronosis. The liver is a unique metabolic and immunological organ which is required to perform the main detoxification function both in the normal physiological functioning of the body and in its various disease. It's also the largest source of lymph production in the body. Liver sinusoidal endothelial cells are the first cells that encounter the macromolecules of waste, pathogens, and toxins in the blood. These cells are characterized by the absence of classical basement membrane, the presence of transendothelial pores, finister, the expression of scavenger receptors, and a very powerful endocytic capacity. So the purpose of this study was to evaluate the influence of chronic 24-hour lighting on the liver expression in liver sinusoidal endothelial cells of mice, and two groups of animals were formed randomly. The control group is intact mice kept under standard lighting conditions, and the next, the 24HL group, the mice kept under 24-hour lighting during 2 weeks. It's a light-induced functional pinealectomy model. We use the following methods in the study. There are a light optical study, immunological analysis of the lymph expression that was performed on peripheral liver sections using the indirect adenine peroxidase method. The relative area and the brightness of the lymph 1 standard zones of the intermediate zones in digital images were determined, and it's necessary to say that the brightness is a parameter universally proportional to the concentration of the marker. The results were compared between two groups, using the man-Whitney test and 3D modeling of the image of the LI1 expression in liver sinusoidal endothelial cells was performed using holographic tomography by the laser confocal nanolith microscope. And next are results of light optical study.
Lina,
of the
please,
liver
sorry,
that
excuse me.
a long-term continuous elimination led to dilatation of the blood sinusoidal capillaries and dilatation of monospaces and lymphatic vessels in the edema of the interlobular connection tissue layers in the liver. There was a stagnation of the overall blood cells in the interlobular central and sublobular veins. In the organ parenchyma, lymphoid nodules or neolymphoid aggregates were sometimes detected. But I can say that no cyrotic changes were detected. Next, results of immunohistochemical analysis of the liver expression. Immunohistochemically in the liver of intact mice kept under standard lighting conditions very well pronounced staining of sinusoid endothelial cells in the periportal regions and especially in the intermediate zones in the hepatic lobes. Significantly less pronounced liver staining in these cells was found in animals with our model. Morphometrically, after 2 weeks of continuous elimination, the liver expression area decreased by 2 times, and the brightness of the staining of this receptor increased by 2%. And all these facts indicated a decrease in its concentration in liver sinusoidal endothelial cells compared with the control group. Next, 3D modeling showed the attenuation of the LIV1 signal in liver sinusoidal endothelial cells of the animals kept under long-term constant lighting. As for analyzing of received data, it's necessary to consider the following. LIV1 is the cell surface receptor in the lymph, at the first in the lymph vessels. But to date, the LIV1 expression is in the sinusoidal endothelial cells of the liver, spleen, and lymph nodes in humans, and also in the non-sinusoidal endothelium of the heart, lungs, and adrenal glands in rodents. The main ligand for the liver receptor is hyaluronan or hyaluronic acid, which is the main component of the extracellular matrix. Also is the glycocalyx component of the endothelium of blood vessels and capillaries. And hyaluronan is a part of glycocalyx on the outer surface of eukaryotic cells. The hyaluronan synthesis occurs on the inner surface of the plasma membrane of the endothelial cells. This long linear polysaccharide with a molecule weight of more than 500 kDa is extruded directly into the extracellular space. And in response to damage and increased levels of reactive oxygen species, these large polymers are fragmented, resulting in the formation of low molecular weight hyaluronan from 1 to 500 kDa and smaller oligosaccharides. And it's known that high molecular weight hyaluronan exhibits anti-inflammatory and immunosuppressive properties. But low molecular weight Hyaluronan is powerful pro-inflammatory molecule. And as for lymph function, firstly, lymph-1 serves as endocrine receptor for Hyaluronan, which is present in the surface glycocalyx of dendritic cells, macrophages, T lymphocytes, and ensures their delivery to the lymphatic vessels. And secondary, and this is important for us, lymph-1 plays an important role in the uptake of Hyaluronan in its subsequent degradation. At the same time, it's already known that the endogenous lymph-1 receptor is constitutively inactive. The lymph-1 receptor must undergo clustering to bind to high molecular weight Hyaluronan. The lymph-1 receptor must be in a salicylated form to bind to small Hyaluronan oligosaccharides, and the binding process depends on Hyaluronan concentration in the extracellular space. Numerous studies have shown that in the liver disease, such as viral hepatitis, cirrhosis, fibrosis, tumors, the lymph-1 expression in the sinusoidal endothelium of this organ decreases. We also revealed a decrease in the lymph-1 expression in the endothelial cells of the liver sinusoids in obesity and type 2 diabetes mellitus. On the other hand, most receivers note in such pathologies a significant decrease in fenestra of the sinusoid endothelium and the appearance of structure similar to the basement membrane, which indicates the beginning of sinusoid capillarization in the affected organ. Studying the expression of lymph-1 in the liver in chronic hepatitis and cirrhosis, Remote and his colleagues noted the most pronounced decrease in the expression of this receptor in liver sinusoidal endothelial cells. In those near active inflammation areas or fibrotic foci, it's known that liver parenchymal cells in the intermediate zones are more susceptible to oxidative stress. At the same time, the authors pay attention to that the Hyaluronan content increases in the serum of patients with severe liver fibrosis. And based on the above facts, we can assume the following chain of events that explains our results obtained in experiment. Apparently, a prolonged keeping of animals under constant illumination creates conditions for an increase in the reactive oxygen species level in the intercellular space of the liver parenchyma and leads to the accumulation of the low molecule weight Hyaluronan. Then, low molecular weight Hyaluronan in turn binds lymph-1 and disrupts the endothelial barrier integrity in the organ. And as a result, we note a decrease in the lymph-1 in the liver sinusoid endothelium. And in addition, liver sinusoidal endothelial cells under desynchronisis, as in chronic hepatitis, cirrhosis, can lose fenestra, which are necessary for the exchange of molecules between blood and hepatocytes. A loss of fenestra directly contributes to the obstruction of hematotissue exchange, impairing lymph drainage in the liver. the development of tissue hypoxia and hepatocellular damage. And thus, a decrease in the liver expression in liver sinusoidal endothelial cells of mice kept for a long time under constant lighting indicates a violation of the endothelial barrier integrity and a decrease in the functional activity of these cells. This contributes to the difficulty of hematotissue exchange, deterioration of liver drainage, and the development of tissue hypoxia in the liver of mice with a model of light-induced functional spinal. Thank you for your attention.
Thank you, Irina. But unfortunately, we have no time for questions. Sorry. Thank you very much. And I'm glad to see on the our conference Irina, Yulia Taskaeva, with presentation ultrastructure features of poorly differentiated hepatocellular carcinoma.
Hello,
Yulia,
hello.
We see you fine, okay.
Dear colleagues, can you allow to screen sharing from name of Natalia Abgatova, please?
It is a question to technical assistance.
Yes, yes.
Sorry, just a moment.
It is okay.
I can share the screen.
Степан, там просят
Okay. You see my screen, yes?
Yeah, fine.
Okay.
Please, please touch F5. F5 for full screen.
Yes, I try, but had a little problems. I'm sorry. Okay.
Okay.
Let's start. Hello, dear colleagues. My name is Julia Gavrilova, and I'm going to talk about ultrastructural features of poorly differentiated hepatocellular carcinoma. Hepatocellular carcinoma is a highly heterogeneous malignant neoplasm, which is characterized by morphological, immunophenotypic, and genetic heterogeneity, and that was associated with increased proliferative activity of tumor cells. The hepatocarcinoma heterogeneity on subcellular level has been confirmed by several studies. In particular, it has been mentioned that the nuclear cytoplasmic ratio in hepatocarcinoma cells is not regular, and tumor cells contain different number of cytoplasmic organelles. Edmondson-Steiner's grade system is used to divide hepatocarcinoma cells to well-differentiated, moderately differentiated, and poorly differentiated tumor subtypes. Generally, ultrastructural changes in well-differentiated hepatocarcinoma has been described and mentioned above studies. Different subclones within the tumor exist in a specific tumor microenvironment and exhibit unique clonal phenotypes. Accordingly, these diverse cell populations can show various sensitivity to the chemotherapy and form drug resistance. Advanced studies of various hepatocarcinoma histological types may contribute to the development of optimal treatment strategy for liver cancer patients. Thus, the aim of the work was to examine the ultrastructure of poorly differentiated hepatocarcinoma and identify morphologic hepatocarcinoma heterogeneity on subcellular level. Male CBA mice of 6 to 8 weeks of age with weights of 18 to 20 grams were used in experiment. Hepatocellular carcinoma 29 cell line was obtained from the Institute of Cytology and Genetics, and for tumor induction, 2 million hepatocarcinoma cells were injected into the right thigh muscle. After 23 days, the mice were sacrificed and tumor specimens were prepared according to the standard methods for transmission electron microscopy. Electron micrographs were taken to analyze tumor cell cytoplasm area, nuclear area, roof endoplasmic reticulum area, mitochondria, autophagosomes, autolysosomes, lysosomes. The tumor was composed of heterogeneous cells located within muscle tissue, and giant multinucleated hepatocarcinoma cells were often evident in tumor. There was greater structural pleomorphism of the subcellular components. Frequently, the nucleus had irregular shape and chromatin distribution. Tumor cells were tightly arranged, forming microtrabecular patterns, and some cells were possessed microvilli. Vesicular trafficking-associated structures were found in tumor cells, and they were often increased in quantity. Caveola and vesicular organelles were rare, but a great number of clathrin-coated vesicles was present. Furthermore, hepatocarcinoma cells were contained multiple endosomes. Autophagy-related structures were found to be abundant. A few number of autophagosomes that contained endoplasmic reticular areas and ribosomes was identified. Heterogeneous content at various stages of degradation was seen in numerous autolysosomes. Electron-dense lysosomes were not frequently observed. Intratumoral and peritumoral blood and lymphatic vessels were identified in the hepatocarcinoma. Obviously, the quantity of intratumoral macrovessels was significantly decreased than those in the peritumoral areas. Such blood microvessels were surrounded by continuous endothelium without fenestrae. Also, some blood vessels were characterized by an enlarged lumen and attenuated endothelial cells that contained few intracytoplasmic structures. Moreover, ultrastructural signs of vasculogenic mimicry were found in intratumoral areas. These vascular channels were formed partly by endothelial cells and were delimited by a thin basal lamina that was surrounded externally by hepatocarcinoma cells. Lymphatic vessels were rare and were occasionally observed in peritumoral area as long, tortuous, and thin channels. The nuclear cytoplasmic ratio was calculated for all studied hepatocarcinoma cells, and it was noted that nuclear ratio was varied significantly among tumor cells. All cells were divided into 5 types. It has been revealed that the hepatocarcinoma population mainly consists of 1 to 3 cell types with high nuclear ratio. The result of the hepatocarcinoma cells morphometry are presented in the table. Among the all studied hunter cells, 48% were found with autophagy-related structures. At the same time, the number and area of autolysosomes were significantly higher in type 4 and 5 hepatocarcinoma cells. However, there were no differences in the number and area of autophagosomes and lysosomes. In addition, the rough endoplasmic reticulum area was higher in type 1, 2, and 3 hepatocarcinoma cells. The main findings of this study can be summarized as follows. From all the obtained results, we suggest the heterogeneity of the basic cellular structures of poorly differentiated hepatocarcinoma at the ultrastructural level. Apparently, the hepatocarcinoma cells could have high levels of basal autophagy because autophagy-related structures were observed in almost half of the studied cells. In addition, vesicular trafficking-associated structures were also demonstrated in the hepatocarcinoma cells. It is known that small hepatocarcinomas are characterized by incomplete capillarization of the blood spaces, and hepatocarcinoma vascularization are affected by the tumor size and histological grade. In our study, the observed phenotypic changes in blood microvessels represent the capillarization of sinusoids, which can occur in hepatocarcinoma. In addition, atypical blood vessels with an extremely wide lumen and surrounded by the endothelial cells with attenuations were found in the tumor. Morphologically, these vessels are similar to the so-called mother vessels, originated from pre-existing venous, since they were characterized by extreme thinning of the lining endothelium and decreased number of intracellular vesicles, including vesicular organelles. And our visualized vascular structures were limited by a thin basal lamina from luminal space, and located externally hepatocarcinoma cells. These ultrastructural findings are corresponded to the phenomenon of vasculogenic mimicry that firstly described for primary uveal melanoma. Our results confirmed the presence of vasculogenic mimicry in a purely differentiated type of hepatocarcinoma. Since the vasculogenic mimicry is used by the tumors to increase nutrients and oxygen supply and to sustain growth and progression after chemotherapy, vasculogenic mimicry targeting represents a promising strategy for the hepatocarcinoma treatment. Finally, further research should provide information about the intratumoral heterogeneity mechanisms in hepatocarcinoma and its relationship with the hepatocarcinoma vascularization and progression. Also, it seems important to evaluate these events in tumors with various degrees of differentiation. Thank you very much.
Dear Yulia, thank you very much for your presentation. Maybe anybody want to ask question? We have some time for this. This topic is very important for oncology, for chemotherapy. Okay, thank you very much, Yulia.
Thank you.
Next presentation also is connected with oncology. Next speaker is Nikolai Latyshev. Nikolai, are you ready?
Hello, yeah, I'm ready.
Yeah, we see you. Please, your presentation. Okay, and full screen.
Okay, you can see?
Yeah, okay, great.
Good morning, dear colleagues. Today I'm going to speak about research work with the title Analysis of Early Acid Carcinoma with Electron Paramagnetic Resonance. The work was carried out at the Krasnaya Scientific Center of the Siberian branch in the laboratory of the organism Extreme States Research. The aim of this study was to evaluate the dependence of changes in the content of metal-containing proteins and free radical forms in the experimental tumor suspension at different periods of tumor growth with electron paramagnetic resonance. EPR spectra were recorded on an EPR Fourier spectrometer analysis of the Krasnoyarsk Regional Center for collective use of the Federal Research Center. First, we analyzed the range from 650 to 4,350 Gauss, and then choose the area of interest from 3,150 to 3,650 Gauss. During all measurements, the sample temperature didn't exceed 85 Kelvin degrees. Figure 2 shows tubes that were filled with 0.8 samples. Next, the samples were placed in a dewar with liquid nitrogen. Immediately before EPR spectroscopy, frozen cone-shaped samples were removed from the shell and placed in an EPR spectrometer. In this experiment, we used outbred laboratory mice obtained in the State Scientific Center of Biology and Biotechnology Vector in Novosibirsk. A suspension of early acid carcinoma was inoculated into the abdominal cavity of mice in the amount of 3 million cells diluted in 2 milliliters of saline solution. Figure 5 shows the pathways for the conversion of nitric oxide in the organism. There is calcium dependent pathway where guanylate cyclase is activated by nitric oxide and converted to guanosine monophosphate, which is a universal regulator of intracellular metabolism. There is also an oxidative pathway with the formation of active radical forms and denitrosyl iron complexes. It is known that some cancer cells are capable of producing nitric oxide, which stimulates the synthesis of growth factors, which inhibited the protective activity of macrophages and improves the permeability of the capillaries of the abdominal cavity for the transport of nutrients, which has a positive effect on tumor growth. We believe that the determination of the metal-containing proteins and free radical forms concentrations using this method can be an important prognostic parameter for assessing the state of the tumor. Metabolic changes will inevitably affect the functionality of cells and as a consequence the dynamics of tumor growth. This figure shows the components that we observed during the experiment. Transferrin is represented by peak 1. The third is the intrusive iron complexes, and the fourth is molybdenum-containing components. A broad ferritin signal is also seen. Signal values from transferrin, ceruloplasmin, and ferritin were stable indicators independent of the phase of tumor growth, since they are found exclusively in plasma, and their functional activity applies to the entire organism as a whole. We also consider the area from 3,150 to 3,650 Gauss. The results showed that the molybdenum-containing components also didn't undergo significant changes. However, the signal of the intrusive iron complexes markedly decreased with the development of early Hepatocellular carcinoma cells. This figure shows high field sections of free resonance curves of the tumor on day 7, 9, and 10 of tumor growth. Table 1 presents data on the total intensity and characteristics of the signal of the intrusive iron complexes. Intensity is defined as the area under the integral absorption curve. Delta H is a line with Hn is a resonant field. After conducting the research, we were convinced that using the APR method, it is possible to assess the changes in the concentration of metal-containing proteins and free radical forms in suspension of early Hepatocellular carcinoma cells. We believe that the method we use can be used to simulate tumor growth. This trend observed during 3 experiments, but the difference is not always large enough. We will continue our research to increase the database. Thanks you for your attention.
Nikolai, thank you very much for your presentation. Very interesting topic. And we are glad that Siberian Federal University is us today. Any questions? No questions at last. Maybe questions will be in chat later, and you have the possibility to answer this later. Okay?
Okay.
Okay. Thank you very much. And we continue. Next speaker will be Danilo Chernopyatov with presentation the confocal microscopy for assessment of transdermal transport of the photosynthesizer chlorine E6 after non-ablative photothermolysis. Danilo, are you ready?
Yes, can you see me?
Okay, your presentation on the screen, please.
Dear colleagues, let me introduce our scientific work on the topic, the confocal microscopy for assessment of transdermal transport of the photosensitizer Hyaluronan E6 after non-ablative photothermolysis. Recently, the interest in percutaneous drug delivery methods has increased, and every day there is more and more information about the development of new transdermal delivery systems. Actually, the development of new ways to deliver drug molecules through the skin not only improves the characteristics of the drug in terms of efficacy and safety, but also there is contributes to the patient's adherence to treatment. Traditional methods of drug delivery, oral or intravenous, have negative consequences, which complicates treatment and causes the patient to be averse to treatment, problems with the assimilation of certain drugs. In this regard, transdermal therapeutic systems are aimed at transferring pharmacological agents through the protective layer of the skin. But the difficulty of creating a vehicle is that many conditions must be met for each drug, since it is necessary to overcome the specific obstacles associated with conventional delivery methods. But despite all the advantages, when used, most drugs are not able to penetrate into deep layers of the skin due to the protective surface layer of the skin, stratum corneum. There are many mechanical, chemical, and physical methods that promote the passage of drugs through the skin, but in most cases, these methods are ineffective and unsafe. Thermal ablation is a method that creates conditions for the transport of medicinal agents through the skin. The local thermal exposure damages the surface layer of the skin exclusively at the site of exposure without damaging the deep structures, which improves the passage of lipo and hydrophilic agents into all layers of the skin. The laser ablation occurs due to the absorption of light energy by tissue cells. The water in the cells evaporates, creating microchannels in the skin. Analyzing the current work on transdermal transport of drugs, we can conclude that the fractional laser photothermolysis is a promising method of exposure for transdermal transport of medicinal agents. As a result of fractional laser photothermolysis, numerous minor skin microtumors occur, while about 95% of the skin remain intact. In contrast to conventional laser technologies, which focus on continuous exposure across the entire surface of the skin, FLP has a fragmentary effect, causing small areas of thermal damage called microthermal zones. The healthy tissue surrounding the MTZ ensures rapid recovery of the epidermis due to the migration of viable cells to the lesion sites. The transdermal delivery of the drug through the microthermal zones created by photothermolysis allows to prevent the drug from being metabolized in the gastrointestinal tract and liver, as it happens when the drug is taken orally or administered systemically. These methods improve their pharmacological effects and at the same time reduce the amount of pharmacological agents. Fully, the above applies to photosensitizers. Photosensitizers are a group of drugs used for photodynamic therapy. These are substances capable of absorbing light of a certain wavelength and converting its energy. Activation of PS by light energy leads to triggering of photochemical reactions and the appearance of the fluorescence effect, which can be detected with the help of special devices. One of more important area of investigation is the intradermal, transdermal delivery and cutaneous penetration of different photosensitizers, including after topical application. The laser scanning confocal microscope is the most suitable method for diagnosing the effect of fluorescence of the drug in all layers on the skin. The purpose of our research is to study the features of transdermal transport of the medicinal photosensitizer Hyaluronan E6 in the form drug of the radiogal after non-ablative photothermolysis using confocal microscopy and calculating the transdermal transport coefficient based on the fluorescence intensity of the photosensitizer molecules. The study included male mice divided into 3 equal groups. The first group of photothermolysis and application of radiogal photosensitizer, the second group of radiogal application, and the third control group. After euthanasia, a scan flap was excited from the abdominal wall of all animals, and clear sections were prepared for laser confocal microscopy. Confocal laser scanning microscopy technique is fluorescence-based image and offers greater resolution due to its point elimination and the detection properties. To analyze radical fluorescence, microscopy was performed at the optimal wavelength for the study. Fluorescence excitation at 458 nanometers and fluorescence registration at 675 nanometers. Results. In the control group, without exposure to photothermolysis and application of photosensitizers, the presence of background radiation and spontaneous fluorescence of the skin was noted. Confocal microscopy of skin, the application photosensitization alone, active fluorescence fluorescence radiogal is observed throughout the entire epidermal layer. Microscopy of the skin treated with photothermolysis with application of radiogal demonstrates the fluorescence of the drug in all layers of the skin and its structures, including hair follicles and sebaceous glands. Presenting the transdermal transport coefficient of the drug as a ratio of the fluorescence intensity in the reticular layer to the fluorescence intensity on the epidermis multiplied by 100, we obtained objective data from which it follows that after a standard cutaneous application of radiogal, the transport coefficient is 3.68 cu, while the application of the drug after FLP provides a 27-fold increase in the transport coefficient, which is 103.7 cu. In conclusion, we can say non-ablative laser photothermolysis significantly enhances the intradermal penetration of chlorine E6 photosensitizer. The use of confocal microscopy allows us to estimate the depth of chlorine E6 penetration into the skin layers, as well as to quantify the increase in the intradermal distribution of the drug by calculating the transdermal transport coefficient of the drug. During the study, we didn't find any references in the literature about confocal microscopy of cryomaterial with chlorine E6, so it can be assumed that such a study is being carried out for the first time. Distribution mechanisms and localization of chlorine E6 in the skin structures are of interest, which requires further research in this direction. We express our gratitude to colleagues for their help in the study, and thanks for your attention.
Danilo, thank you very much for your presentation. It is a very interesting topic, and me as a co-author with Danilo, we are going to investigate, to continue our investigation in near future about transdermal transport of photosynthesizer in the different structure of skin. Maybe anybody wants to have a question? in my opinion, it is our last presentation in our session, and I think it's a very interesting section because the different area of medicine, like laser medicine, oncology, and endocrinology was in our presentation, and the last presentation have a bridge to physics, to another discipline, because laser medicine and paramagnetic resonance and so on, it is very important team for next investigation in modern medicine, because modern medicine is closely connected with another area of knowledge, for example, mathematics, physics, genetics, and so on. Maybe anybody ask some questions or tell your opinion about this session. Thank you very much for all of participated in our session, to our guests, and we have 15 minutes to next session. Next session will be devoted in silico medicine, yeah, and I think Dr. Shatrov, yeah. You will be next chairman, yes. Please switch on your microphone. We don't. Oh, okay.
Yes, yes.
Yes. Are you chairman of the next section? Yeah.
Yes.
Yes. We have 15 minutes and less to next session. Thank you very much. Where are you from?
Well,
next session will begin
yes, yes.
15 minutes yes, after 12. Okay.
Good morning.
Thank you.
Visualization of blood erythrocytes sedimentation process on transitive layer erythrocytes plasma.
Unfortunately, can you share your screen?
Content. Actuality of the presented problem for diagnostic signification of the standard erythrocytes sedimentation rate ESR test. 2. A review of publication of mathematical modeling and experimental methods for process of ESR. 3. Device for visualization and graphic recording of the erythrocyte sedimentation process. 4. Results of visualization information erythrocyte sedimentation process of the red blood. Conclusion. Actuality of the present is by widespread usage of laboratory blood test. The purpose of the SIS work is to prove a specific character of SIS analysis. Process is registered by devices for visualization of blood cells deposition with high degree of space and time resolution in real time. The research of
Dear Anatoly,
dynamic
Anatoly, sorry.
Yeah, yes,
Your presentation will be 2:30.
yes.
Yes, because I thought that you are chairman of next session. Okay.
Yes. Okay.
Yeah. Yes. We have a break. 10 minutes to beginning of next session.
Yes.
Thank you very much. Thank you.
Thank you. I speak? Yes?
No. Your presentation will be 2:30. After break.
Да,
After sections.
да, вы подключились не вовремя, к сожалению. У вас презентация в 2:30.
I speak? No?
У вас презентация позднее. У вас презентация в 2:30 по новосибирскому времени. Видимо, вы неправильно...
Я считал по уральскому времени.
У вас уральское время, да. Вот у вас презентация позднее просто. Вот останьтесь, да, пожалуйста, сейчас у нас кофе-брейк, и после этого мы продолжим.
Тогда какое время у меня будет?
Я сейчас вам скажу. Давайте я посмотрю точное время по уральскому, да?
Да.
Окей.
Но я живу по московскому времени.
Окей. Я понял. Одну секунду. Так, ваше выступление по московскому времени в 10:30 по Москве.
Окей.
Да. Тут, видимо, все-таки, да, произошло недоразумение. Приносим извинения за...
Окей.
Да, спасибо.
Thank you very much.
Да, thank you.
Dear colleagues, we have some technical problem, but now we continue. We begin the next session of our conference, and this session will be named in-silico medicine. And today, our speaker is Alexander Lykov. Alexander, are you ready?
Yes.
Okay. Please switch on your presentation.
Так, I have a problem. It's okay. 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 Are you see?
Yes, yes. And please touch F5, please, for full screen presentation.
Mm-hmm.
Okay. Yeah, great. Please.
Dear colleagues, I will present our experimental study concerning for the toxical effects of new made non-selective sorbents. This study was done in our institute, and you can see who is for this. The main purpose of this study was to establish possible to use this new based in gamma aluminum oxide and polymethylsiloxane and carbon nanomaterials modified non-selective sorbents in humans. In this, you can see the main stage of preparing our non-selective sorbents. It began with the modification of gamma oxide aluminum oxide with polymethylsiloxane in aqueous phases, drying, and then it will be modified with single-wall carbon nanotubes with different types of dispersions such as povidons or carboxymethylcellulose. And next, we use the carbon nanofibers synthesis in Boris Koch Institute of Catalysis. Next, we have obtained human platelets using special plasma lifting tubes. It's processed to enrich the number of platelets, and during the examination, we studied the effect of co-incubation platelets with new prepared sorbents on oxidative reductase activity, and also quantitated the number of granules in platelets plasma. Okay, excuse I topic to end again. Excuse me. In this table, you can see the our results of study. You can see that the samples of large and intermediate of gamma aluminum oxide particles modified with polymethylsiloxane significantly reduced oxidative activity, and you can see that modification of this complex of single-wall carbon nanotubes with dispergent povidone also reduced oxidative activity in platelets. We can see also that when used single-wall carbon nanotubes with dispergent methylcellulose, we can also indicate that oxidative activity of platelets significantly reduced. Another types of sorbents and with single wall carbon nanotubes or with carbon nanofibers now have significant influence at platelets accelerated plus activity. Whereas when we can look here, you can understand that all types of sorbents can significantly process to decrease number of granules in platelets. It's maybe as a result of activation in these platelets. And at the end, we think that long-term exposure with platelets and new prepared non-selective sorbents process to in sometimes inhibition of auxiliary reductase activity, and it's process to activation of platelet cells. It's usually observed when used another type of sorbents during detoxification of people with some diseases such as poisoned or infections. Thanks for attention. It's all. Okay. No. Vladimir Valeryevich, I finished. Are you here? No? Hello. Where are you? Wait. Vladimir Valeryevich, where are you? It's now. Okay. Maybe any have questions to me, then Vladimir Valeryevich absent. No? Okay. Wait. Sometimes.
Alexander?
I finished.
Yes.
Yes. And good have questions now.
Yes, but our session today is very short, and Alexander continue this session. His next presentation, biocompatibility of the complex aluminum oxide and polymethyl siloxane modified with carbon nanofiber. Alexander, your next presentation.
Okay.
2.
1 second. I will be. Escape. 1 second. Okay. Just a moment. Just a moment. Okay. Excuse. Okay. This part of my speech is done in collaboration with Institute of Catalysis in Novosibirsk. And some colleagues.
Alexander, we don't see your presentation.
Okay. Okay. Escape, excuse me. I will be here. Okay.
Okay, sure. Great.
This study was conducted with the Institute of Pathology, Novosibirsk, and with our laboratories institutes. The main idea of this study was to examination possible compatibility of new non-selective sorbents based on gamma aluminum oxide modified with polydimethyl and with carbon nanofibers. Because all newly made subject compositions can be studied to possibly toxic for human or animals. Okay. It's similar as in the first presentations. We previously obtained the composition of waste aluminum oxide and polydimethyl and next we used modification surface using different type of carbon nanotubes and obtained new non-selective sorbents. Okay. The cells which were used in this study is the human-born neuromesenchymal stem cells, which were obtained by traditional methods, which you can see in this slides. Okay. The next, we studied the toxicity and proliferative activity using MPTSA. Next, then we studied the effect of these non-selective sorbents to possibly the production of stable nucleic oxides in supernatants from this bone marrow erythrocyte stem cells. And at last, we examined the rate of apoptosis using special stain, such as orange and Ezidium bromide, excuse me. Okay, in these tables, you can see that we used a different type of material walls to prepare our carbon nanotubes. It's all are hydrocarbons with different type of carbon in its structure. Also, during this preparing of carbon nanofibers, we have changed temperature and time exposure during these preparations. And you can see that obtained carbon nanofibers have different type of specific surface and have a different volume of pore. Also, under electron microscopy, you can see that they differ by the length and the width of obtained carbon nanofibers. Using these carbon nanofibers and using the very small particles of aluminum oxide and very big particles of aluminum oxide, we prepared some samples of non-selective sorbents modified with carbon nanofibers. You can see that when you used small particles of aluminum oxide, we also increased capability to absorb some chemical reagents and then increase the content of carbon nanofibers. But when we used particles with large size, we can see that increased content of carbon nanofibers possessed to increase the absorption ability of these obtained non-selective sorbents. Okay. During our examinations, we observed that only 3 samples have significantly increased proliferative rate of bone marrow mesenchymal stem cells. It was a sample of number 45, number 38, and number 39. It's all based off small particles of aluminum oxide. Another type of sorbents have no significant influence of proliferation of bone mesenchymal stem cells activity. When we studied the levels of production nitric oxide in supernatant of these bone marrow mesenchymal cells, we can see that samples number 32 and carbon nanofibers DNR181 and NN3 also significantly increased levels of the nitric oxide production by mesenchymal stem cells. Another type of sorbents or initial carbon nanofibers now have significant influence on nitric oxide production by bone marrow mesenchymal stem cells. At the end, we can see by this picture that all type of obtained sorbents and initial carbon nanofibers significantly increase the level of apoptosis in these bone marrow mesenchymal stem cells. The reason of this may be that this bone marrow mesenchymal stem cells will obtained from patients with chronic diseases, and it's were older. It's known that the age and presence of illness can affect functional activity of bone marrow mesenchymal stem cells. Okay. This study requires continuous using another type of cell type. So it's okay. It's finished.
Dear Alexander, thank you very much for your presentation. Maybe anybody have some questions to Alexander, because this session is very short, because in silico medicine it is maybe a very serious area of medicine and chemical. No questions?
Okay.
Alexander, thank you very much. And we wish you another future investigation and new results in your topic. Thank you very much.
Thanks.
And now we have a break till 1 o'clock. Thank you very much, all of you, for all of participants of our session. Thank you. See you next section at 1 o'clock.
Добрый день, коллеги. Будьте добры, слышит меня кто-нибудь из поддержки, из техподдержки конференции?
Не из техподдержки, звук хороший.
Звук хороший, да.
Спасибо большое. Я хотел бы, если можно, техподдержка, представитель техподдержки или соорганизации, не мог бы я задать один вопрос?
Секунду, сейчас я попробую позвонить.
Хорошо, спасибо большое. У меня не технический вопрос, а скорее организационный. Может быть, вы мне можете помочь?
О, попробую, давайте.
Все очень просто. Меня попросили быть председателем этой сессии, но, к сожалению, у меня нет информации об аффилиациях участников. Есть только имена, имена, фамилии и название докладов. Ну и, собственно, город. Если этого достаточно, то не проблема. Если есть возможность оперативно мне как-то переслать информацию об аффилиациях, я буду благодарен, но, на мой взгляд, это как бы не супер критичное.
Ну, вы знаете, я вел предыдущие сессии, в общем-то, представлял авторов. И вчера тоже на сессиях было то же самое. Поэтому,
Отлично, давайте тогда так и поступим.
да, авторы представляют себя самостоятельно, и аффилиации видны на первом слайде, как правило.
Хорошо, большое спасибо вам. Все, я тогда в режиме ожидания, я уже на месте.
Хорошо, спасибо, всего доброго.
Спасибо большое. Dear colleagues, are you here?
Yes, fine.
Okay, cool. We have less than 5 minutes before the session, so I can see here almost all participants except one, I guess. But anyway, we can prepare for our session, if you don't mind. And I would like to thank the organizers for their invitation of me to be a chair of this session. And I would like to remind that today we are at the session of IEEE Euro-Siberian Conference on computational technologies in cognitive science, genomics, and biomedicine. And I'm very happy to participate here as well. So also I would like to say that we have a very dense program for this session. And basically a reporter will have 10 minutes for his or her report, and also 5 minutes for answers and questions and answers. So I beg everybody to be in time and to be as dense as possible to avoid any prolongation of our session, because we have a very important next session with plenary lectures. Okay, so we can wait for next 2 minutes before the beginning of our session. Is it okay?
Sure.
Okay. And also, I apologize. Unfortunately, I have no information about your affiliations, so I would possible only to call you by your name and the title of your presentation. Okay, so it's time to begin our session. And the first report, first presentation today will be done by Vladimir Gorchakov from Novosibirsk. So the presentation is called as parameters of hydration and structure of lymph nodes during the maximum development of lymphoid tissue. Okay, so please go ahead.
Let's go start.
Yes, please.
Dear colleagues, my report, parameters of hydration and structure of lymph nodes during the maximum development of lymphoid tissue. Emergence of the concept of the lymphatic region defined no approach to studying lymph nodes proceeding from the regulatory role in maintenance of water homeostasis. The mechanism of maintenance of water homeostasis is the lymphatic drainage with ensuring immune function of lymph nodes. The efficiency of these functions depends on hydration characteristic of lymph nodes and is connected with originality of the drainage of lymphatic regions. It represents relevant to study the influence of food component on structure and function of lymph nodes. The purpose of work is the estimate influence of parameters of hydration of forming and more functional status of regional lymph nodes of different localization. Design of research. The experiment was executed on the 15 fire threads of V-star. Their age is 3-5 months, young animals, period of the maximum development of lymphoid tissue. Object of research are inguinal, mesenteric, and tracheobronchial lymph nodes. Message used in work. Morphological message. Lymph nodes fixed in chin neutral formalin. Further lymph nodes subject to the classical scheme of the paraffin filling with preparation microsection of the microtome. Microscopic sections painted hematoxylin eosin and by Masson. Thermogravimetric method. The principle of method consists in drainage of a lymph node at a constant temperature with registration on analytical scales of change of weight. Definition of different fractions of water, common, free, and bound, coefficient of hydration and density. Statistical data processing is earned out by means on the license program STAT plus Pro. Assessment of liquid content in lymph nodes is important for characteristic of condition of lymphatic drainage and immune function, and establishment of correlation of water balance with construction feature of the lymph nodes of different lymphatic region. The circulation of liquid or water is condition of life activity of organism. Maintenance of water homeostasis is represented important function of lymphatic system, which basis is the lymphatic drainage of internal environment of organism and the ecological space. Liquid in lymph nodes is distributed by sector as the free fraction of water is a lymph in lymphatic science and blood in vessels as the related fraction of water is intracellular and extracellular interstitial liquids relative to cells. It is necessary to attend to quantitative distinctions in the content of the common water and the structural fractions and characteristic of water liquid component of lymph nodes. The quantity of liquid is not a standard indicator for different tissue and organs. The volume of liquid varies depending on localization of lymph nodes. The mesenteric lymph node distinguishes most content of the general water in comparison with inguinal and tracheobronchial lymph nodes. Content of the common water can influence the size of fluctuation of indicators of free water. The most part moving free liquid lymph is concentrated in the lymphatic sign of lymph nodes. The free water correlates with the lymph volume of lymph nodes sinuses. It is noted that lymph volume of sinus is maximum in the mesenteric lymph node. There is a minimum lymph volume in the tracheobronchial lymph node. There is an intermediate value of a lymph volume in the inguinal lymph node. Volume interstitial and bone water change similar in lymph nodes. The effective lymphatic drainage is caused by development of lymphatic sinus. The trend of decrease of hydration coefficient forms the following row: inguinal, mesenteric, tracheobronchial lymph nodes. The degree of density of lymph nodes is other important indicator. The trend of the indicator forms the progressive increasing row: inguinal, mesenteric, tracheobronchial lymph nodes. Degree of the site density is connected with a cellular and fibrous component of lymph nodes. We obtained interesting data on hydration of lymph nodes. We obtained the evidence of difference in performance of lymphatic drainage functions by lymph nodes from different areas of a body. The lymphatic drainage is connected with development of lymphatic signs. So, mesenteric lymph node possesses high, inguinal lymph nodes possess average, tracheobronchial lymph node possess low activity of lymphatic drainage, lymph transport through lymph node. We defined a morphofunctional specialization of lymph nodes proceeding from prevalence of time Cyrus-dependent and or Cyrus-independent zones, the size of a science system. The variant of structural organization of lymphoid tissue is associated in the inguinal lymph node with performance of drainage function and the immune response of the cellular type, test zone. The variant of structure of the tracheobronchial lymph nodes is characterized by prevalence of immune function at the immune response on humoral type, B zone. The variant of structure of the mesenteric lymph node is combined alone with drainage and immune function of the mixed type of the immune response. Conclusion: parameters of hydration are objective quantity criterion of morphofunctional status of regional lymph nodes during the maximum development of lymphoid tissue. Each of regional lymph nodes had special morphological option structure that allows to allocate lymph nodes with prevalence of drainage function for inguinal lymph node, with prevalence of immune function for tracheobronchial lymph node, and equally expressed drainage and immune functions for mesenteric lymph node. The received results confirm basic provision of the concept of lymphatic region. The principle of the region should be considered as predictive assessment at a research of lymph nodes in physiological conditions and pathology. Thank you for attention.
Thank you so much for this very interesting report. Do you have any questions, dear colleagues? We have 4 minutes for... Окей. Уважаемые коллеги, есть какие-то вопросы у вас?
Окей.
Пожалуйста, мы имеем 4 минуты.
О, я уложился. Хорошо.
Если вопросов нет, тогда, если можно, я задам вопрос. И если можно, пожалуйста, Владимир, переключите на слайд, где вы показываете три графика, три плота. Вот, да, вот этот. Нет, предыдущий. Вот этот. Мой вопрос, я заранее прошу прощения, может быть, я не совсем уловил, что было сказано. Мой вопрос таков. Вот, скажем, на первом плоте, который здесь изображен, показано три столбца. Общее содержание воды, связанная вода и свободная форма воды. А в каких единицах измерений представлены эти столбики? Вот мой первый вопрос. В каких единицах?
Ну, первые три столбика, это касается общей, связанной и свободной воды, это представлено в процентах. А вот объем лимфы в лимфатических синусах, это перерасчет жидкости именно на синус. Это в миллиграммах, как там, миллиграммах на миллиметры кубические. В этом плане перерасчет.
Хорошо, спасибо большое. Тогда вот, если можно, я хотел уточнить по поводу воды. Мне не совсем понятно, почему сумма свободной и связанной воды не дает 100%. То есть почему общая вода здесь представлена величиной 43,9%. Мне не совсем понятно. Ведь вы же оцениваете...
35 прибавить 8, это будет 43. То есть если вы сложите...
Нет, насколько... может быть, я просто неправильно уловил. То есть, правильно ли считать,
но там
что...
есть потери. Когда мы это рассчитываем, там могут быть потери. И поэтому 100% может не быть. Может даже быть больше или меньше. То есть это зависит, во-первых, от математических расчетов, как правило. И мы не подгоняли. То есть какие цифры получили, те и поставили. И потом часть воды может теряться и не учитываться. Видите? Учитывали кровь, например. Поэтому может быть несовпадение.
Хорошо, спасибо, я понял. Спасибо большое. У меня больше нет вопросов. Может быть, у участников есть вопрос. У нас есть 2 минуты.
Спасибо.
Ну что ж, спасибо большое, Владимир. Очень интересный доклад. Тогда, if you don't mind, we can follow to the next report. The author is Anna Shurligina from Novosibirsk. I apologize as well. I don't know the affiliation of this participant.
Олег, it's me. Instead of Anna Shurligina, because she has some problem, and she asked me to present our results here. Dear colleagues, thank you for possibility to present our study. Just a moment. Yeah. Do you see my presentation?
Yes.
Oleg? Okay. Okay.
Everything is fine.
Yeah.
Okay, we can see.
Dear colleagues, today I present the results of pilot study devoted to quantification of the interaction of hemosorbents with red blood cells using scanning flow cytometry. Study was conducted in collaboration of Research Institute of Clinical and Experimental Lymphology and Institute of Chemical Kinetics and Combustion named Voevodsky. Two parts of this study. The first part, this is a medical part concerning of using of sorbents in medicine, because medical solvents mainly based on charcoal are widely used for treatment various diseases. The first of all, renal insufficiency and drug and other intoxication. Sorbents are aimed at removing toxic agents from the body and which were provided by their porous structure and the chemical nature of surface. And mechanism of action of sorbents is to fix various toxins in the porous space and remove them from the body naturally when ingested. And this type named enterosorption. And another direction at the passing or perfusion blood through columns with sorbents, and in this way, this direction named in Russian and post-Soviet Union space hemasorption, but another word has the term hemiperfusion. And another direction is the local application of sorbents to wound, and this named vulnerosorption. And medical sorbents must be mechanically strong. It's the first requirement to this one. And another requirements, they will be compatible with biological tissues. First of all, with in case of hemiperfusion, with cells of blood, red blood cells and platelets. And our presentation devoted to interaction between sorbents and red blood cells. And from this point of view, mechanically strong sorbent with we investigate mechanically strong sorbent with improved organoleptic properties. You can see here this sorbent on the basis of gamma aluminium oxide with polybidimethylsiloxane. It has white color, and another sorbent for comparable is sorbent SUMS the first. This abbreviation SUMS the first is Siberian углерод минеральный sorbent, Siberian charcoal mineral sorbent, this abbreviation. And the aim of this study to investigate the effect of this original sorbent and sorbent SUMS the first, and their influence to biometric parameters, functional parameters of red blood cells using method of scanning flow cytometry. And this method was conducted in Institution of Kinetics and Combustion in the laboratory of cytometry. And the features of these sorbents, this one and this one, consist in this technology of obtaining of sorbent SUMS the first require very high temperature, about 700 degrees Celsius, and this technology is not good and a little bit harmful for ecology. here you can see the porous of sorbent, and this porous is comparable with another molecules of toxic agents and bacterial. Here you can see the Staphylococcus aureus on the surface of sorbent. Here you can see investigated sorbents. This is a carbon-free hydrophilic and hydrophobic sorbent on basis gamma aluminium oxide and polydimethylsiloxane, and sorbent above mentioned sorbent SUMS the first. Here we present experiment, schematic installation with glass column with sorbent. The volume of sorbent about 2 grams, and through this glass column we using infusion pump with constant speed, about 4 milliliters per minute, and blood obtained from patient after joint replacement perfused through this column with sorbents, and collected in tubes with anticoagulants. After this special sample preparation of blood, function parameters were determined on a scanning flow cytometers in Institute of Kinetics and Combustion in laboratory of cytometry. Average data for 6,000 red blood cells. Here you can see the results of study of physical and chemical properties of sorbents with a grain size of 0.2 to 0.8 millimeters. Structural data on the specific surface area, pore volume, bulk weight, sorption activity by methylene blue dye, and sorption of Staphylococcus aureus are presented. You can see that structural parameters of sorbents are similar, which indicates the uniform distribution on hydrophobic ingredients, carbon and polydimethylsiloxane over matrix granule, aluminium oxide. At the same time, the sorption activity of SUMS the first was significantly higher compared to gamma aluminium oxide and polydimethylsiloxane and aluminium oxide alone, with a similar, with a smaller specific surface area of the sorbent and a smaller pore volume. In terms of sorption activity against Staphylococcus aureus, SUMS the first is significantly superior to the sorbent aluminum oxide with polydimethylsiloxane. Here you can see the data on functional parameters of red blood cells, particularly anion exchange rate and ultimate membrane extensibility, evaluated on the basis of data on red blood cell hemolysis using the developed mathematical apparatus developed in the laboratory of cytometry. And this table shows the data of number of active BAN3 protein and the values of ultimate extensibility of erythrocyte membrane. This is a ratio of stiffness to elasticity. And so the transmembrane protein band 3 is the main exchanger of the erythrocyte, and a polyfunctional membrane glycoprotein involved in the transfer of anions through the phospholipid bilayer. In the form of the overall amount of CO2 at the intracellular pH in the range between 7 and 8. So the protein is very important in the exchange of oxygen and CO2 in the body. The ultimate, the ultimate extensibility of erythrocyte membrane is characterized the ratio of the stiffness of the membrane to its elasticity. This parameter is expressed in terms of the maximum relative increase in the membrane area during erythrocyte lysis. The ultimate extensibility of the membrane is an important parameter for assessing the availability of the red blood cells and their resistance to osmotic lysis. The higher this parameter is, more than red blood cell is transformed. Here you can see that the sorbent aluminum oxide with polydimethylsiloxane reduces the number of active band 3 protein to the least extent, and the sorbent SUMS1, the first, cause a decrease in this indicator in red blood cells almost twice as compared to blood after surgery. And the value, the ultimate extensibility of the red blood cells membrane increase 3 times. This probably indicates of the integrity of the membrane and the launch of the cascade of reaction that lead to a change in the throughput of band 3 proteins. However, this change is lay between samples don't exceed the normal range measured on 20 donors. As a conclusion, this comparative pilot study of numerical parameters of physiochemical and biological properties of 2 different sorbents obtained on basis of aluminum oxide and various hydrophobic agents, organic silicon, polymer, and carbon. This study conducted by scanning flow cytometry on the effect of sorbents on the biometric parameters of red blood cells in the conditions of gamma perfusion didn't reveal a traumatic effect of sorbents on the parameters of red blood cells in comparison with the red blood cells of the original blood taken from a patient after total joint replacement surgery. But in the case of sorbent with a hydrophobic organic silicon polymer, there was a slight decrease in the hemoglobin content on the blood compared to the carbon mineral sorbent. The same sorbent reduces the number of active band 3 proteins to the least extent. The results obtained allow us to conclude that the use of scanning flow cytometry is a perspective method for monitoring of biometric parameters of peripheral erythrocytes and during surgical and blood transfusion exposure. And this method is much promising. I repeat, this is a pilot study and we continue our investigation. Here you can see our team from Institute of Kinetics and Combustion and from Research Institute of Clinical and Experimental Lymphology, particularly Pharmaceutical Technology Laboratory of Operative Lymphology and Lymphatic Education, and Connective Tissue Pathology Laboratory. Thank you for your attention. Finished.
Thank you, Vadim. Thank you, Vadim, so much for this interesting report. Unfortunately, we have no room for any questions. So the next speaker, can you please start your presentation?
Okay, just a second.
Thank you. Sorry, Vadim.
Thank
we
you very
have
much.
no any room.
Okay.
Sorry. Okay, the next presenter is Alexey Zhdanov from from medical equipment area of the Ural Federal University.
So can you hear me?
Yes.
Okay. So actually I have two report. So let me introduce myself. I'm research engineer of Ural Federal University and also medical equipment area chief of Ural Optical and Mechanical Plant. This both organization is located in Ekaterinburg city in Russia. So dear audience, today I present scientific and practical work about mixing device developed based on electric drives controlled by an electromechanical system. So the introduction focused on the equipment for transfusion therapy developed within framework of research grant. Equipment for transfusion therapy should have several conditions. On one hand, the transfusion therapy equipment should have automatic rocking of blood bag for uniform mixing with cryoprotectant. Cryoprotectant is a special substance which allow blood product to preserve the properties for a long time. On the other hand, equipment should have automatic control of the speed and volume of blood supply. And at the same time, equipment should have additional weighting and balancing of blood bag before centrifugation. Figure 2 shows an electrical diagram for connecting motor to an electric drive. Since the maximum weight of blood bag is usually equal to 1,000 grams, it is possible to use typical decisional speed of more than 30 revolutions per minute. In particular, drive system, Europe DC generator, gear motor, and the reversible motor drive with a maximum output current equal to 0.7 ampere. The motor are controlled by a signal from microcontroller. The showed approach of design integrated to strain gauge transducer. And the signal processing is given by STM32 microcontroller. To form the rectangular pulse, comparators have used. The comparator's input voltage level was set by the supply voltage, and the developed electronics were tested in experimental sample. The test result shows that the developed solution meets the requirements of the equipment for transfusion therapy. And in conclusion, I want to note that the transfusion therapy was included in the list of critical technologies in Russia, according to the decree the President of Russia in 2011. And use of clinically effective components of blood and its product in many medical fields is impossible without using blood service devices. So thank you for your attention, and I will be glad to answer your question, or I may continue to the next, if you wish, to the next report, which is connected to the first one. So what are you guys?
Okay, thank you so much. Does anybody have any questions?
Probably we will get more questions after next presentation. So it's really
okay. Proceed, please. Okay.
So it's all about the same thing. So here on the slide, you can see some devices. It's called blood mixer. And then I explain results. So my next report is about development of weight sensor based on strain gauge transducer. This topic is continuation of the previous report, and it contains the simulation results. So the blood mixer is a special device that allow weighting a blood bag during transfusion. When the blood bag is filled with the set mass, the device clipper the line and inform about the end of blood collection. That's how it works. The device price has a significant influence of weight sensor cost. Therefore, it is necessary to develop a weight sensor based on strain gauge transducer, which is really cheap, and many strain gauge transducer is manufactured in Russia. So here, from a mathematical point of view, the implementation is simple. The output frequency are equal to the sum and difference of the input signal frequencies, and the multiplication function is realized typical trigonometric equation that you can see on equation 1. So here, transistor Q1 and Q2 performs the implementation of capacitive 3-point socket self-oscillator. Quartz resonator in the socket are connected between the base and collector of transistor. And here on figure 3 shows output signal from Q1 and Q2 emitters of transistor. The signal from Q2, the green graph, has some parts, but this is not critical since the signal will be multiplied and filtered by a low-pass filter. And figure 4 shows the final signal after mathematical operation and low-pass filtering. So here on figure 5 shows the output of weighting sensor socket. The upper signal shows the information about weight of the sensor. So if the weight of blood bag is increasing during transfusion, the time of this pulse is getting longer. And the lower signal shows information about the temperature state of sensor. Why it's so? I mean, usual strain gauge transducer is consist of 2 transducer. The first one is for defining the mass signal, and the second about temperature. Why so? Usually the temperature has a really significant influence on output signal, especially the upper one. So the developed solution is already introduced into the work of Bimix blood mixer. Bimix blood mixer is transfusion therapy, which is manufactured by Lead Core company, which is located in Ekaterinburg city. Such company affiliated with the Belarus company, which is sell transfusion therapy equipment. And the validation of characteristics of weight sensor board is performed using the industrial oscilloscope. So that's all. Thank you for your attention, and I will be glad to answer your question. Thank you.
Thank you so much, Alexey. Has anybody any questions for these two connected, linked together lectures, reports? Okay. Thank you so much, Alexei. I personally have no any questions.
Thank you. Thank you so much.
Thank you so much. Thank you. Let me go to the next speaker, and I must apologize personally because I made a mistake with the schedule of our session. The next speaker is Vyacheslav Chudinov from Perm. Would you mind, Vyacheslav, to present your presentation? Vyacheslav?
Oh, sorry?
Oh, okay. Thank you so much, and sorry again for my mistake. Okay.
Thank you. Thank you.
Okay, please go on.
Dear colleagues, I will present a report on the topic to avoid a foreign body reaction for polyurethane implants modified by high energy ions. First, about problem of foreign body reaction. Inserting of all synthetic medical implants induced a complex of local cellular and tissue reaction to a foreign body, and you can see collagen shell as result, and for a body reaction, this collagen shell that's isolates the implant from tissue of organism, and the most important reasons of at its same cases, this isolation breaks functionality of implants, and for example, biomechanical disruption. The capsule can calcify it and increase the deformation of the implant. And the most important reasons of reoperation is capsular contractura. And the formation of connective tissue, for example, the formation of connective tissue capsule blocks and the electrical impulses of neuroprosthetic devices, for example, cochlear implants, exotic device for science, and it is necessary to increase, to apply voltage on microelectrodes, which can to break tissue and cells. For this reason, we need new medical materials without foreign body reaction. And one of the solutions is to create carbon coatings on implants. We synthesize in our laboratory polyurethanes, medical grade of polyurethanes, and the polyurethane sample was modified plasma immersion ion implantation in the University of Sydney in Australia. For modification, we used vacuum chamber. We regenerate plasma, then we applied high voltage and accelerate gases, ions, nitrogen gases, ions towards polyurethane. Then bombard the polyurethane surface and change the thin surface layer of polyurethane, while bulk layer no change. As a result, we get a carbon coating on polyurethane, and one of the moment we create our implanter in PEM to creation carbon coatings by a similar method. What about carbon structure in polyurethane? We can see on this picture Raman spectrum. The Raman spectrum of treated polyurethane, this red line, include two new peaks, new lines related to vibration or carbon structure upset like graphite. And where spectra shows the presence of free radicals in treated polyurethane, and for this red line for treated polyurethane, and thus active graphite-like layer was created on the polyurethane surface as a result of plasma ion implantation. we can read in more information in our article. This slide shows the result from microscopy. The topology of polyurethane film surface varies significantly after treatment. The surface of the treated polyurethane is characterized by a large number of folds, and surface deformation is the result of internal stress after ion plasma treatment. And these data show a significant increase in the elastic modulus of the carbon layer with increase in the treatment fluence. And one of the moments is very important moment. We have a hypothesis high activity free radicals in vitro facility of the modified polymer surface. Carbon layer provides a certain biological activity of the covalently attached proteins. We think it is the reasons of biocompatibility carbon coatings. In this article, we write about it. We study amount of attachment protein on untreated and treated in plasma treated polyurethane before and after wash up in SDS solution at the temperature 70 degrees. After washing, the amount of protein on untreated samples decreases, but the amount of protein on ion-treated samples remains higher. And thus the protein is attached covalently on ion-treated polyurethane. And you can read more information on this article, this publication. And what about animal trio? We have polyurethane samples, flat polyurethane samples. These polyurethane samples were implanted into 6 rats. It's a first experiment. 5 samples we implanted into each animal, untreated and treated by our implantation with different fluids. The duration of exposure of implants is animal was 1 and 5 months. From each implants and surrounding tissue, this section were prepared and stained with hematoxylin and eosine. We study thickness of the connective tissue membrane and density of distribution of granulocyte neutrophils and eosinophils in histological sample by microscopy. Picture show that connective tissue capsule that is a weight implant. The implant more around untreated than treated. The density of granulocyte was calculated as the ratio of the calculated absolute value of cells to calculate area of connective tissue capsule. We used analysis of variance NOVA and QK tests to identify intergroup differences. Average values of neutrophil density around plasma implants from 100 to 200 less than for untreated polyurethanes. Average value of eosinophil density around plasma treated are from 400 to 800% less than untreated polyurethanes, and this result is reliable according to the statistical test used. And after 1 month of exposure, the thickness of the collagen capsule around untreated implants is 50% bigger than around those treated with plasma with some fluids. And after 5 months of exposure, the thickness of the collagen capsule around untreated implants is from 70 to 100% greater than around those treated with plasma with some fluencies. And for the implant treated with plasma with maximal fluence, the higher average thickness value is explained by the more rigid carbon layer and large wave size. We suppose that even with small movements of tissue or an implant, there is a mechanical effects on the surrounding cells and tissue of the rigid road surface of the carbon layer. And about conclusion, increase in fluence causes an increase in the difference in height and size of waves, as well as increase in the rigidity of the surface layer. Histological analysis for tissue or certain polyton samples is animal material show a decrease in foreign body reaction for plasma treated implants compared to untreated ones. However, with an increase in the exposure time of the implant in living organisms, the effects of mechanical influence of the surrounding tissue on the rigid wave surface of the carbon layer is observed. The threshold values of treatment fluence reach the foreign body reaction increases with prolonged contact of the implant with organ tissue. And thank you for your attention.
Thank you so much, Vyacheslav. It was very interesting. Does anybody have any questions? The lecture was very
I have a question.
Okay.
Скажите, пожалуйста, вы вот смотрели, как приживается имплант, и смотрели клетки, в основном я посмотрел, вы показали эозинофилы и нейтрофилы, те, которые характеризуют как бы воспалительную реакцию. А вот как развивается соединительная ткань вокруг имплантов за счет фибробластов, вот это вы не смотрели, не учитывали?
Эта оценка была качественная. Скажем так, качественно оценили. Мы не использовали какие-то CG-маркеры.
Ну, вы гистологию там
приводите. I'm sorry, colleagues. I'm really sorry. I must ask you to continue in English because... Okay. So can you please use English?
We don't use CG markers. We use histology, easy method of histology.
Okay.
Thank you so much. Any other question? So if no, we have almost 20 minutes before the next report. So if you don't mind, Vyacheslav, may I ask you for some issues regarding your report? And can you please my first question is, you said that polyurethane sample has carbon molecules, and you said that there is some kind of protein adhesion to the surface.
Yes.
So is it possible, in principle, use specially produced it would be actually not implants, but for example, specially produced rods from this material to use there for cell adhesion, cell attachment, I mean isolated cells. Is it possible in principle?
Cell attachment, sorry, understand.
For example, I can have an isolated cell, for example, cardiomyocyte or muscle cell, and then I can use the rod-shaped implants or rod-shaped things produced from the same material to attach to this cell and to apply some kind of mechanical perturbation to this cell. Because, as you said, this material can effectively adhere to the cell surface, to the proteins, and so on. I can repeat my question. We have enough room in our time to discuss. So my interest. Okay.
Maybe you're right.
Okay, this material used for implants can effectively adhere to the biological tissue, isn't it?
Yes.
Okay, so in principle, if I put a single cell, isolated cell, it doesn't matter each cell, red cell or red blood cell or muscle cell or cardiomyocyte, if I put the cell to the material, it can adhere to this material quite effectively due to the
before implantation. Before implantation.
Not any implantation. It's just the interaction between material and cell membrane. Is it possible to adhere?
It's possible.
Okay, so then if I prepare very thin rods from this material to use there to attach to a single cell, for example, from different sides of this cell, I can provide enough adhesion of this material to this cell membrane, and then I can make some mechanical changes in.
Okay.
It's just the mechanical attachment of a single cell. Is it possible in principle?
We don't try this.
Anyway, what is the actual physical forces provided, which provides adhesion of living tissue, biological tissue to this surface? Is it electrostatic force?
It's provide free radical in localized on carbon structure in carbon structure, and I show a pair spectra. A pair spectra indicate this radical, and this film we studied from after treatment 2.5 months. 2.5 months, we save film, treated film, and after that we register spectra, and we have a signal of free radical on this structure. And we said is free radical provide active carbon layer, and we can attachment of protein and cells, and provide biocompatibility of material.
So it's rather biochemical interaction, not electrostatic.
Electrostatic
as well.
Maybe and electrostatic, but we don't investigate this. But it may be free radical and electrostatic. Maybe.
Okay. Thank you. And also, I would like to ask about the whisker plots you presented. Unfortunately, I can't say the number of your slide where you shown the differences between untreated samples and samples treated with different extent of fluence. Not this one, the next one, the next one. Oh, this one. No, previous. Can you?
Yes.
Okay. So here, as I can see, there is no any specific or important difference between the extent of fluence, because I see there is only statistical difference between untreated and any of treated.
Yeah.
Does it mean that, and also you show here the measures of immune response of the tissue, and does it mean that actually it's really not important for this sample to be treated more than at some lower limit? I mean, if you achieve a desired limit of preparation of your sample, it's not important to provide more treatment on this sample with fluence, I mean. So because as I understand your report, the fluence actually changed the structure, the surface structure of the sample, and you investigated whether the change in the waves, the composition of waves, the height of these waves can affect the implantability of this material. So, and here I can see that this pretreatment doesn't induce any change in the immune response. So possibly for mechanical or for connective tissue and so on, but not for the immune response. Okay, the geometry of the implant doesn't affect the immune response. Okay, is it true?
Yeah.
Not?
In next slide, I say about negative reaction for long exposition in organism. For the more rigid and more rigid surface, its surface we create by the maximum fluences, and it is negative. We don't use some maximal of fluences. We need medium fluencies or small fluencies for the provide biocompatibility material. But I don't show don't present about activity of fluencies, activity of activity surface in the different fluencies, and we optimized safe activity of carbon layer and not use very bigger fluence. I don't know. Understand? You understand me? No.
Okay. Okay, I see. But possibly I'm wrong. But here you show just the thickness of connective tissue, which can grow due to the traumatic damage of the tissue during implantation. It doesn't relate directly to the immune response. I mean, the immune response is the same because you transferred the foreign body, foreign material into the biological tissue. Okay, during the implantation, you inevitably damage surrounding cells, and this create this zone of traumatic damaging. Creates actually the potential zone of connective tissue around. It doesn't relate to the immune response. So your previous whiskey plots show that immune response is not dependent on the fluence of the material. So there is difference between traumatic damage and immune response to the foreign material inserted to biological tissue, I guess, especially in the case if you in some way in some way prevent any how can I say any bacterial or infectious compositions on your sample. So if you, I'm sorry, I forget this word in English, if you make pretreatment to release any biologically active foreign objects from the material before implantation. Do you understand me?
It's question about bacterial.
No,
No.
actually, okay. Just let me explain with more usual words. For example, if you want to prevent the transferring any bacterial or infectious substance during surgical operation and so on, you can pretreat your material with heating or special liquids which remove the bacterial or any other components from the instrument, from the samples, I don't know, scissors, syringes, and so on. I forget this word in English, I'm sorry. This is the quite common term when you prepare your material before any surgical treatment to prevent transferring any potentially dangerous components to the tissue. Antiseptica. Okay.
I think.
Okay. So if you, I'm sorry, if you pretreat your material before implantation, and if you implant the material being pretreated correctly in the terms of aseptic preparation, then you could expect no any significant immune response, just the local immune response, okay, because the traumatical damaging of the surrounding tissue, and just due to release to the surrounding area some intracellular substances which are autoimmune components, local autoimmune components. So in case if you implant dirty material, bacterially dirty material, or for example with some kind of infection or some kind of foreign biological components, then you can expect a system immune response during the implantation, and this is completely different from the first situation. So if you prepare your implants with purely aseptic conditions, you just could expect the local immune response, which is not dependent on the mechanical and physical properties of your implant, isn't it? It's just to return to the previous plot, we discussed it with no any difference between no, not this one, yeah. No any difference between the local immune response. Okay, so thank you so much. I'm sorry for this very extensive discussion. Does anybody have any question? Because we have another 5 minutes to discuss something more. I thank you so much, Vyacheslav, for this very interesting discussion, and I see quite potential steps for using this material. For me personally, it's interesting to use this kind of material for single cell manipulation. Possibly it would be very interesting.
Thank you.
Okay, thank you so much. If you don't mind, we can go to the next speaker, Alisa Podgurskaya from Moscow. Can you please start your?
Yes. Good afternoon. Can you hear me?
Yes. You have 4 minutes more plus your time, so you can use this
time. Do you see my presentation?
Yes, yes.
Good afternoon, dear colleagues. Today I present the study phengyramine arithmeticity testing using monolayers of human iPSC-derived cardiomyocytes. Firstly, I will explain the in vitro arithmeticity testing that was developed before, and then move to the phengyramine arithmeticity. Developing in vitro tests that could predict arrhythmias are important for different classes of drugs, and as the heart disorders are still the first reason of death worldwide. Most of the pharmacologically used cardiotoxicity tests are based on ion channel screening using isolated cells. However, it is highly important to consider the interactions between the cells. There are different approaches to assess complex cardiotoxicity using hard slide technology, cell clusters, cell monolayers, three-dimensional microtissues. We use cardiomyocyte monolayers of patient-specific iPS cells. After cardiomyocytes characterization by immunocytochemistry and achieving the same electrophysiological properties as in an adult tissue, we conduct optical mapping with calcium-dependent fluorescent dye. Also, we use cardiomyocytes from neonatal rats. We measure 3 values: conduction velocity, maximum capture rate, and the number of re-entry occasions. Validation of the test was done with erythromycin that directly blocks IKR without affecting IKS, IK1 fast sodium currents and connexins. The second drug is cyclophosphamide that do not affect ion channels and connexins, but it disrupts cells integrity. Diphenhydramine blocks potassium channels and connexin 43. Let's move to the results. With erythromycin, we found no changes of conduction velocity. The maximum capture rate decreased up to 40% under 45 micromoles of the drug, and re-entry occurrence was in 33% cases. There are activation maps on the right panel that illustrate the re-entry formation on the standard linear obstacle in the monolayer. We use it in order to level down the differences in homogeneity of obtained monolayers. All the mentioned effects were found in human cardiomyocyte monolayers, but not on the rat test model. These results are consistent with IKR current blocking effect of erythromycin without affecting IKS current. IKS current has small density in the red cells. With cyclophosphamide, conduction velocity was not changed. Maximum capture rate also decreased up to 32%, and no re-entry was observed. These effects were also found in red cardiomyocytes. Maximum capture rate itself, maximum capture rate decrease could be induced either by disruption of the ion channels or intercellular contact functioning, or damage to the cell's structure's integrity. No effect of cyclophosphamide was found on voltage-gated ion channels level of connexin 43. Conduction changes that we found were caused by alpha-actinin disruption. It was detected both in human cardiomyocytes, that is showed on the left panel, and also we found it in neonatal ventricular cardiomyocytes on the right panel. Then we calculated C value for human cardiomyocytes using fast Fourier transform. It allows to analyze the distribution of structures orientation presented in the input image. F-actin parallel bundles cross-linked by alpha-actinin, and it appears in distribution charts as peak values compared to the baseline intensity. And damage of cellular structures lead to a decrease in the peak intensities and an increase in baseline intensity. And we've calculated this contrast and indicated it by C value. So C value was reduced more than 2 times after 30 minutes exposure to cyclophosphamide. With difenhydramine, conduction velocity decreased up to 65%, and maximum capture rate decreased up to 60%. Re-entry were found in 50% cases. Conduction velocity decrease is connected both with fast sodium channel blockage and gap junctions blockage under difenhydramine. Maximum capture rate decrease caused mainly by potassium channel blockage. Re-entry formation evidences mainly the ion channels blockage caused by difenhydramine. Let's move to the conclusions. I will sum up all that I said. Conduction velocity reduction indicate fast sodium channels and gap junctions blockage. Here we showed it for difenhydramine. Also in our previous works, we demonstrated how conduction velocity reduction is connected with gap junction inhibition by the other chemical compounds. Maximum capture rate reduction is connected mainly with potassium channels blockage, but also with the mediated influence. The results obtained allow us to conclude that arithmeticity testing works both with ion channel blockers and drugs with indirect mechanism of action. Thank you for your attention. We thanks Ranzikian's lab for providing iPS cells and Russian Foundation for basic research. I'm ready to answer the questions.
Thank you so much. Has anybody any question? Dear colleagues, we have 10 minutes to discuss this very interesting report. Colleagues? Okay, if you don't mind, Alisa, can you please show me the slide with confocal images?
Yes.
This is number 6.
Do you hear me normally?
Yeah, with some noise, background noise from your side, but it's not annoying. Okay.
Okay.
Okay. So as I can see here, are there living myocytes?
Excuse me? Which myocytes?
Are they living? This is
living.
live myocytes, living.
No,
living,
they are not
yeah.
living. They are fixed in formaldehyd, and
okay.
then we died for F-actin and
Okay.
alpha-actin.
Okay. I just wanted to ask about this because I, by mistake, I thought that these myocytes are living. And my question was about alpha-actinin. I know that there are some methodological approach to stain alpha-actinin in living tissue to see the composition of myofibrils and sarcomeres in the living cells. It would be very interesting to see the same situation in the living beating, maybe not beating, okay, but with electrical propagation of the signal of the electrical wave. It would be very interesting to see the same under physiological conditions, not in vitro state, actually. Okay, so
thank you.
Okay.
So here we see the cells after. We see the cells in control, and after 5 minutes exposure to cyclophosphamide, and after that the cells were fixed, but they're not living, unfortunately.
Okay, but anyway, the exposure was under living conditions, and then you fixed the tissue and stained the tissue. Okay, yeah.
Yes. Yes.
Okay, thank you so much.
And all the optical mapping experiments were also in the living condition. Optical mapping advantage is that we can see the propagation in the real time, and we can manipulate with the cardiac tissue as we wish for hours.
Okay. Okay. By the way, can you go to the slide number 4 to show this? 4 or 5? It doesn't matter. Okay. Because we have 8 more minutes to discuss, may I ask another question about the optical mapping? I can see here that the map is not actually with quite high spatial resolution. Because I can see here the spatial resolution was, I can't say exactly, but it seems the camera, the sensor, the CCD camera here was maybe 40 pixels to 40 pixels, isn't it?
Oh, it's not, it's a little bit different.
Okay.
Here I've built activation maps in Wolfram Mathematica with concrete radius of approximation. So these maps are not exactly the propagation that we see at optical mapping. Here you can see 6 time areas, and at each time area, activation map indicates where the excitation wave is. So
yes, but anyway, each map was obtained. Yeah.
I can choose a lesser time points.
Okay, but anyway, each map was obtained by optical device, isn't it? Is it optical acquisition?
After optical mapping, we obtain video, and video.
Yeah, I see. Yeah. Okay, so
yes, this data is from optical mapping, but it is.
It's just the wave from time-related wave from wavefront of the spreading of the excitation.
Yes.
Okay. Then I would like to ask you about the optical resolution of your system. How much is it?
Resolution here was 512 per
512 to 512.
Yes, and 30 frames per second. Oh, not 130 frames per second.
Sorry. How many frames per second?
130.
40?
130.
140.
Yes.
Oh, okay. 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 What do you think about the smaller heart? For example, red heart or mouse heart. The faster the propagation of the electrical impulse, and then you can increase your FPS of your device to follow the correct propagation wave, the front of the propagation wave. So if you, if you use red heart, it's possibly enough to have 140 FPS. But possibly if you will use a smaller heart, you can increase your temporal resolution. So you can increase FPS to follow correctly to the wave. Because otherwise you will get more distance between two consecutive waveforms. Okay. Okay, it doesn't matter. Thank you so much. Does anybody have one more question or two questions? We have 3 minutes. Okay, just let me ask my final question to you. Because we have a little room before the next speaker. Okay, which dye do you use for potential measurement? The membrane staining and so on, the electrical optical mapping?
Here is calcium-dependent dye, so we see calcium waves.
Calcium-dependent?
Calcium, only calcium, yes.
Okay, okay. In this case, did you block the mechanical response in this situation? Or it was beaten tissue?
We use pacing stimulation in order to get electromechanical response.
Yes, but you should electrically induce the propagation, so you should stimulate your tissue.
We've done it.
Okay, so you stimulate it by external electrical device, and then the tissue was stained by a calcium indicator?
Yes.
Not potential? Okay.
Not potential.
So
yes.
It was not electrical mapping, it was calcium mapping?
Yes.
Oh, okay, okay, I see here. Okay. Which dye did you use for calcium measurement?
Fluor 4 AM. Fluor 4
okay.
AM.
Yeah, I see. Okay, thank you so much. It was very interesting to discuss this report. So, thank you. We can go to the next speaker, Anatoly Shatrov from Kirov.
Yes.
Good day. Can you please proceed your presentation? Thank you.
Yes, yes. Demonstration of ekran.
Уже ты сюда пришел, на молодежь.
Может быть, мы можем
Good evening, good morning, dear colleagues. My presentation is visualization of blood erythrocytes sedimentation process on transitive line erythrocytes plasma.
Unfortunately, we can see your presentation.
Well, content of presentation. One, actuality of the present problem for the diagnostic signification of the standard erythrocyte sedimentation rate, ESR test. Review of publication of mathematical model on experimental methods for express form ESI. Device for visualization and graphic recording of the erythrocyte sedimentation process. Result of visualization information of erythrocyte sedimentation process of red blood. Conclusion. Actuality of the presented problem is widespread usage of laboratory blood tests. The purpose of this work is to prove specific character of this analysis. Process is registered by devices for visualization of blood cells deposition with high degree of space and time resolution in real time. The research of dynamics changing of border and transitional layer of erythrocyte plasma character gives change to considerably on enlarge the content of diagnostic indicator of erythrocyte sedimentation rate. This research presents in the data obtained during the process of visualization of erythrocyte sedimentation rate in a pilot study in order to improve their diagnostic value. To understand the observed process and trend formation mechanisms, the comparative analysis of humans or animals, both healthy and with individual physiological characteristics, the presence of disease of exogenous influence was carried out. Review of publication. Soviet Russian research in problems of blood erythrocyte sedimentation process, mathematical modeling, and experimental research. Chizhevsky, Novosibirsk, Voyeikov and Moskov, Ananyev, Aristov, Tomsk, Regiler, Losev, Moscow, Berdnikov, Kazan. Foreign research, King, Copley, United States, Schmidt, Schoenwein, Klose, Rebush, Germany, Reinhardt, Swiss, Miralit, Haran, Tateshi, Maeda, Japan, Yamaikina, Vazyan, Ukraine. Description of experiments. The students were considered with using the blood of humans and animals, rats of white line, Vista males weighing 200 grams. Human blood sample was carried out in special laboratory by standard finger prick method. Capillary pre-washed the three substituted sodium and citrate dilution 1:4. The blood in experimental animals was sampled from the jugular vein into a syringe with sodium citrate. In the basis of ESA study lays the micromethod modified by Panchenkov. For the research, two laboratory devices working simultaneously were used. The device for automatic recording of erythrocyte sedimentation process and the device for visualization of process in the transitional layer plasma erythrocytes of setting blood. A device for experiments, a block diagram. 1. Computer. 2. Special capillary with blood for microshooting of sedimentation process. 3. Digital high resolution camera eyepiece, 4 controller, 5 electric carriage, 6 light source indicators lamp, 7 power sampling of control electric carriage, 8 heat filter, filter hills, and lens for warming parallel light flux. Research results of ESR indicator. In this figure, visualize information of erythrocyte sedimentation process of the red blood animals where is a state of physical norm. Oscillation is regular. Research results of ESR indicator found by preparates number 4 and number 6 energy protectors based in acid with stimulate the body after putting into a stressful condition. They are irregular and non-linear changes in nature of oscillation. Oscillation is different in this figure. This figure visualizes information of erythrocyte sedimentation in red blood with experimental toxic hepatitis. In this figure, oscillation is non-regular and non-linear. This figure shows data of layer thickness changes during the stressful condition of the animal. Analysis were taken after a swimming rat begins to show sensory excitation. Oscillations in this case, in spite of the apparent regularity in the behavior of average value, demonstrate a significant deviation at moments of time. In this figure, show two locations of oscillation. Is a slide video information obtained using a device for visualizing process in the boundary layer plasma erythrocytes offsetting blood. 6 frames in 5 seconds. Conclusion. The parameters of the cures of erythrocyte sedimentation and changes in the boundary layer with various effects in animals have significant differences. The present method makes it possible not only to detect quantitative changes in the ESR, but also established a high quality nature. The obtained data suggests that this method is highly sensitive and might be used in basic and applied research. In fundamental aspects, is important to study the blood factors affecting the nature in the dynamics of the erythrocyte sedimentation, boundary, and phenomena in transitional layer. The point of this aspect is in terms of mechanics, two factors affect the setting blood cells. They are for force of gravity of combination with the buoyancy forces resulting in aggregation of blood particles. In the transition layer, it regards as a dynamic system. The presence of the small parameter, the ratio of typical time, my regards for features of the oscillation atom on transition layer tranquils. Presentation finished. Thank you very much.
Thank you so much. Does anybody have any question? We have 3 minutes more to discuss this report. Colleagues, please. Okay, if you don't mind, I have one short question, or rather wondering about the oscillatory behavior of the sedimentation process. I have never know about this, so it was very surprising for me to know that this process is not regular. Okay, it would be non-linear or linear, but to see very prominent periods of oscillations was very interesting for me. There is any explanation for this phenomenon?
Excuse me, please, question in Russian.
Окей, я задам тот же самый вопрос на русском языке. Я был очень удивлен, я просто не разбираюсь в этой теме, но был удивлен, узнав, что существуют периоды осцилляций на кривой осаждения эритроцитов. Существуют какие-то гипотезы, объясняющие этот феномен?
Основная гипотеза, которую мы здесь пытаемся донести, заключается в том, что в упрощенном варианте сложная динамическая система при наличии малого параметра, в данном случае малым параметром является отношение времени осаждения под действием силы тяжести и формообразования вот этих вот комочков эритроцитов, так называемых форменных элементов крови. И наличие малого параметра в сложных системах такого вида приводит к колебательным процессам. В частности, появляется так называемый предельный цикл. И вот в данном случае предельный цикл для нормального состояния организма является устойчивым и регулярным. В случае каких-либо аномалий, в частности, подтравливали тетрахлорметаном крыс, появляются вот эти вот всплески относительно большой толщины слоя, переходного слоя, эритроциты плазмы. Причем, конечно, для того, чтобы сделать такой тест, как говорят в врачебной практике, специфическим, необходимо иметь большую статистику. По крысам, по животным эта статистика в принципе есть. И она приводит к тому, что в случае аномальных поведений животных мы наблюдаем не один предельный цикл, а несколько. Часть из них является вложенными предельными циклами, а в случае, когда предшествует в виде отравления тетрахлорметаном, и после этого наступает уже, собственно говоря, смерть животного, там мы наблюдаем разрушение всех предельных циклов, и это было видно на нерегулярном характере изменений толщины слоя.
Я понял. Спасибо большое. К сожалению, у нас настало время следующего доклада. Большое вам спасибо.
Да, thank you very much. Спасибо.
Thank you so much. The last report for today will be presented by Mikhail Volkov from Yekaterinburg. Can you please, Mikhail, start your presentation?
Good afternoon to everyone.
Good afternoon.
Are you hear me? Do you hear me?
Yeah.
Very good. Так, as I understood, you see the presentation, isn't it?
Yes, we see.
Okay, let's start. Let me present our subject. It's not related to mathematical modeling or computer bioscience, but relating to bioinformatics and little bit explain genetic mechanisms in real clinical cases. And I would like to present you an unusual family case of severe combined immunodeficiency with adenosine deaminase deficiency in our patients in Sverdlovsk region. It's a very big collegial work, and main researchers, of course, is clinical doctors who was real attending doctor of that children. What is a primary immunodeficiency? I can explain you from the position of more understandable for non-doctors. The new name is inborn errors of immunity. It's a very large group of disease, already more than 400 disease, which include and high susceptibility to infection and autoimmunity, autoinflammation. Autoinflammation, usually you can know it in the real life as temperature without any reason, and malignancy, I mean cancers and so on. And it's very complicated pathology that could be on the doctor meeting, in the clinical, in every physician. Not meaning that only immunologists meet them. Absolutely not. Usually other doctors meet that patients and don't know what to do with them. And the rapid growth of the number explained easy. We start using NGS technology, new genome sequencing technology. And if, for example, in 2009, it was only 200 diagnoses, today it's already more than 430. And you see the graph of rates in Sverdlovsk registry, I mean Sverdlovsk region in total is 6 million people. And the rate of this rare disease rising up every day because we developed diagnostics in Yekaterinburg and start do a genetic analysis in Moscow. Of course in Yekaterinburg too, but not everywhere. What is other deficiency which I speak about? Is ADA is a housekeeping enzyme, very important gene which work in every organs and systems in the human body. And we can explain that when we see the broken housekeeping, when housekeeping gene is broken, it very damages for body and very heavy clinical picture, very severe symptoms. For immune system, the main is lymphotoxicity, and lymphotoxicity leads to severe combined immunodeficiency. But when we're expecting such big list of symptoms, we sometimes can't recognize light form of this pathology in children's on the attending, I mean in the clinic, in ambulance and so on. You see that it's not just immune pathology like respiratory infection, mycobacterium or viruses. It's already, we know that is a serious neurological and development problems, serious problems in skeletal, I mean skeletal abnormalities, especially with fingers. Is a toxicity effect which lead to DNA reduced DNA synthesis and repairing. It means that it's very high risk of malignancy. And of course, the first symptoms which every child developed is neutropenia and eosinophilia. Of course, all these symptoms, like in the school book, not 100% of the symptoms in every patient. Every patient different. And I would like to introduce this case because it was very interesting for us. First brother, older brother, was born healthy. He was vaccinated. But from 2 months of life, he developed recurrent ductal cystitis. These are infections of eyes. And then in the common blood analysis, doctors found eosinophilia. High eosinophilia, not so super high, but higher than normal. And they, after 1 month, he developed recurrent eosinophilic pneumonia. I mean, it's not usual pneumonia. It's like allergic pneumonia, but without neutrophils, but with eosinophiles. It was strange. Doctors took chromosomes for analysis and didn't found no chromosomal aberration, but they didn't genetic testing. In 1 year, the pneumonia was so severe that he started receive immunosuppressive therapy, and he started to be healthy. It was good treatment, and they did another chromosome research and found some chromosome aberrations. Not in every cell, but some cell in bone marrow was already damaged. After 5 years, he developed again eosinophilic syndrome. And in this case, in the bone marrow analysis, doctors found the symptoms of leukemia. It was acute myeloid leukemia, and after 1 year, he got hematopoietic stem cell transplantation from actually identical unrelated donor. But unfortunately, he was already suffer from a very wide spectrum of viruses. And the chronic infections as background for donor transplantation is a very bad case. We hadn't any possible ways to save his life, and after 2 years after transplantation, he died. His brother was born after 3 years of birth date of brother, three years different, but he developed a little bit another clinical picture. And it was, of course, with leukopenia, neutropenia, and eosinophilia, but also hypogammaglobulinemia. It means that immunoglobulin G was on low levels. It was unusual. It was different from his brother, but doctors was already in big attention to this family, and they found pneumonia early and start do intravenous immunoglobulins at once. But it helped, and pneumonia was finished very soon. Then he developed bacterial infection every 3-4 months. And it was for all his young life in kindergarten. And when he goes to school, he already suffered from respiratory infections at autumn-winter period and neuroorthopedic syndrome. So he started developed neuroorthopedic damages. It was dangerous already. It means the nervous system was affected. And this period, doctors found immunologist, and it was today he have constant intravenous immunoglobulins replacement therapy every month. It saved life therapy, and many of our patients use it. And only in this period, in 7 years after died of his brother, doctors did whole exome sequencing. And what we found? We found 2 pathogenetic mutations in the 1 gene. Sometimes it happened, but it was not so usual for this gene, first of all. And second, all mutations was pathogenic, but nobody of parents had the symptoms of this disease. First mutation was described already in journals. And using databases of pathogenicity and prevalence in the population, we understood that this is a rare mutation. So it could be not benign, it could be pathology. Second mutation was the same, rare and lead to pathology. And when we did Sanger sequencing for all members of family, I mean, we took material from died brother, we took blood from mother and father, we found that and first and second brothers had 2 mutations from both parents, but every parent have one of the mutations and have no any symptoms. How to explain this? First of all, we should understand that not every mutation could be pathological. It's easy to understand. Yes, for example, on the right picture, we see that in 2 different positions could be mutation, which lead to changing of third dimension structure of protein. But if the region of protein which have no any strict function, any dangerous if it will delicious, the patient will have no symptoms. For example, mother have only 1 mutation, and the protein function we can think maybe not so good as 100% of full function, but maybe 90%. Father, for example, 70%, and it's okay for covering the symptoms. But children doesn't have mathematical sum. They have 2 alleles of these genes with pathological structure, and they have no ability to be healthy. And when we researched the pedigree, we understood that even their grandparents, which often we found in grandparents the symptoms, even then great parents hadn't that symptoms. Only one of grandparents died of melanoma. But symptoms was really different. And what can I say? That is really very important, first of all, to start genetic diagnostic as much as possible early, and treatment too. Because today we have new screening methods when we, even without deep mining, deep thinking about the problem, just do a screening. And when we find some differences, some variables, we send this patient for deep research and save them life, because then early we do transplantation of stem cells, then healthy they become. Thank you for your attention.
Thank you so much. Dear colleagues, do you have any questions? Okay, it was interesting to know about this very, I hope, very rare case. If you don't mind, Mikhail, can I ask a very short question? It is about one of the first slides, maybe first or second or third. No, third, number 3, yeah. Number 3.
3? Mm-hmm.
Yeah. As I can see from this plot, the number of diseases in the Sverdlovsk region is about 600, isn't it?
Yes.
Okay, but if I will see to the right part of this this slide, I can read here the worldwide cases, isn't it?
I'm sorry, it's not so explainable.
It's just
not
okay.
It's diagnosis, different diagnosis.
Okay.
Left.
Okay.
Maybe
so
20 diagnoses.
Actually, okay, 406 in 2020. It's just 406 different diseases.
Yes.
Genetic disease. Okay, and in the case of Sverdlovsk region, it is the total number of diagnosed diseases.
Patients. Diagnosed patients.
Okay. Thank you. Did you mean that a single patient could have several diseases, genetic diseases?
Yes, it's approximately 30 different diagnoses.
Okay. Thank you so much.
You're welcome.
Thank you so much. If dear colleagues, if you have no any questions, I would like to invite you after 13-minute break. I would like to invite you to the plenary session with our foreign speakers. I believe it would be very interesting to hear these lectures, to listen to these lectures. So thank you so much. It was very interesting for me and for you as well, I guess. And see you next time on the next conference. Thank you so much.
Коллеги, меня видно, слышно? Здравствуйте. Василий Ильич, не слышу вас. Олег.
Добрый день. Да, перерыв пока.
Ага, хорошо. Олег, Василий Ильич, если не возражаете, я вступительное слово скажу, а потом уже Олегу передам все эти бразды председательства, окей?
Да, хотел сказать, просто уточнить, у нас же получается по расписанию сперва выступает Александр Николаевич и потом наши спикеры. Я правильно понимаю?
Да, все правильно. Ну, я же могу перед всей пленарной?
Конечно, да, конечно, хорошо.
Хорошо. Олег, еще такая просьба, ты же будешь всех троих представлять, правильно я понимаю? Вот, и первого тоже.
Вот я как раз хотел спросить, да, первого спикера, конечно, наверное, мне разумно представлять, но я думаю, он основной председатель.
Почему?
Разве не он следующий?
Нет, ну я думаю, что... Почему? Я думаю, что ты лучше представляешь.
Хорошо, как скажете.
Вот, нет, ну сам себя как-то неудобно.
Нет, его-то я как раз представлю, а следующий...
Мне кажется, что это же наши люди, что он их не знает, ты про них хоть что-нибудь скажи. Кто, откуда, что мы там совместный проект с Панфиловым. Ну, хочешь, я представлю?
Как вам удобней.
Давай, представляй. Ну,
хорошо.
Только разверните.
Поразверните.
Да.
Так, давайте тогда немножко отрежиссируем. Ну, просто и Александр...
Вступительное слово про наши совместные конференции, там про то, 5-10, вот, и все, и передам тебе слово, ты начнешь представлять людей.
Да, я просто к тому, что я знаю о вашей очень плотной коллаборации с Александром Панфиловым, но в меньшей степени
А у нас с
знаю...
Игорем как таковой нет, просто нужно сказать, что вот Игорь приезжал с лекциями в Екатеринбург, очень большой интерес вызвали его исследования. Ну, вот что-то.
Хорошо.
Окей?
Хорошо. Хорошо.
Смутил тут меня совершенно, что-то Саша не с нами, он, здесь, да. Ефимов прислал письмо, что он там все время перепутал, сказал запишет. Саша сказал не запишет. В общем, в итоге записи я не получила, а будет он или не будет, я тоже теперь уже не понимаю. Но будем надеяться, что будет.
Последний раз писал, что будет.
Ну, будем надеяться. Да. Так, мне, наверное, надо будет надеть гарнитуру, а то опять у меня будет фонить. Вчера фонила без гарнитуры. Так, нормально у меня голова по центру более-менее торчит?
Да, по центру.
Да,
и, кстати, ваш звук не фонит, совершенно нормально все слышно. Можно...
Ну, я не знаю, может быть, когда запустится в эфир трансляция. Вчера люди говорили, что когда без гарнитуры, то фон шел.
Ясно.
А мне надо еще, знаете, что сделать? Мне нужно отключить звук на трансляции. Сейчас я отключу.
Ольга Эдуардовна, по поводу аффилиации Игоря, я ее позаимствовал ровно так, как это опубликовано на сайте нашей конференции.
То есть ты не сверялся, да?
Нет, честно говоря, нет.
Все правильно, декан факультета биомедицинской инженерии университета Джорджа Вашингтона.
Здравствуйте, Александр Николаевич.
Может, про нашу договоренность тоже Александру Николаевичу сказать? Александр Николаевич, здравствуйте, Ольга Эдуардовна.
Здравствуйте, Александр Николаевич. Я Олег Лукин.
Так, Александр Николаевич пока звук не подключил, сейчас, видимо, подключится.
Кто-нибудь скажите что-нибудь, чтобы я звук настроила на наушниках, пожалуйста.
Эта конференция очень замечательная, очень интересные доклады.
Все хорошо, спасибо.
Спасибо.
Да, фидбэков очень много, и все очень-очень положительные. Так что всем большое спасибо.
Спасибо.
Сейчас я Сашу спрошу, он вообще с нами, нет?
Я здесь, Ольга Эдуардовна.
Он вообще-то всегда заранее выходит. Саша, Панфилов подтвердил, что он у тебя все получил?
Вроде бы, да.
Саш.
Сейчас проверю.
Саша Курсанов.
Да, сейчас проверю, Ольга Эдуардовна.
Саша Курсанов.
Он здесь, он вам отвечает, он слышит.
У меня в наушнике, господи. Подтвердил, да?
Да, он что-то присылал.
Не поняла.
Сейчас проверю еще раз. Вроде присылал.
Как так вроде-то? Мне не отвечает сейчас. Не знаю.
Александр Николаевич, нас слышно? Вам слышно нас? Спасибо большое. Александр Николаевич пишет в чате, ну, наверное, все прочитали, что звук, проблемы со звуком на той стороне.
Так, у вас звук не идет, в смысле, у вас микрофон не работает, вам нужно выбрать звук. Вот, Александр Николаевич, вам нужно, вот где микрофон в левом нижнем углу, выбрать источник звука.
Трансляция.
Ой, по-моему, ой, Александр Викторович, привет. Я уже немножко волнуюсь, потому что я тебе на WhatsApp написала.
Я не видел.
Ага, и позвонила, да. Все, спасибо большое, Саша.
Хорошо.
Да, спасибо, Танечка. Я уже Таню напрягла. Да.
Что,
я сейчас ему скажу.
Хорошо, но я тогда здесь, когда надо, вы мне скажите.
Да, все, конечно, Саша.
Ладно. А ты будешь председатель или кто?
Ну, я сначала сделаю первую такую вводную, а потом Олег Лукин будет уже представлять.
Александр Викторович, здравствуйте. Да, я здесь.
Да.
Привет.
Здравствуйте.
Ладно, хорошо, ну тогда звук отключаю, чтобы не фонить там.
Давай, хорошо, Саша, спасибо огромное. Как у нас у Александра Николаевича дела? Он же у нас первый доклад. Александр Николаевич, вы нас слышите? Видите? Почему-то вот тут камера, вас не видно.
Насколько я понял, Александр Николаевич, он в чате написал, нас-то он слышит, а вот с микрофоном на его стороне могут быть какие-то сложности.
Ну вот у него, когда смотришь
вот сейчас
участник...
сообщение. Вот сейчас сообщение, он пишет, что...
Я видела.
Да, так что...
А что будем делать?
Как вариант, ну, если... Ну, у нас еще 4 минуты, в общем-то.
Нет, ну давайте какой-то запасной вариант. Если что, поменяем, что ли, последовательность?
Я думаю, да, это единственный вариант.
Саш, ты не возражаешь? Саш Панфилов, не возражаешь?
Сейчас.
Угу.
Алло, да?
Да. У нас, потому что, видишь, первый докладчик, Александр Николаевич, у него какие-то проблемы со звуком.
Да, я дозвонился до Новосибирска. Сейчас они перезагружаются. Видимо, что-то не работает микрофон. То есть он нас прекрасно слышит, а мы его нет. Что-то сейчас они
мы его и не видим, хотя у него вроде камера горит.
Да, видимо, какие-то проблемы с подключением, надо перезагрузиться.
А можно уточнить, кстати, мы вот снизу по времени, мы же не лимитированы. Я правильно понимаю, после 15:00 в потоке ничего другого не запланировано?
Да ты что, там 2 сессии, Олег, о чем ты говоришь, там прямо следом идет.
Я прошу прощения.
Нет, у нас очень жесткий регламент. Не.
Все, ясно.
У нас следующая сессия, я тебе сейчас скажу, начинается. Следующая сессия начинается ровно в 15.
Нет, она в 15:15, там есть маленький перерывчик.
Да, 15:15.
Мы заложили 15 минут вот на всякие...
Да, 15:15. Так что нет, у нас тут никакой свободы нет. Мы сегодня до вечера, до 19:30 работаем. У нас там наша последняя сессия, где все наши выступают с 17:30. Так что все жестко. Ну, у нас вроде все такие очень дисциплинированные.
Нет, это чисто технический момент. Тут ведь не в этом дело. Но у нас 2 минуты до начала, я не знаю, как.
Коллеги, может быть, если у Александра Николаевича проблемы, может быть, тогда поменять сейчас спикеров местами?
Мы так и решили, что мы Александра Викторовича тогда выводим. Давайте все. Тогда
так
я могу...
15:30 нет, товарищи, и Александр Николаевич онлайн нет.
Нет,
мы его не слышим.
У него проблемы с звуком.
Оль, а Игорь будет в записи или нет?
Нет, он должен быть лично. Он, по крайней мере, Саша Курсанов написал, что он будет лично. Так что мы его ждем вообще-то, потому что он запись-то не прислал.
Слышно?
Сейчас слышно, да.
Да, замечательно. Ну, я подписан как Андрей Бочаров, ну ладно, это буду все равно я.
Ну все прекрасно, все супер, все на месте. Александр Николаевич, мы тут уже немножко обсудили. Я сделаю вводное приветствие, вот, а потом передам слово.
А потом я буду делать, хорошо.
Да, ну вас Олег сначала представит, Олег Лукин, это сопредседатель.
Хорошо.
Потом, значит, вот. Ну и Олег может представить наших и остальных гостей, потому что он их знает хорошо и про них что-то может рассказать, если вы не возражаете. Вы нас слышите?
Я, да.
Хорошо. Вы просто не ответили на мой призыв.
У меня что-то было сломано, я отвечал, но машина не реагировала на мой голос. Я поменял машину, бежал до соседа, выгнал его из компьютера.
Понятно. Ну, вы какой-то отчет дадите, Василий Ильич?
Да. Мы, в принципе, вот через все мы начинаем.
My dear colleagues, let me open our last plenary session at our conference. I would like to say that this is joint symposium of the Conference on Experimental and Computational Biomedicine, which was held in Ekaterinburg, and our combined conference, IEEE Euro-Siberian Conference on Computational Technologies in Cognitive Science, Genomics, and Biomedicine. I would like to say that first 2 days were very successful. We've had 1 day of real talks in Ekaterinburg, and then great day of online session yesterday, both at the IEEE conference and Ekaterinburg conference, and today is our joint conference. And I would thank our plenary speakers at this session. Them are great, and we wish to listen their talks, and I'm excited about listening to that. Thank you them for contributing to our conference, and I give the rules of the chairman to Oleg Lukin. Oleg, please introduce our first speaker. Thank you very much.
Thank you, Olga Eduardovna. I thank all of us for visiting these brilliant lectures, and I would like to present our first speaker, Alexander Savastyanov, Doctor of Philosophy, Scientific Research Institute of Neuroscience and Medicine from Novosibirsk, Russia.
Okay.
So,
hello, colleagues. I see my my presentation.
yes, we see.
Okay, very good. Just a moment. I would like to present my talk. Topic of this talk: combination of EEG and linguistic methods for searching of neurological markers of personality traits and mental disorders. The personality traits are psychological characteristics of individuals which can be stable during a whole period of life. The study of these traits is important for many different areas, including the diagnostics of inclination to mental disorders, such as depression, autism disorder, stress disorder, anxiety disorder, etc. Psychological questionnaires are a typical tool for estimation of individual personality traits, but such tool has many disadvantages due to the great subjectivity of the answers to questions. And in this reason, there is an actual task to develop an approach to assessing the personal characteristics of individuals in norm and pathology by using objective methods for assessing their behavior or patterns of brain activity. In our view, the combination of linguistic methods of stimulation with recording and analysis of EEG gives an opportunity objectively estimating human personality traits and inclination to disorder. Here you can see one of our experimental models. We select 5 categories of sentences. First, it's neutral, non-emotional sentences about unanimated objects. Second, it's a set of sentences about my own anxiety or my own fear. Third, it's sentences about anxiety or fear some other person without names, but we used pronouns such as he, she, her, him, etc. And other category of sentences, it's sentences which describe my own aggression, and sentences which describe aggression of some other person. And what is experimental paradigm? Totally, we are using 200 sentences from 5 categories, and half of these sentences include grammatical error. And another half of sentences are grammatically correct. And experimental task, person should read a sentence and estimate it is grammatically correct or not. Person, experimental subject, doesn't know this about emotional categories of this sentence. Yeah, he or she concentrates only on grammatical structure of sentence, but we try to find markers of sentence emotionality. Yeah, how subject reacts to emotional content of this sentence. And here you can see experimental methods. Yeah, in the screen appears some signal, be ready, next presentation of sentence, and next subject response. And simultaneously with recognition of sentences, we are doing recording of EEG, and such parameters of EEG as evoked potential, CRP, or event-related spectrum perturbations can be used as a marker of brain activity, which related to process of speech recognition. Here you can see plot of event-related spectral perturbation. In this X, you can see time, and here it's frequency band. And red color, it's synchronization of frequency band, and blue color is desynchronization of frequency band. And you see it is signal be ready presentation, it is presentation of sentence, and you can see synchronization in delta and theta frequency band, and desynchronization in alpha and beta frequency band. For us, the most important characteristic is synchronization, means power increase in theta band after approximately 200 milliseconds after sentence onset. Please understand the idea. Just 200 milliseconds after sentence appears on the screen. It's very short time. It's extremely short time. Person cannot recognize this sentence. Yeah, can you read sentence in time period 200 milliseconds? Yeah, of course not. Reaction time, it's about 2, sometimes 5 seconds after sentence onset. But brain, human brain, can recognize emotional content of this sentence and relation of this sentence to person himself or to some other person. In the right side of this slide, you can see time of stimulus recognition. Yeah. It's sentence about object, sentence about my anxiety, anxiety of other person about my aggression and another aggression, and you can see that time of recognition depends on emotional content of sentence. Yeah, and especially important sentences about my aggression and sentences about aggression of other person. Difference only in one word. I am angry, he is angry. I like to kill some other persons. He like to kill other persons. Yeah. I am aggressive. He is aggressive. Yeah. Same errors. All other words are same, but time. Yeah. Very strong differences between reaction time for sentences about my aggression and sentences about aggression of other person. And same differences, not so strong, but also present about my fear and fear of other person. In behavior, we see that almost all persons, yeah, demonstrate different behavioral reaction on information about myself and about other persons. It is this slide in right side. It's quality of task recognition. And we see that quality of task recognition depends on a group of people. We organized this study in different regions of Siberia. We used 4 different regions. It was big city Novosibirsk, It was small villages around Novosibirsk with Russian population. It was region of Republic Tver and region Republic Sakha, Yakutia. And you can see these 2 points, my aggression and aggression of other people. Very strong differences in behavioral parameters of reaction between city population and population of rural regions. Yeah, especially strong differences between Novosibirsk and Tver. In Novosibirsk, people recognized sentences about aggression of other persons better than about own aggression. But people in Tver recognized sentences about aggression of other person with extremely bad quality. Yeah, they didn't see grammatical error in sentence if this sentence described aggression of other person. It is behavioral markers, yeah, but it is EEG markers. First of all, it's quite easy to recognize which language is native for participant. We have 3 situations. First, it is 2 similar tasks, but one in Russian language and second in English. Yeah. And it is cortical topography of P600 local peak, and you can see very strong differences in reactions on native sentences, sentences on native language, Russian for Russians, and foreign language, yeah, English. We can recognize, okay, it's this language is native, this language is foreign. This is Tuvinians, yeah, and we apply same task in 3 different languages, English, Russian, and Tuvinian. And you can see from Tuvinians no difference between Russian and Tuvinian language. They recognize both language as native, and strong differences between Russian Tuvinian and English. Yeah, English is foreign, Russian and Tuvinian are native language for these people. But interesting reaction in Yakuts, yeah, people from Yakutia. We applied 3 language, Russian, English, and Yakutian, and we see that only Russian language is native for these people. They are Yakuts, yeah, and officially Yakutian language should be native for them, but no, Russian language is native, yeah, but Yakutian and English are 2 foreign language for their brain. Okay, we can recognize which language is native. Also, by using of EEG, we can estimate degree of emotionality from different sentences and for different categories of people. You can see it is theta synchronization from 4 to 8 Hertz in first 300 milliseconds after sentence onset. It is all groups and 5 different categories of sentences. And you can see that green diagram, its reaction to neutral stimulus, and other diagrams, its reaction to emotional related stimulus. And you can see that reaction to aggression much stronger than reaction to sentences about anxiety and to neutral sentences. This is for groups and all categories of sentences. Yeah, it's average reaction for all categories of sentences, but for rural Russians, city Russians, Yakuts, and Tuvinians, and I don't know why, but the strongest emotionality demonstrated by Russian people from small villages. And it is reaction to sentence about my aggression and aggression of other people. It is rural Russians, no differences. It is Yakuts, also no differences, but very strong dispersion, yeah, in reactions. It is comparison of 2 categories for city population. Yeah, you can see that emotionality of sentences about my aggression much stronger than emotionality of sentences about other aggression. And opposite reaction, for Tuvinians, they react strongly from information about aggression of other people in comparison with information about my own aggression. Okay, we see that EEG allows us to understand, estimate subjective level of emotionality of brain reaction to some information. People, participants thought about grammatical structures of this sentence, but brain thought about emotionality. And we can estimate implicit non-controllable reactions of brain in different kind of information. Okay, and it depends not only nationality, not only region, but also from psychological traits. All people participate in psychological testing by means of standard Big Five questionnaires. And we see that different components of EEG reactions strongly correlate with scores of different parameters of psychological tests. Especially strong effects were found for extraversion, neuroticism, and psychotism, which were measured by Goldberg or Ising test. And we see that higher level of extraversion demonstrate stronger brain reactions to emotional related information, and higher level of neuroticism and psychoticism demonstrated decrease in brain reaction to emotional information. And we see correlation between psychological traits, which are measured by psychological tests, and behavioral and EEG reaction during recognition of such linguistic stimuli. Next, we try to compare group of people who two groups of people who are living in a stable condition and trait migrants. We checked three groups. First of all, it's residents of Novosibirsk, mostly students who lived in Novosibirsk during more than 10 years, and we organized experiment immediately in Novosibirsk. Next group, it's residents of Yakutia who also lived in habitual for them climatic and social conditions. And third groups are migrants, about 60 persons who migrated in Yakutia from southern regions, mostly from Tajikistan and Uzbekistan, and group about 20-25 persons migrated to Yakutia from Africa. It's of course they are in stress condition, and we checked this group twice, immediately after their migration and after approximately half of year. It's two periods of adaptation. Actual adaptation immediately after transfer, and adaptation, long time adaptation after some period of living in new conditions. Here you can see results of psychological questionnaire, and you see that for migrants, after immediately after their transfer, it's typical to feel negative emotions. They demonstrate high level of neuroticism, high level of anxiety. They demonstrate low level of emotional intelligence, and they demonstrate low level of agreeableness. It means that migration is some stress factor which decreases ability of person to regulate own emotionality, own emotions. And it may be reason in disorder in communication with other people. And here you can see difference between migrants and Yakuts in their brain reaction to emotional related sentences. And you can see that in brain opposite, they demonstrate stronger brain reaction than local people who constantly live in region. It's maybe strange, looks strange. In behavioral level, they demonstrate difficulty in recognition of emotions, but in brain level, they demonstrate deeper activity of brain regions which related to emotional recognitions. And after half of year, migrants demonstrate decrease of hyperactivity of brain and increase in behavioral ability to recognize emotions. And it's strongly correlated with level of depression. People who demonstrate high level of depression after half year of adaptation show it bad behavioral ability to recognize emotions and hyperactivity of brain during emotional recognitions. But people who demonstrate low level of depression also show it high ability to recognize emotional speech and low level of brain activation. And it was correlated between each other. Second group, it was meditators. We specially organized expedition in Baikal Retreat Center. And we organized study with 60 persons who practice meditation. And compare their behavior and their brain reactions in same experimental paradigm. In psychological tests, meditators, here I write Baikal. Baikal means group of meditators, Buddhistic meditators who practice sahaja, no, not sahaja, shamatha meditation. And they demonstrate low level of negative emotions, low level of anxiety, low level of neuroticism, but also low level of ability to recognize emotions of other people. They demonstrate low level of not only negative, but also positive emotions. Yeah? They had low level of negative emotions and low level of positive emotions and low ability to recognize emotions of other people in comparison with people who have not meditation practice. Yeah? And in brain, it's interesting situation. It is reactions in information about I and other in non-meditators, and it is reactions of brain in information about I and other for meditator. And we see that in ordinary people, I am ordinary person, I never had meditation practice, brain reacts in information about me and about other by a very different manner. But in meditators, no difference in information about me and other. And also we see that information about anxiety and aggression very different for non-meditators and not so different for meditators. And the most sensitive frequency band is theta band, which relate with limbic system of brain. And this band is a marker of meditation practice. We can recognize this person practice meditation or no. Yeah. Theta band is the marker of participation in this specific religion psychological exercises. And the last group which was studied by our methods is patients with major depression. We have a group of patients who received medical help in clinic, and they also participated in our study. And we compared reaction in theta band for persons, for healthy persons and for patients. And it is 5 categories of sentence which were presented for our participants. Blue diagram is healthy persons, and red diagram is patients with depression. And you can see reaction to my aggression, almost no difference. Reaction to information in my own aggression, no difference for healthy person and for patients. Yeah, but strong difference in reaction on information about aggression of other persons. You can see that people with patients with depression demonstrated more strong reaction to this information in comparison with healthy persons. And also interesting situation, my anxiety and anxiety of other. Yeah, for my anxiety, own anxiety, anxiety of participant, patients demonstrated stronger reaction in comparison with healthy participants, but sorry. Anxiety of other person opposite. It means that this patient very sensitive to information, negative information about themselves, but sensitivity to negative information about other people decreased, but also increased reactivity to information about some danger. And interesting situation, they also demonstrate emotional reaction to neutral information. For most of healthy participants, neutral sentences didn't induce any emotional reactions or induce just slow emotionality. Yeah, but for patients with depression, neutral information, it's emotionally negative stimulus. Okay, and in conclusion, we can use this model for study of emotional related reactions of human brain. We can recognize a difference in brain reaction to information about me, about other people, difference in reaction in neutral information, information about fear, information about aggression. And we can see that many different factors such as cultural specificity of persons, psychological traits of persons, living events such as migration, participation in some specific practice such as meditation, all of these factors are influenced to brain reaction on this type of emotional information. And also we see that difference in this reaction can be used as a marker of depression. Not only depression, because we have similar results for autism, but for depression it's much more clear than for autism. And in future we can try to apply this model for diagnostic of affective pathologies and efficiency of therapy of such pathology. Okay, thank you for your attention. I am ready to answer to all of your questions.
Thank you so much, Alexander. It was very interesting to see these brilliant results, but unfortunately we have no any time room to provide any question. I'm sorry, but we should follow to the next presentation.
Thank you for your attention, colleagues.
Thank you so much as well. The next speaker is Alexander Panfilov. He is a professor at the Department of Physics and Astronomy in Ghent University in Belgium. And also he is guest professor at the Department of Cardiology of Leiden University Medical Center, Netherlands. And I would like to say that our team in Yekaterinburg, in very deep and long collaboration with the team of Alexander, and importantly, the collaboration relates the modeling of electrical events in myocardial tissue. So it's my great pleasure to invite Alexander to proceed his report. Thank you.
Okay, thank you very much. So first of all, I would like to thank you for invitation for this meeting. As I understand, part of this meeting is still related to the legacy of Professor Marhasin. And of course, I knew Vladimir Semyonovich for many, many years since I was a student. And we talked with him a lot on different subjects, and he influenced a lot of research, my research and research of my group. And I also wanted to say that Oleg already told that we have strong collaboration with Yekaterinburg. And Yekaterinburg, if I may say so, became my second home in Russia. And I always enjoy visiting you. But unfortunately, due to obvious reasons, I cannot do it on this occasion. Okay, so this is my talk. And this research I did in laboratory of Daniel Pineapples, and his team is present here. And my main aim here is to tell you in a little bit more detail about our recent publication, which was published in journal eLife, and it deals something with unusual way of defibrillation of the heart. I put also few very basic slides because of I don't know who is online for my talk now. But when we talk about sudden cardiac death, then at least in the West, people usually see such posters. But even such posters already show that a problem of sudden cardiac death is electrical problem. And it is related to internal electricity of the heart. However, in order to treat it, only way of doing it is defibrillation. And defibrillation is again done with electricity. But here we apply huge electrical stimulus from outside of the heart. But what is going during all these processes? And this is my basic slides. We know that all bioelectricity in heart and nerve tissue is a result of different concentrations of sodium and potassium channels outside and inside membrane. And therefore, we have a so-called difference of potentials at membrane in normal conditions. Then, if due to some stimulus, sodium gates are open, then we have inward current, which depolarizes our cell, and it brings potential somewhere from -90 to something to +10, +20, +50 millivolts. And then after a while, potassium gates are open, and they produce current in the opposite direction. And because of that, we have repolarization of a pulse. And this is what we call a normal response of cardiac cell. When we talk about spatial propagation of these effects, then we also talk about this in slightly different terms. We talk about three phases, big phases of action potential. The phase when our tissue is at rest, noted here as a white. Then red is a state in which cardiac cell can excite its neighbors. And then there is also important refractory period during which the cell cannot be re-excited anymore. But after that, cardiac cell recovers its properties, it becomes again white. And therefore, we call this tissue as a tissue with recovery. And because of these recovery properties, there are possible so-called vortices in the heart, different types of vortices.
And onset of these vortices is normally results in cardiac tachycardia or fibrillation, and this happens because the period of rotation of such vortices is very high. It's like, I don't know, 200, 300 beats per minute. Then what is defibrillation in that sense? Then if, for example, during such source of arrhythmia, a huge external electrical current applied to the chest of a patient, then it moves all cells which are addressed to the excited state. Then all these cells will be refractory, and then after refractory state, it will go again to the resting state, and arrhythmia disappears. And this is like classical defibrillation. However, question what we studied also, what I would like to tell in this talk, but can we also depolarize and thus defibrillate the heart without external electrical current? Because I told you we have a lot of electricity already inside the heart, as we have different concentrations of different ions, and therefore playing with it, like all this electrical activity occurs. And the answer is yes. By the way, one of the way of doing it is mechanical deformation, and former speaker at this symposium, Peter Cole, is a world specialist on that. But here I would like to talk about different thing, which is called optogenetics. And optogenetics came to our area from neuroscience. And optogenetics means that in addition to existing channels in cardiac cell, we add additional channel, which is can be opened by light. And using it, we can open this channel by flash of a light, and therefore can induce inward current, and therefore we can initiate this depolarization signal. And let me illustrate it on one work which Daniel Pineapples did a few years ago, when they expressed this rhodopsin channel in the heart of a rat. Then they put additional electrode on the heart of the rat, and by burst spacing of this electrode, they induce an arrhythmia. This is shown here by ECG, so that this is normal, how we call sinus rhythm. This is burst spacing, and this is ECG of arrhythmia. And now let us see how it can be controlled by light. So in this video, we see it like sinus rhythm. Then this is burst spacing. Then you see ECG and arrhythmia, and now have a look to the heart, a small heart which is here. You see flash of light, and then we see sinus rhythm again. One more time, arrhythmia, flash of light, and this disappearance of arrhythmia by this. And this is, of course, what we think is much better way of doing it, of defibrillation, because there is no external electricity and all comes by internal things. Therefore, it's much less damaging and much more efficient than we think normal defibrillation. Optogenetics now is used widely in cardiac research, and I also wanted to show you another example of our work with Daniel's lab, where we did the following. So, like, because we can like create a lot of simple experiments using this light. For example, we can have a system, and the system can project a spot of light on cardiac tissue, and then we can move this spot of light, and so on and so on. And here was an idea that using it, we can maybe control vortices in cardiac tissue. And this is like also paper published in Life also a couple of years ago. Idea here was the following. So for example, we have a vortex, rotational activity in the heart, which normally would exist here. But then what we do, we put a spot of light somewhere. And then when spiral anchors and starts rotating around this spot, we slowly move it to the boundary, and then this vortex disappears. Let me illustrate it also on video, which has in silico and also in vitro research in cardiac tissue. This happens exactly what I told you here. We move the spot, it moves to the boundary, and disappears. Therefore, here it's also working, and and we can test a lot of hypotheses. We have already several papers published on that. But how realistic to do it in patients? Daniel in his group did some additional steps in that direction. They also published, I think, last year paper, and also very good journal, Science Translational Medicine, where they put this idea a little bit forward, and they put small LED lamps under the chest of a rat. And then they were able to illustrate that this process also works in vivo. However, you see that even on experimental animals, it's extremely challenging to do it, because you need to modify heart with, you need to put this light source and so on and so on. Therefore, frankly speaking, we do not think that optogenetics in its classical view, how I presented it to you now, will be used in clinics soon. But working on that brought to an idea, and this was idea of Daniel also for many years, idea of biodefibrillation. Because we see that optogenetic channel can open and remove arrhythmia, can we maybe possibly make an additional step and therefore invent some channel which not only opens and removes arrhythmia, but also will be able to sense arrhythmia by itself. Therefore, basically, idea was to construct an ion channel, we would call it, for example, the defibrillation channel, which can sense arrhythmia, open and stop it. And when this idea came, Daniel, and also at that time we're already working with him and discussing, but what should give a signal to a channel to become open? Therefore, this is a question: what happens in cardiac cell during arrhythmia, which is not present in normal situation? And then, of course, there are many changes, like changes of calcium transient, some concentration of ions, etc. But the most obvious thing, of course, is that there is a different frequency during arrhythmia and outside of arrhythmia. And how we can sense frequency? And of course, there are a lot of systems which perfectly sense this frequency. And both of them are actually relevant for today. It can be, for example, temporal summation, which people observe in muscles, or it can be very famous mechanism of presynaptic potentiation of axon, by also by temporal summation of signals. Therefore, basically, what we need to do, we need to do something for cardiac cells. How can we do it? Again, I don't have time to tell you in all details, but I can tell you a few additional steps, and they're quite elementary. First of all, if we want to invent a model of a channel, then we have something that's called Nernst potential, which needs to bring voltage to some defibrillation part. And then there should be an activating gate for this, an activating gate described by this equation, and we see that this A goes 0 for some threshold value, for example, -16, and then it goes to 1. Okay, then if we now have an action potential, then part of this action potential, it will be above this threshold value, part below. Therefore, part of time it will move upwards, part of time it will go downwards. And then by playing of these rates, because this is a model, we can do everything, whatever we want. By changing these rates, we can invent a channel which will do this temporal summation. And this is one of examples of also simulations where we see that for low frequency, and this is this is variable A, it just oscillates, but for high frequencies, it accumulates and goes up. But we had a problem. If you do it in such simple way, then if channel is opened, the defibrillation channel, and if it depolarizes the cell, then this variable A will be at its maximal value, and this channel will not close. What to do here? And here again, we can just do very simple thing. We just add to addition of activating gate, also inactivating gate H, which has opposite dynamics. And this gate, after a while, will close this channel, and then all should work. And as a result of that, we invented a model, which later became model 2, which described here. It has also some additional things, so-called clock variable, but I don't have time to tell you all small details here, what we did. And then, after a while, another model was proposed also in collaboration with our PhD student in my group, Nina Kudryshova from Moscow Institute of Physics and Technology. She created a slightly different model of channel. It also had a temporal summation, but an activation mechanism was different. And also in our group, by talking with different members, we also invented another model of this channel, which, like if you look to green, you see this green somehow active when we have a front of a wave. Therefore, this green somehow counts number of wavefronts. And by count, and if you have more wavefronts, this channel will open. And this was also like one of ideas of what we can do. And when we had these models, we started to test them. And we had a lot of movies. I just show you one of them. This is virtual tissue with channel, and this is without channel. And in both tissues, we will induce an arrhythmia. So just wait a little bit. Now arrhythmia occurs. And now here, this channel starts working, and we see that it modifies excitation, and we go back to sinus rhythm. But here we have fibrillation. Therefore, we see that in this case, channel was successful. And we checked it for all models, model 1, model 2, model 3, and in all cases, it was working good. And in addition, we also checked it for all arrhythmias, which we knew, like ventricular tachycardia, ventricular fibrillation, arrhythmia which occurs due to fibrosis, arrhythmia which occurs as a rotational blood anatomical scar, and so on and so on. And we also did it in the whole heart. It's nicer movies, which shows the same movie, but in anatomical model of ventricles. And we see like detection of arrhythmia, and it's like defibrillation by this channel. So it's a little bit longer. Yeah. So all restored. And we also have the same for atria. This is even longer movie, but it will show you the same. I will stop it at some stage, but also the same idea that when we induce something we called atrial fibrillation, this channel can check it, like record it, detect it, sorry, and remove it by its opening. And we did it, and we were like very happy with our results. And we submitted our paper to eLife. And it was really a very special paper for us because we like kind of, I don't know, invested a lot of time, a lot of people were busy with that. And we thought, oh, we have excellent paper, we have excellent journal for that. But unfortunately, we had a rejection. And in this rejection, I highlight its long report. It says something like that in absence of even a small biological experiment, the work remains a theoretical proposition that may not prove to be correct. And then we also tried another journals and so on, but only high profile. And it was unsuccessful. And we were thinking, okay, but what should we do? How can we do it? Because we cannot, of course, during a few months, invent a new channel and put it in experiment. But here came a very interesting idea. Again, it came not to me. It came to a new postdoc in Daniel's lab, Balázs from Hungary. And he was specialist in voltage clamp, and he told us, but why don't we use an action potential clamp? And what is it? So like, I don't know how many of you, but in cardiac electrophysiology, people use a lot of so-called patch clamp when they use a small pipette, which they using some suction attached to a cardiac cell, and then using this pipette, they can measure a lot of things, and they can also influence this cell by putting some electrical current through this pipette. And then, like a long time ago, in 1996, a paper in the Netherlands was published by Ronald Wilders, who did the following. He studied a connection between two real cardiac cells, but he connected them via these pipettes, and he wanted to see how one cell influenced other cell. But he did not connect them directly by like gap charts, etc., but via these electrical devices. And at some stage he thought, but why can we do the next step? Why we do not replace one cell, real cell, by a virtual cell, by a computer cell, and do connection between these two cells and study what is going on. And I think Olga knows what will be my next slide now. Actually, idea of interaction of virtual and real biological thing was widely like developed and used by Professor Marhasin. And he used this virtual duplex where one cell was real and one virtual. And actually the same was done by like Ronald Wilders. Again, I don't know like really timeline, who did first, etc. I would imagine that Vladimir Semyonovich did it before him. But I know for sure that he knew this paper by Ronald Wilders, and we discussed with him, and he was very also positive altitude about this thing. And therefore, when this idea came, we went, basically, it was like Tim de Coster, my former PhD student, and now postdoc in Daniel's group, went to Ronald Wilders. He's now working at in Amsterdam at a medical center. And they did experiments. They isolated real cardiac cells. They created this system where they had a real cardiac cell, put a pipette there, and they wanted that this pipette will be fed by electrical current, which is calculated by a computer, in that sense by model 1. And what Tim told me, that he did not change anything from equations which were written in the paper. He literally just programmed this very simple equation to a computer, and then computer was producing this current, and then it was acting on this cell. And then he got what we wanted. Now again, in orange, it's a control cell, and in the black is cell in which this virtual channel, it's still virtual channel, but it influences real cell. And we see here, we see this current which is generated by this virtual channel. And we see that indeed this current is activated after high frequency. We have a peak, and it defibrillates the cell. And we also did it with other frequencies and have other results. But anyway, it was proven. It was, in that sense, that minimal experimental proof of a concept. And finally, our paper was published last year. Again, biodefibrillation is one of big research directions in Daniel lab. I'm also a member of that lab now. And we work now in several directions. So first of all, we're looking for existing ion channels which can be performed directly as a defibrillation channel. We're also looking for a modification of existing ion channel which can be used for biodefibrillation. And finally, we are also thinking about creation of new synthetic channel which really can be designed and maybe put to a cell which can do the job. This is like in general direction. Again, we work on this, couple papers are already in the pipeline, and we can present about these papers
to you shortly. But finishing, I wanted to say, but for me, independently, what will be results of that studies, I think this idea, and I may say so because it's not my idea, of Daniel, is in principle very good and great idea. And if I formulate it in a general sense, I could tell something like that. So what we tell, that maybe by modification of ion channel, we can create an action which can help heart to fight with arrhythmias. And I think it's very, very much plausible to do it. Maybe the channel, or most likely the channel, will be not the same channel which were written in our computer, but I'm pretty sure that like guided modification of ion channel can indeed improve our heart and make it resistant to arrhythmias. So this is once more time a lab, or I would say maybe even our lab in which we performed, and thanks a lot for your attention. Okay.
Thank you so much, Alexander. It was very, very interesting presentation. Does anybody have any question? Colleagues? Alexander, can I then ask you for one thing?
Yeah, sure.
It's about the putative defibrillation channel. If I understand well, it can transfer ions. And what's important, what might be important, the type of ion. For example, can it transfer calcium ions?
I mean, yeah, of course, in model, we tell our channel to transfer all what this channel wants. And of course, like right now, we're working with so-called inward currents because we want to depolarize our cells. Therefore, most important for us are like sodium ions, or it can be how people call non-selective channel. Also, any peak channel will do the job. But we're also looking for other channels like calcium. I wouldn't myself design a channel which would put additional calcium inside the cell, because you know that calcium is a huge activator of many intracellular processes, and I don't know how healthy it would be. But we're thinking about calcium also as possible regulator, because calcium also changes during arrhythmia, and maybe calcium itself can indeed be a trigger of open this virtual channel.
Okay, it's very interesting. Okay, thank you so much. We have 2 minutes more. If somebody want to ask Alexander. Otherwise, thank you so much again, Alexander. Then we can proceed to the next speaker. I would like to present our speaker, Igor Yefimov. He is professor, Dean of Department of Biomedical Engineering, the George Washington University from USA, Washington. And I also very happy to say that Igor is a very brilliant lecturer. For me personally, it was a great pleasure to listen to several of his lectures. And he also visited our team in Yekaterinburg, where he, for example, presented very beautiful lecture about different areas of biomedical science, including optical mapping, and also including the using of new approaches and new methods for implanting the electrical devices which can control the heart rhythm. So it's my great pleasure to present this speaker. Igor, can you proceed, please?
Yes,
Thank
hello.
You.
see my slides?
Yeah, we can see.
Okay, great. Well, thank you so much to the organizers for giving me this opportunity to present. And I'm particularly grateful because indeed I visited Ekaterinburg, I guess, 2 years ago. And I talked about our work on implantable bioelectronics, on new generation of bioelectronics. And I'm happy to present now a continuation of this theme with our new work. And so here's my talk. So first of all, I would like to make some disclosures. I will talk about bioelectronics, which some of it is part of one of the two startup companies, Cardiotoxicity and Nucera Biosystems, which we recently started. And my research was funded by National Institutes of Health, NSF, LaDuke Foundation, and the American Heart Association. Also, I would like to to acknowledge contribution of my lab members, Rose Yin. She is the PhD student in my lab and leading researcher on this topic. Cardiac surgeons Ben Lee and Sheena Chen, who helped to develop surgical procedures to implant miniature implantable devices into very small animals, mouse and rat. And Kedar Aras, who helped to develop a catheter, which I will present here for treatment of arrhythmias. And of course, I'm happy to present our collaborators from Northwestern University, Professor John Rogers, who invented material science behind this electronics, Philip Gutruf, who was his postdoc and now he's assistant professor in Arizona, University of Arizona, Mandy Han, and David Choi, and also Kwan-San Yang. So let me start by referring to this very interesting article, which was published in 2012 and dedicated to the bicentennial anniversary of New England Journal of Medicine, arguably the most reputable, most prestigious medical journal. In 1812, when French troops and Napoleon were in Moscow, the first issue of this journal was published. And this journal, this first issue, contained one article about angina pectoris, or chest pain. It was description essentially what we now understand as myocardial infarction. And at that time, of course, it was not entirely understood. And since that time, a lot was done to alleviate pain and death from myocardial infarction. And this article, published in 2012 by Nabel and Braunwald, leading cardiovascular researchers from Harvard Medical School, showed that statistically since 1970 approximately, there was a dramatic reduction in mortality from cardiovascular disease in the United States and worldwide, but this article was only about the United States. And from 1970 to 2010, approximately, there was a 70% reduction in mortality. And if you look at the major contributions which led to this reduction in mortality, primarily these are bioengineering advancements or surgical advancements, which frankly, I think it's very similar engineering advancements. Very few are actually pharmacological advancements or biological advancements, such as, for example, beta blockers, also the HMG-CoA reductase inhibitors, but primarily it's open heart surgery, angiography, then the CABG, which bypass surgery, defibrillator, then stenting, resynchronization therapy, etc., and left ventricular assist device is the most recent advancement. So therefore, bioengineering is really important in treatment of cardiovascular disease, and that's essentially what I will be talking about. So how is defibrillation done? I saw just Sasha Panfilov just described in his presentation from a biophysical point of view, but in practical terms, basically a defibrillator is implanted and then electrodes are fed through superior vena cava into the heart. Electrode is put into the right ventricle, and then shock, as shown here, about 10 millisecond long shock, biphasic shock, consisting of first phase and second phase, is applied between the coil and device itself, and that's how defibrillation is stopped. For many years in my lab, I studied defibrillation with optical mapping, and basically optical mapping allows you to map with very high resolution. And we know now how arrhythmias look like. So this is example of recordings of arrhythmia in the rabbit heart. And you can see there's a rotor which essentially maintains arrhythmia in the rabbit heart. This is a typical representation of such small heart. Interestingly, many years later, Michel Heisseger's group from Bordeaux mapped arrhythmia in humans, which is spontaneously occurring ventricular fibrillation using this non-invasive technique, which is called electrocardiographic imaging, invented by Dr. Yoram Rudi from Washington University in St. Louis. And you can see that arrhythmia is very similar to what we see in the rabbit heart. But this is only in the beginning of arrhythmia. As arrhythmia deteriorates with time, as this rotor breaks down, it becomes more and more complex. And unfortunately, with such technique, it's very difficult to follow. It has limited resolution. Therefore, in our lab, we studied arrhythmias extensively. This is one example published 2 years ago when we will take a human heart from a donor who died from non-cardiac death, from brain death usually. We excise a small segment of myocardium, cannulate it, perfuse it, and we can basically study arrhythmia by imaging it from 4 sides, from an epicardium, endocardium, and 2 cut surfaces. So I'll skip, of course, all the details. And this is how arrhythmia looks like. So this is fluorescence recordings, phase space or phase analysis of the same recordings, and then wavefronts. So you can see that the dynamics of this arrhythmia on 4 surfaces, endocardial, epicardial, and 2 transmural slices, is quite complex, actually. It's not simple rotor. And you can see here, for example, on transmural surface, you can see there are some rotors, like right here one, but it's very short-lived. It's primarily maintained, fibrillation maintained by multiple wavelets, which are short-lived, they collide, annihilate, and start over again. But of course, what is important to emphasize always when there is a reentrant arrhythmia, there is this blue area, which is essentially excitable gap. This is excitable tissue. And my dream was always to create a technique which can track this excitable tissue and then apply very small stimuli, not defibrillation shocks, but stimuli on the range of 1-2 volt, which can terminate this arrhythmia. And this is what we are working on, how to approach this goal. So, but first, I'd like to very briefly mention that towards this goal, we first invented a technique which allows dramatic reduction of energy in defibrillation as compared to the single shock defibrillation. So here is an example of one of the preclinical studies published 3 years back in a dog, in which we implanted 2 coils, and one biphasic shock was replaced with a sequence of pulses shown here, and we call it multi-pulse therapy. So if such sequence is applied, we dramatically reduce an energy. For example, with single biphasic shock for this particular ventricular tachycardia, atrial fibrillation in this case, it requires 1.5 joule or 165 volt for leading edge voltage here. With our technique, with multi-pulse therapy, it goes down to 0.16 joule. As you can see, almost 1 order of magnitude reduction in energy. So subsequently, we conducted a clinical trial in patients with paroxysmal atrial fibrillation, which was just published in this year in the Journal of the American College of Cardiology, Clinical Electrophysiology. In this case, we enrolled 16 patients, which were coming for ablation. And in these patients, these coil electrodes will be inserted into the heart temporarily just for the study. Arrhythmia will be induced. Then we apply our therapy, evaluate, if necessary, change parameters. If it's not terminated, we repeat the therapy again. If it's terminated, we will have to reinduce arrhythmia and repeat it over and over again for about 45 minutes, which was allowed by our protocol. So this is how such study looks like. In one particular patient, 47-year-old male, you can see here arrhythmia in left and right atrium. This is electrocardiogram, applying therapy here, and resumption of sinus rhythm. So we repeated this therapy. As you can see, each dot here is one application of therapy, and played with energy to determine what was the amount of energy required for termination of such arrhythmia in a human patient. And what we found was very interesting, that if you apply higher energy, such as 1.2 joule, you achieve almost instantaneous termination. If you apply half of this energy, 0.5 joule, after therapy, it takes about 1.26 seconds to terminate. And if you apply even lower energy, it takes much longer, about 5 seconds. So essentially, there's a clear correlation between amount of energy and time, inverse correlation in time of termination. But statistically, again, without going too much into details, you can see that we achieved this termination in 71% of episodes, which is very good efficacy, with energy of 0.036 joules and voltage of 42 volt on average. For comparison, previous attempts to terminate intracardiac defibrillator to terminate atrial fibrillation required 6 to 7 joules. So we essentially are more than order of magnitude below that energy. However, it still requires traditional coil-based electronics. It's still application of energy at a large scale, although pain-free perhaps, but it's still not what we wanted. So therefore, as I already said, we focused on bioelectronics, which will allow to overcome this particular requirement for still relatively high energy. So this is a sequence of papers which led to this presentation, and I'm happy to say that the first 3 papers I already presented in Ekaterinburg during my previous visit a couple of years ago, and now I'll talk about these new 3 papers, one of which, number 4, was published in Nature Biomedical Engineering in 2020, and number 5 and 6 are not yet published, but they are accepted in press. Nature Biotechnology paper will be published actually in June, not July, and the Nature Materials paper will be also published during summer. We don't know the date yet. So briefly to introduce what led to our development is, so John Rogers' laboratory is famous for inventing so-called organ-conformal bioelectronics. In this case, basically, electronic circuit is printed on a membrane which is flexible and stretchable. In this case, it's shaped as a heart, can be put on the heart, and you can design this device any way you want. You can put electrical sensors and actuators, can measure mechanics with strain gauges, can measure or stimulate with light. pH can be measured, temperature can be measured, ionic concentrations, and on. So there is a whole toolkit which you can use to make such devices. We showed that electrical sensing works quite well, comparing epicardial electrogram recorded maps with optical mapping. You can see they are essentially identical, in this case during sinus rhythm, not during pacing. We can measure pH. Here's a map of pH. So we created next generation of this electronics. In each case, we equipped each sensor with its own circuit. So you have here 400 such sensors. Each of them has a microcircuit. We call them chiplets. In this case, it's a capacitor and amplifier for signal conditioning, and another switch for multiplexing, so which you can allow you to collect data or to stimulate at will where you want to apply the stimulus. So conceptually, what we really want to achieve over time is the following. It's still the concept. I will be working on it for the next 10 years, perhaps. So we want to create a circuit, a device, which will consist of 3 layers. One layer is sensors. Another layer is computational layer. It will use neuromorphic networking networks for computing features of propagated waveforms and determining those excitable gaps, as I showed you before, this blue area. And finally, there are a layer of actuators, electrodes, which will apply stimulus to extinguish arrhythmia by applying stimulus exactly at this blue area, as shown here. So conceptually, we looked at our data on human ventricular fibrillation. So here's a snapshot of the data I showed you before. You can see here is 1, 2, and 3 waveforms propagated throughout the muscle on the epicardium. And we designed a chip with a young assistant professor, Gina Adam. And this chip essentially can conduct local computation and then can calculate where such stimulus can be applied. At this point, this is still computational, but we are working on implementation of this microchip in actual hardware. But towards this aim, we started making first steps. So first of all, we made the first iteration of this device, which will be applied for ablation. So how ablation is done now? So a patient with atrial fibrillation, as I showed you before in our study, comes for treatment of arrhythmia. In this case, patient basically will be lightly anesthetized if necessary, put on the table. An incision will be made here to expose femoral vein, and then ablation catheter and mapping catheters, 2 catheters usually, will be inserted in this vein. and tunneled all the way to the heart through inferior vena cava and put in the atrium. So there are two catheters. One is a mapping catheter, which will be essentially used to map arrhythmia and find sources of arrhythmia. In this case, it's a so-called lasso catheter, but it could be basket or some other design, but it's basically just mapping. And another catheter is ablation catheter, which is consisting essentially of an electrode, one electrode which will be placed sequentially point by point. It takes many hours sometimes, and you have to burn essentially continuous lines around pulmonary veins. There are 4 of them in the left atrium, then connect them, and it's a very, very tedious and difficult procedure. Also, it requires X-ray because you have to see where those electrodes are located. Otherwise, you will be blind. So currently, there are a number of methods how this ablation is done. So most widespread are RF ablation, when radio frequency essentially current is applied for such catheter as I showed you before. There is also cryoablation, then essentially tissue is frozen to about -20°C and killed, cells are killed in this way. And RF, by the way, increases temperature to about 65 or 60°C, also essentially cooking those cells locally. There's also laser-based ablation, which is done by some investigators, and different configurations of such electrodes of ablation systems are used. A new method which is currently being developed is called pulsed field ablation, which is based on electroporation. So electroporation basically is a very brief shot of current at very large magnitude, but it's only microseconds, and it kills cells instantaneously without much collateral damage. So the way it's implemented is shown here. For example, sequential ablation, as I said already, within one of the pulmonary veins, you have to go point by point. It's a very difficult procedure. And you can see the success rate, depending on the study, varies, but typically in good hands is about 70 to 80% with this particular technique. Balloon is another technique when you insert something inside the pulmonary vein and you ablate basically through the balloon. You can ablate an entire line circumferentially around balloon. Again, success rate about the same, 70, 60 to 80%, depends on the experience of a physician. And pulsed field or electroporation based is done also with several different catheters, like for example, one shown here. And one study so far published showing 100% success rate, almost 100, which is quite remarkable. But again, future will show how it works. So what we designed is the following. So we combined all that, instead of having several different catheters in one device. There's an electrode array, temperature sensor array, and pressure sensor array. So they all are designed on the same system. They are all interpenetrated and can work at the same time or sequentially as needed by the protocol. So here's how electrode array looks like. It can be put on a balloon. It can be expanded, put it from inside the heart, like I showed previously, or it could be actually placed on the epicardium as well. But in this case, configuration of the system will be different. So the pressure array, it looks like this. It consists of very small pressure sensors shown here. These are little pop-up structures, which are essentially when you come in contact with tissue, they are slightly flexed, and deformation will cause change in strain, and this will, which is parallel to pressure within the normal range. And strain can be measured as electrical conductivity or resistance, and this way we can make sure that we have good contact between our sensors and the cardiac tissue. So here's an example how ablation is done. When we apply a RF ablation, as I said already, it's a slow process. It takes tens of seconds. As you can see here, depending on the amount of energy, we will increase temperature. And you need to reach, as I said, about 60 degrees, 55, 60, 65 degrees, which requires a certain amount of energy. And you need a real-time feedback for this type of ablation. And namely, you have to measure temperature. That's why we need a temperature array. And also, because you have multiple electrodes, you can connect them as an arbitrary line of ablation and conduct ablation simultaneously from several electrodes. Here are some examples of point ablations by this method. Alternative method, as I said, based on pulsed field or electroporation, is achieved from the same electrodes by instantaneous application of very large field, microsecond duration. And again, we achieve very reliable ablation shown here. And I believe this method will be preferential because it's fast and maybe more safe So this method is great. We're already working on developing a catheter, but still it's only interventional method. You have to insert this into the chest of the patient and then essentially remove the electrode after ablation is done. But we want to have a real-time device, and we have to develop, first of all, a platform for real-time device. So many years ago in 2013, we published this first paper in which we asked ourselves, how do we make a very small device which will be first step towards achieving such goal? So in this paper, we published essentially very simple battery-free device in which you have a coil, a couple of diodes, and 2 silver wires which are sutured to the heart of the mouse. This device does not have a battery. It's inserted inside surgically inside the chest, attached to the heart surgically. And then from outside, you will bring the coil. You will transmit energy using inductive power transfer, the same way like a transformer works. And in this case, you can stimulate the mouse. So this pacemaker worked. However, there was one big problem. It was stiff. So these wires are not flexible enough. And as a result, a lot of mice died because of heart perforation. So it was a big disappointment in this respect. And I met at that time approximately Professor John Rogers, who developed soft electronics. And I asked him if he can think of redesigning such design. And he put to work Philip Gutrov, who was his postdoc at the time. And they designed this beautiful device, which has a serpentine design of the electrode. And this electrode essentially is attached to the mouse or rat heart here through these holes, sutures. There are 2 platinum electrodes for electrical stimulation. And also we also put a light emitting diode to do optogenetic stimulation, which Sasha already described its principle. So it's I don't need to go into that. So we applied this system, and here is an example for electrical stimulation. So electrode works, this is just in vitro demonstration of principle. So first 2 bits are sinus rhythm, and then now here we will turn on the pacemaker. So now it will be paced beat right here, and then now will be another paced beat again right now. And you can see also on electrograms, first 3 recordings of during sinus rhythm, and these are paced electrograms. You can see they have very different QRS complex. We also were able to pace optically. So here is an example by applying pulses of light. You can stimulate the heart at different frequencies. You can see here electrograms during normal sinus rhythm, pacing at 5 Hertz, 6 Hertz, almost 9 Hertz, and 10 Hertz. So now this was done in vitro. Now we're testing in vivo. For in vivo, we developed a procedure. I mentioned already Ben Lee and Shina Chen were responsible for developing this complicated procedure. When chest of the animal is open, you stitch electrode to the heart, and then you tunnel this flexible electrode through the rib cage, outside the rib cage, and put it under the skin subcutaneously on the side of the animal for transmitting energy into this device. So this is how step-by-step surgery looks like. Again, you're shaving side of the animal, opening it, open the chest, attaching the electrode, closing the chest, and device itself subcutaneous, and then basically closing the skin of the animal. So we have a great video to show this procedure. Unfortunately, I don't have time, but a paper was submitted just recently with this surgery, and it will be available. So now how it works, here's example. So rat has an implantable device, and outside circuit is this red wire. So I'll show the movie. So now animal is, so spacing is working right now, as you can see. And now student will remove rat from this coil, and now you can see electrogram changed. It went actually into sinus rhythm, and now animal will be placed back, and you will see it will go back to paced rhythm. As you can see here, much faster rate compared to sinus rhythm, because coil now is transmitting energy into the animal's chest. So we looked at long-term parameters, are there any side effects, and found that it's minimally damaging. So during implantation, there was a reduction of body weight of the animal, but then with time, everything recovered. Animal felt very well. You can see that the fibrosis was minimal, as shown here, only at the site when we attached electrode to the heart. So for long-term pacing, we, as I showed you already, we place this cage inside another coil, attaching to the external box, and you can see here day 3, day 5, day 6 after implantation, up to 1 month we already can pace. Animals can stimulate the heart with such devices. But the next step was in this work, we wanted to create a temporary device. There is one very interesting procedure, which is done quite routinely on many children who are born with septal defect, with heart defect. And in this case, a temporary pacemaker is required because after surgery, one of the complications is AV block. So temporary pacemaker is required for approximately 5 days until AV block will resolve and heart can be stimulated by its own pacemaker by sinus node. But for 5 days, surgeons will leave a wire inside on the heart during surgery, and the wire will be tunneled outside the body through incision on the side of the skin here and attached to external generator. And then after condition will be resolved and external pacemaker is no longer needed, typically it's pulled out. Unfortunately, it also can lead to dramatic consequences because you can also pull out a piece of heart because of fibrosis. So quite often nowadays surgeons will just cut this wire and leave wire inside the patient's chest, and such patients have to live on for many years after that with this wire, which is also not a good thing. So we thought that we can develop a pacemaker which will be transient, made of bioresorbable materials. It will work like I showed you before for 5-7 days as required, and after that it will dissolve. So John Rogers' laboratory already developed many different materials which are bioresorbable. They have those required properties. So here they are listed. As I said already, this paper will be published in June in Nature Biotechnology. So for example, for conductors, we use magnesium. Magnesium actually is bioresorbable. Silicon nanomembranes used as diodes. If they are a nanomembrane and nanometers in thickness, they actually also dissolve quite readily. And also different other materials for encapsulation, for interconnections, etc. So when such pacemaker is made, as you can see here, and put in just a thyroid solution for at 37 degrees, day 0 it looks, of course, normal. And then at day 20, it's already almost dissolved, at least part of the electrode, and day 35, very little left here. So it works the same way as I showed you before for non-bioresorbable pacemaker. And here is an example how it works. So we place coil, and now we turn on the pacing. As you can see, now we turned on, and you can see very high frequency stimulation from the device. After that, we will remove the coil right now, and you can see heart return back to normal sinus rhythm. So principle is the same as I showed you before. We tested it for functionality in a mouse heart, rabbit heart, and a small segment of human myocardium. As you can see, it works in all 3 cases. So here is, for example, in the mouse sinus rhythm, the first 3 beats, and then we turn on the pacemaker, and you can see morphology of signals changed. And we can map it also. You can see that the excitation originated from this point where electrode was. Same with a rabbit heart and same with the human heart. Now, we developed the same procedure for implanting the device. So you open the chest, you attach the electrodes, you close the chest, and then you put coil to test. And you can see again, like I showed you before, we pacing sinus rhythm was at 350 beats per minute, and we paced at 400 beats per minute to overcome sinus rhythm. You can see it's functional. Now, animal is recovered, and we can test it at day 1 after implantation, day 4 and after implantation. As I said, we programmed these devices to live approximately 5 to 7 days, after which time they will start dissolving in the body of the animal. So here how it looks like. So this is at implantation. 1 week later, you can see there are already signs of decay of this device. At 2 weeks, it's already collapsed and some parts already gone. And at 4 weeks, you can see essentially nothing left. Device is completely dissolved. So we looked at histology. So you can see that immediately after implantation, there is, of course, very little fibrosis. At 3 weeks, there is some fibrosis. And at 6 weeks, again, this is magnified image fibrosis at the site where electrode was attached. But overall, behavior of animals showed that side effects are minimal. Again, body weight of subjects reduced after surgery, but then recovered very quickly, and animal felt really well. So ejection fraction was maintained at normal range with between 70 and 80%, which is healthy range for rats. And biochemistry of blood markers of inflammation also show that there was no or very minimal inflammation. So our collaborators from Northwestern University also tested this in dog. So again, same way, a device was implanted in the dog chest, attached to the heart, the animal was closed, and then coil was brought in to this area. And you can see, like in previous experiments, 3 first beats are shown as sinus rhythm, After that, patient was turned on, and you can see QRS complex and T wave show clear signs of pacing. This is how pacing ECG looks like in human patients as well. So it works safely, and our colleagues are now conducting long-term studies in dogs. So now, last but not least, a picture from this new paper, which just was accepted for publication. We ask ourselves a question: how do we replace surgical stitching, sewing of the electrode to the chest, to the heart surface, which is very tedious and very difficult procedure to do, but also causes some little bit of inflammation. So John Rogers' laboratory developed this new, very interesting bioadhesive, which is also biodegradable. So it's essentially glue, biological glue, which you can see we can glue this device. This is just example of non-biodegradable device. After 10 days being implanted, you can see it maintains very good contact. As you can see here, we can pace up to, like I said, already 30 days with this glue. And glue has beautiful properties. It's electrically transparent, has very little impedance, and also optically transparent. Can be used, for example, for optogenetics or for measurements of fluorescence. We also tested it for mapping. So we glued this matrix consisting of multiple electrodes for electrical mapping. It works really well. We also, of course, used it for many other applications beyond cardiology. So this bioadhesive, I think, is a big breakthrough, and a paper was accepted by Nature Materials just a few weeks ago. Will be appearing during summer. So let me conclude. So I believe I showed you that we developed already real-time low-energy therapy for cardiac arrhythmias, and it's already in clinical trials, first of which was already published. We also developed organ-conformal bioelectronics, which is a high-resolution platform for network sensing, neuromorphic data processing, and network actuation or therapy, such as stimulation or ablation. And first design was published. We're already working on clinical implementation of that. And also, we developed bioresorbable version of bioelectronics, which allows you to place temporary pacemakers. We're thinking about temporary defibrillator or temporary neural stimulator. These are now in development, and I hope to report it in the next few years, developments in this area. But overall, I believe a future is bright for this bioelectronics, and this newly developed, fully implantable, battery-free, and bioresorbable organ-conformal bioelectronics provides amazing opportunities for implanted, injectable sensors, actuators, therapeutic and diagnostic devices, which will be developed in the next 10-15 years. And that's what my group will be working on in collaboration with Professor John Rogers from Northwestern University. And I thank you, and I will be happy to answer any questions. Thanks.
Thank you so much, Igor, for your fantastic report. Unfortunately, we have no any time to give you any question, to give you possibility to answer. Thank you so much again, and I must to say also it's very big thanks to you from organizers and from listeners to you. Very early report. It's about 6 a.m. at the place of Igor. Thank you so much. And let me
maybe we have exception. We can accept more few questions for Igor. I think it's
okay. Just a single question, please.
Alexander Nikolaevich, please talk. Alexander Nikolaevich.
Yes.
Vasily. Olga Dordona, maybe you?
You have to turn on the microphone.
Sorry for that. Hi, first of all, so the early morning in Washington. Igor, I would like how light could be delivered. So as I guess the light should also be energy dependent, and that's why there is some charging for the device. So.
Well, light will be delivered the same way. So, I mean, the source of light is light emitting diode. So we make a very small diode. It could be micron scale device, very, very tiny. But energy for it will be delivered also through inductive power transfer. Basically, you feed energy through inductor. Like I said, same principle as capacitor. I'm sorry, as a transformer. and then it will be essentially electrically feeding the light-emitting diode, and it will be using for optogenetic stimulation. However, I have to say that optogenetic stimulation is a great concept, beautiful platform for studies. However, I personally really doubt it will be used clinically, because clinically, in order to use optogenetics, you need to make a transgenic human being. And I don't think anybody would agree to be transgenic. So, however, there are other principles by which you can optically stimulate the heart. For example, we are now developing 2 different methods in which light will be used for stimulation, but it will be not through transgenics. For example, one principle discovered by Alex Savchenko from San Diego now, but he is an alum of Moscow Institute of Physics and Technology, but as I am and Sasha. So it's a graphene-based interface. You can put layer of graphene on a cell. If you shine light on graphene, graphene transduces a power of light into essentially changes in electrical field and stimulate the cell. You don't need optogenetics for it, but you can optically stimulate. So this is one principle by which we will be designing those devices. And I have a grant on this right now, so we're working on it already. But thank you, it's a great question.
Oh, that is fantastic. Actually, you presented fantastic stuff, and we believe that biosensors and biomaterials in the future. So any more questions, please, guys?
Igor, can I ask you just technical question? I mean, not about this fancy electronics, of course, it's amazing, nothing to say. But about your first part, about atrial defibrillation, like with multiple stimulus, do you maybe have higher incidence of ventricular events? Because, yeah.
Yeah, it's of course, for us, it was question number 1 as well. I mean, if this would have happened, it would have been death of our method. And of course, we made every precaution not to do it, and we didn't have any events like that. The way we do it is we trigger atrial defibrillation by QRS complex of ventricular activity. So basically, our therapy is applied after QRS of ventricles during refractory period of ventricles. And that's how we avoid ventricular complications.
But in your case, stimulation goes more than 1 second. Are you still able?
Yes and no. First part of multi-pulse therapy, several beats, are going through actually very quickly, about 100-150 milliseconds during refractory period.
Yeah.
But the subsequent pulses are below capture threshold of ventricles. So subsequent pulses cannot stimulate ventricles. So that's why we have phase 1, phase 2, phase 3 of this multi-pulse therapy. So essentially, we designed it exactly to address your question, because obviously this question was for us also number 1 question. It's a very important question.
Okay. Okay. Thanks a lot.
May I also ask you to clarify? So you showed that if you use low energy pulses, it takes about several seconds to recover after fibrillation, but it's too much, yeah? People can die during this.
For ventricular defibrillation, I agree with you, it would be too much and will be unacceptable. For ventricular, therefore, we actually also developing this therapy for ventricular defibrillation and tachycardia. It's a little bit different plan. We need to achieve spontaneous or very quick recovery. You are right. But for atrial fibrillation, it's not a problem because, as you know, people live with atrial fibrillation. Not die immediately. Some live for tens of years with atrial fibrillation. And essentially, even if you have 50% success rate, only 50%, it means essentially on average you have to apply your therapy twice and you will succeed. But if therapy is of such low energy that patient does not sense it, it's not painful, it's not, there is no discomfort, then it's acceptable. And that's why even if you have few seconds, in some cases after application of therapy, it's not a big deal because patient will recover from fibrillation.
Okay, great. Sorry, guys, we have 5 minutes before next session and we have to finish. I would like to thank all our plenary session. We had fabulous sessions, so great talks. Thank you so much for contributing to our conference, and see you in future in Ekaterinburg personally.
Thank you from IEEE Russia.
Thank you so much.
Thank you.
Thank you. Bye.
Alexander, are you hearing me? Anybody here? I want to check my microphone.
Костя, вас слышно и видно, все
Thank
хорошо.
you.
Да, спасибо, что вы на месте. Через 5 минут начинаете.
But you're from Taiwan.
Hello. Hello, everybody. My name is Konstantin. I'm chair of this section. I'm glad to see you here, and it's a very big pleasure to see you all. And
we waiting the. Okay, I'm waiting Lou Michel. It's a first speaker of our section. Are you here?
Yeah, can you hear me?
Hello, thank you very much. Sorry, we have a problem with echo. Pleasure to see you, Shival. Excuse me, some noise, some voice repeat twice. Okay, I'm waiting to Michelle. It's the first speaker of our section. Are you here?
Yeah.
Excuse me. I ask you to fix problem with voice because a very strong echo appears. Yeah, and sorry, we have problem with echo. Pleasure to see you, Shival. Excuse me, some noise, some voice repeat twice. Excuse me. Could you use your? Excuse me. I asked you to fix problem with. Excuse me, we have very big problem with echo in your speech. Okay. We start. In any case, we start the first presentation. Please, yeah, please start. Let's start. Excuse me, we have very big problem with echo in your speech. Okay. We start. In any case, we start the first presentation. Please, excuse me, are you here? Maybe we start your presentation.
Okay. Yeah. In this talk, in this study, we try to link P200 components in language task with the ECG recorded under resting state condition, and we found something new. And this study has been collaborated with several experts, include Professor Yuwen Su from Graduate Institute of.
Are you here? Maybe we start your presentation.
Okay. Yeah. In this study, we try to link P200 components in language task with the ECG recorded under resting state condition, and we found something new. And this study has been collaborated with several experts, including Professor Yuwen Su from Graduate Institute of Linguistic at National Taiwan University, and Professor Alexander Sebastiano. And now I want to briefly describe our experiment. And the experiment includes the recognition of a linguistic ambiguity in Chinese sentence. And the stimuli contain 106 sentences, including 52 ambiguous and 54 unambiguous sentences. And we recruit 50 healthy subjects, and including 26 females and 24 males. We try to match their age, but it wasn't successful. Subjects went to our EEG lab, and we give them the state trait and anxiety inventory, and give them the implicit anxiety test. And also we give the implicit anxiety test twice. And after that, the subjects complete the resting state ECG registration with 4 minutes eyes closed and 4 minutes eyes open before the language task. Under the eyes open condition, subject had to maintain eye fixation on the central cross. And subjects complete the self-referential source skills, and the skill contained 4 sub-skills, including drowsiness, emotion, attention, and the self-referential thoughts. And all subjects complete the language task in which they were instructed to recognize whether a sentence shown on the screen could be interpreted in one way or more than one way. And here I'll give you some example about the ambiguous sentence, like 爷爷昨天走了, grandfather left or died yesterday. This is a lexical ambiguous. 走了 can means left or died. And 父亲听到母亲称赞自己. Father heard that mother praised him or herself. 称赞自己, 自己 means can mean my father or my mother herself. This is a syntactic ambiguous sentence. Also, finally, we have Mama 叫我把外套裹紧. Mother asked me to wrap myself up or to pick the coat tightly. So this is both lexical and syntactic ambiguous sentence. Here is our experimental trials. We show first on the screen, we show the central cross, and subject has to do the fixation for 500 milliseconds, and followed by sentence presentation for 8 seconds. And subject had to make decisions during 8 seconds. Otherwise, we treat this particular trial as missing. Also, we have a 4 to 7 seconds interstimulus interval. After the data pre-processing, we manually check individual subjects' ERP. And we pick out those subjects who clearly show, we check the left temporal and left frontal regions, clearly show the P200 components, and those persons would only show the N200 component. Lydia and me did the same things, but we cross-validated with each other's results. So finally, we classify subject into 2 groups. One group clearly showed the P200 component for both the fixation period and for the sentence recognition period. And the other group didn't show such kind of components. We call this group as a no P200 group. This is a ERP show the similar components, but the ambiguity is much smaller in the right temporal and right frontal region. And P200 group also shows stronger P3 components in the frontal, midline frontal, and the midline and occipital regions. And this is the ERP in the central electrodes. But also we can see that P200 group shows a slightly larger P300 components. But in the left or right parietal occipital regions, we don't see much difference between those 2 groups. You see the stronger N200 components. Okay, this is the right parietal occipital regions. Now we separate the ambiguous sentence and the unambiguous sentence. We plot both, and you can see that after 300 milliseconds, after the sentence onset, you can see that the P200 group show distinct ERP for ambiguous and unambiguous sentence. But the no P200 group, they show similar ERP after 300 minutes and post sentence onset. This is a frontal and temporal, left frontal temporal regions. And for the parietal, left parietal occipital regions, you can see that for the unambiguous sentence, both groups show slightly larger P300 components. This is the same for both groups. Now we plot the delta oscillation during the sentence recognition period, and 200 to 300 milliseconds corresponding to the P200 components. You can see that no P200 subjects, the group show much stronger delta synchronization in the parietal occipital regions. But P200 group show slightly stronger theta synchronization in the occipital parietal region compared with the no P200 group. But now we go back to the fixation period, and we pick the fixation period, which also shows more P200 component. But we can see that P200 group shows stronger theta synchronization in the occipital parietal region compared with the no P200 group. Also, no P200 group, they show stronger alpha desynchronization in the occipital parietal regions and during the sentence recognition period. Now we see we can compare the two groups on our behavioral data. Those data are not significant to differentiate those two groups, but in general, no P200 group, they have a higher, slightly higher state and trait anxiety. And they show they have a higher level of arousal and pay negative emotion to the resting state recording procedure. And also they pay higher attention to the recording procedure. And also they tend to less recording what happened in the past during the resting state recording. And now you can see that for the implicit anxiety, when the anxious words is share the same pattern with self, and no P200 group subjects, they tend to have a much longer reaction time. Okay, compare with the session which is anxious words share with others. So this is the same. We test them twice. They give quite consistent results. Compare with the P200 group. And for the response accuracy and reaction time to our language task in general, P200 group they took a longer reaction time compared with the no P200 group, but the response accuracy is also higher for this group. Okay, but both groups show had a perform equally well on the unambiguous sentence. See, the accuracy is 0.85. It's the same, almost the same for those two groups. Now we try to link the P200 component with our resting state heart rate variability indices, and we calculate more than 20 indices, but only we found a detrended fluctuation analysis index, alpha 2, which measure the non-memory dependence of ECG time course. And this index can be approximated by very low frequency power divided by very low frequency power plus low frequency power. Those are the range of the very low frequency, and high frequency power. And we calculate the ratio between, because we have eyes closed and eyes open station, we calculate the ratio between those two alpha 2 values. And this ratio per se can predict the membership of P200 group with 70% accuracy. Okay, after control gender and age. Now we add the trait anxiety, score, age, and the very low frequency power. And those four variables can improve the prediction accuracy to 90%. Okay. But now this conclusion, although the neural source of a P200 is unknown, empirical studies have indicated that this ERP component may reflect neural process recruited while a visual input is compared with an internal representation or expectation in memory. But according to our experiments, we found that stronger theta synchronization in the parietal region during the eye fixation period is a marker for the P200 group. Okay, members in this group show parasympathetic control under the eyes closed resting state, and the sympathetic control under the eyes open resting state. So the no P200 group show the opposite or reverse ANS pattern. So their resting state patterns are totally opposite to each other. So that's why this variable can have a very strong prediction power. That's the end. Thank you.
Thank you very much. We have some time for one or two questions. Questions, please. Okay. In this case, one question from me: how much electrodes is necessary for correct work of your processing?
Our equipment, we have a 128 channel. It's a high field channel.
Yeah. Thank you. Okay. Let's go to the next presentation. It's presentation of Vitalia Milchenko, cardiac valve involvement in ankylosing spondylitis patients taking different treatments. Let's start.
Can you hear me?
Yes.
One moment, I switch my presentation to present mode.
Also, we're waiting when we share your screen. Yes, thank you. Let's start.
If you see full screen.
Yes, we see full screen with your presentation.
Good morning, dear colleagues. I am glad to represent our simple but important for real medicine practice clinical investigation on the study of cardiac valves involvement in ankylosing spondylitis patients taking different treatment. All authors declare no conflicts of interest. Some moment about ankylosing spondylitis is a form of inflammatory arthritis that primarily affects spine. Ankylosing spondylitis symptoms usually begin before age of 14 and often as early as the teens and 20s. The lower back pain is worse after inactivity or when patients wake up. The final stage of this process is ankylosis. That's why early diagnosis and treatment of ankylosing spondylitis is critical. Treatment aims to reduce pain and symptoms, help you maintain normal posture, reduce complications and comorbidities. One moment. While chronic inflammatory lower back pain is a pivotal symptom of ankylosing spondylitis, the disease can have a wide-ranging impact on patients' health and well-being. The development of extraskeletal ankylosing spondylitis manifestations, including heart valve damage, impairs patient health and increases risk of death. Commonly prescribed medications for ankylosing spondylitis include non-steroidal anti-inflammatory drugs and different classes of biologics, which are targeted drugs that act on immune system pathways to reduce inflammation. The most common cardiac complications for ankylosing spondylitis are conduction disorders and valve insufficiency. These conductions were found up to 20% in ankylosing spondylitis patients. Because inflammation plays a pivotal role in ankylosing spondylitis and other autoimmune diseases, pathogenesis, and cardiovascular diseases, the effects of anti-inflammatory treatment on cardiovascular disease progression are being studied. According to a series of studies, the ankylosing spondylitis therapy by non-steroidal anti-inflammatory drugs, disease-modifying antiarrhythmic drugs, and biologics has an ambiguous effect on the cardiovascular system. Therefore, the assessment of cardiac damage in patients with different therapy is relevant. So the aims of our study was to determine the frequency of heart valve damage in ankylosing spondylitis patients with different therapy. 49 patients with ankylosing spondylitis were examined in rheumatology department of Research Institute of Clinical and Experimental Lymphology in Novosibirsk. The diagnosis met the modified 1984 New York criteria. All patients had signed informed consent. Patients were 39 years. All patients were male. The most of patients had moderate or high activity. The disease duration was at least 14 years. 38 patients received non-steroidal anti-inflammation drugs. 30 patients have a sulfasalazine, 8 methotrexate, and 1 combination of therapy. 16 patients received biologics. All of patients were treated in accordance with clinical recommendations, and we done all this procedure of investigation. Our result, the most common manifestations were left atrium dilation, aortic insufficiency and mitral insufficiency, and diastolic dysfunction. And these clinical manifestations is not correlates with other comorbidities, for example, with arterial hypertension. Left atrial dilation is the most damaged manifestation. Cardiac valves were less likely to be affected in patients with NSAIDs. It means non-steroidal anti-inflammatory drugs. Mitral insufficiency were found in 13% patients from NSAID group, compared with 4 patients, 44% cases in opposite groups. Aortic insufficiency was found in 18 cases, 80% cases, compared with 15 ones, respectively. Diastolic dysfunction were found in 15 patients with NSAIDs, compared with 60 cases in patients without NSAIDs. No significant differences were found in frequencies or cardiac valve lesions between the groups of patients taking biologics and DMARDs. So we present the conclusion that therapy with non-steroidal anti-inflammatory drugs may be useful to prevent the cardiac valve damage in patients with ankylosing spondylitis and have a cardioprotective effect. Thanks for attention. This is my business card for communication to take additional information and scientific cooperation. Thanks.
Thank you for presentation. We await some questions from our colleagues. Okay, one question from me. Could you explain how you use statistical processing? What do you do and what criteria you used for p-value?
Thanks for questions. We use a package of SPSS, and because group is small, we use non-parameter methods. At least description statistics and Mann-Whitney with statistical
Mann-Whitney test. Okay, thank you. Any other questions? Okay, in this case, thank you very much. Thank you for presentation, and we go to the following presentation. A brain tumor classification based on MR images using GAN as a pre-trained model. Hello?
Hello, am I audible?
Yes, but sound maybe you increase volume of your microphone. Could you say something to check?
Am I clear now?
Okay, it's good enough. Let's start. Let's start your presentation, please.
Sure. One second. So, good evening, everyone. I'm Dinesh Reddy. Today, I'm here to present my our research topic: brain tumor classification based on MR images using GAN as a pre-trained model. So here is the outline that I will be discussing here. So first starting with problem statement, like like the presence of brain tumor among humans has increased in large amounts in recent years. Like radiologists require a lot of time to manually annotate and segment these images. So like this, like if someone can achieve these things, like during like the well-timed detection and classification of the tumor can add to accurate treatment and can increase the survival rate of patients. Over the several years, like deep learning technologies has been like playing a vital role in the field of computer vision. Like one of its key uses in the medical industry is to minimize misdiagnosis and also the amount of time taken to annotate and segment these images. Like there are several researchers going on. So as part of this, like even my research is also to minimize this to minimize misdiagnosis and to improve to reduce the complexity involves in MRI image classification process. So like before, there are several existing methods for brain tumor classification, but like most of the researchers like lack high accuracy. So I want like in my research I try to improve accuracy along with that to improve the performance, like, and to reduce the complexity of classification of MRI images. So coming to the introduction, like as we brain tumor is an abnormal growth of tissue in the brain, which can lead to death if not detected early on every stage. So brain tumors can be cancerous, that is like malignant, and or non-cancerous, that is benign. So uh, like there are most common types of brain tumors are meningioma, glioma, and pituitary. So one most common widely test used to diagnose brain tumor is MRI scan. Why? Because MRI scan is used because it is less harmful and more accurate than CT brain scan. So my research is also on MRI scan images. So coming to the data set description, so here like I have used two data sets, like publicly available data sets. One is from Nanfang Hospital and General Hospital from China. So like based like this data contains like 3064 T1 weighted CEMRI images, like contrast enhanced MRI images from 233 patients, like which consists of 3 tumors like meningioma, pituitary, and glioma. So like glioma has 1426 and meningioma consists of 700 and 708 slices, and pituitary tumor 930 slices. So for better differentiation, I have also included no tumor images along with these three tumor images. So this data set I have collected from University of North Carolina. So basically this data was obtained from 20 healthy volunteers comprising MR brain images of 100 healthy subjects, which were scanned for a decade with different age groups and with exclusion of any subject with a history of any disease that affects the brain. So it's like a healthy brain MRI images, which I have considered as no tumor. So here are the samples of uh, um, here are the samples of the different types of MRI images, like glioma, brain tumor images, glioma, meningioma, meningioma, pituitary, and healthy, like that is no tumor. So coming to the method, like here I have used GAN as a pre-trained model. So coming to pre-trained models, like we have some transfer learning techniques. Like there are several pre-trained models like ResNet 50, VGG 16, exception, inception, V3. Like we have several more, several transfer learning techniques, but all these techniques have trained on some different domains, but not on, especially on like MRI images. So like if these can be used everywhere, but uh, here, like for my research, like I have used GAN for training MRI images, which we can use as a pre-trained model for further classifying new MRI images. So here, like in this research, we introduced new deep learning approach for brain tumor classification. On MRI images, a deep neural network is pre-trained as its discriminator in general adversarial network on MRI images by using MSG-GAN with auxiliary classification to extract the features and to classify the images. So this is like so this GAN is like normal GAN, which consists of generator and discriminator, but here in the discriminator, like we have I have included auxiliary classifier as well for better here I have added auxiliary classifier which can help the discriminator to maintain good feature extraction and prevent a mode collapse. So also this auxiliary classifier make sure to generate data the data by maintaining some condition, like for suppose if you want to generate only meningioma images, like this auxiliary classifier can be used. So as I said, like the fully connected layers of the auxiliary block of the discriminator consists of two blocks. One is auxiliary classifier and another one is like normal adversarial, which is common in other GANs. So here is the architecture, which for generator architecture, here like first the latent vector and the label are multiplied and passed to the embedding layer, which during train would learn the label representations from the latent vector, and it is multiplied to it. So then the output from the embedding layer is passed to the convolutional 2D transpose layer, where the deconvolution takes place and produces the image of size 4x4 from the latent vector. Then it is subsequently passed to n number of upsample and convolutional blocks to produce an image by increasing the size of the resolution of the image, like from 4x4 to 8x8, 16x to 16. So it depends on the depth what you are using. So here like I have used the depth as 6. So my like the output generated from this image will be with the resolution of 128 by 128. So the output from the generator, that is 128 by 128 resolution image, will be the input for the discriminator. It is that like this image will be downsampled by 2 at every stage by using a convolution block and average pooling. Then it is passed to mini batch standard deviation. Here the standard deviation of each feature of the activation map is calculated and then averaged over the mini batch. So the output from the a mini batch standard deviation will be passed to the convolution block and then passed by two fully connected blocks parallelly, that is separately. So one of the FC like fully connected blocks acts as an auxiliary classifier and another as an adversarial. So here the adversarial will classify whether the generated image is real or fake, and the auxiliary classifier will gives the class of the image which was generated. So by using the generator, some of the generated image samples of after training, some of the samples I took it from the images which was generated. So here are the images, 128 by 128 resolution. So here so since the original image size is of 512 by 512, but here I have used only 128 by 128, so there might be some loss of information, and the image is not quite most of the images are similarly generated, but not exactly as a human eye you can say most of the images are looking similarly, but if you clearly look into it, some images were not cannot be compared with the original training images. So here are the training loss and validation loss. Here are the here are the training loss and validation loss after the discriminator after the discriminated after the discriminators performance assessed as a classifier to classify the type of the tumor. So these are the results from those. Here I have achieved 98.57 percentage. So here when compared with other techniques, here the here this more here this discriminator pre-trained model has fewer number of trainable parameters when compared to other pre-trained like Resnet 50 or VGG 16 or some other which has more number of trainable parameters. But here the discriminator here the discriminator we have used the future here we have fine-tuned the here we have fine-tuned the discriminator of the fully connected layer of auxiliary block. So after fine-tuning we have less number of parameters when compared to other models, so which is pretty good. And also we have achieved better accuracy when compared to some other pre-trained models. And also thing is we can use this technique for generating high resolution MRI images, since MSG GAN has the capacity it can support 1024 by 1024 resolution of images, but it requires high computation power. Since our data is of 512 by 512, I have used 128 by 128, but if we can increase it to 256 by 256 or 512 by 512, I think we might the accuracy might get increased. So yep, that's it. Thank you for your attention. Any questions?
Thank you very much. We are waiting questions from our colleagues.
Hello, I have a little question. You said you used 2 databases, right?
Yes, 2 databases.
Have you encountered any problems with combining these databases into one learning data set?
So yeah, that's a good question. So when actually this second data set, this is just for differentiation between tumor and no tumor. Actually, if we see that this first data set is of T1 weighted CE MRI images, but the second data set is just T1 weighted MRI images, not contrast enhanced images. So some of the professors has reviewed this thing and they asked me similar question whether this whether my research is correct or wrong. Then after sitting for some time I my classification results have given no tumor with 100% accuracy. Why? Because it's like just T1 weighted, not T1 weighted contrast enhanced MRI images. So I couldn't able to find T1 weighted contrast enhanced images for healthy MRI brain scans. So it was one issue I faced it with, but I have to continue for differentiation. So after including this, like my model was predicted 100% accuracy for no tumor images. But like if we find some data set for T1 weighted contrast enhanced for no tumor, then we might like there might be decrease in, like, I'm not sure if they're like 99% or like 100%. I'm not sure. I think it will be 100%. Why? Because like tumored images will have like some pattern like of tumors, but like in normal healthy brain tumors, like we won't be finding that small portion of tumors. I think it will give 100%. But like as I said, like I couldn't able to find T1 weighted contrast enhanced images for no tumor. It's just for differentiation between no tumor and tumor. I have added this fourth tumor as for differentiation.
Okay, interesting. Thank you.
Thank you. One question from me. If you use a gun, this technology has problem with model collapsing when you obtain images which is close to each other. How you avoid this in your studies?
So like by using auxiliary classifier, we can avoid this mode collapse. So like if you there is one technique called AC GAN, like auxiliary classifier GAN. So from that I took like I have inspired from that paper along with MSG GAN. So actually this auxiliary classifier will try to help the discriminator to maintain to maintain good feature extraction and prevents like mode collapse. Especially also like it will make sure that our generated data maintain a condition of like whether like which kind of class you want to generate, like glioma or meningioma. So here this is helpful for preventing mode collapse.
Thank you very much. Thank you. It's very interesting presentation. And we go to the next speaker. It's Ishchenko Irina, digital 3D technology in the assessment of lymph-1 expression in skin eczema and type 2 diabetes mellitus. Irina, are you here? Could we check your sound? Say something, please.
Слышно?
Yes.
Thanks. I welcome all colleagues to our conference. I present to your attention a report on the topic digital 3D technology in the assessment of lymph-1 expression in patients with eczema, with skin eczema, against the background of type 2 diabetes mellitus. Due to the increase in the number of patients with type 2 diabetes mellitus, the problem of treatment of skin diseases occurring against the background of carbohydrate metabolism disorders remains relevant both for dermatologists and for endocrinologists. The development of dysfunctions of blood and lymphatic vessels in various tissues and organs plays an important role in the pathogenesis of vascular complications of diabetes mellitus. The lymphatic system is involved in the transport of tissue fluid, proteins, immune cells, lipid absorption, and cancer metastasis. Disorders of the lymphatic system can lead to edema and fibrosis. Recently, special attention has been paid to the study of the structures and molecular cellular mechanisms of the initial stage of the formation of lymph and the lymphatic system in various tissues and organs. And in our investigations, we show the role of pre-lymphatics in the development of liver function disorders in type 2 diabetes. Studies of the skin lymphatic system mainly concern lymphatic vessels stained with the LIM1 receptor. However, the morphological features of the skin lymphatic system and its roots, pre-lymphatics, in diseases associated with the presence of exudative processes in the epidermis and deep layers of the skin remain unexplored. Sorry, purpose of the study was to carry out research on the morphological features of various links of the skin lymphatic system of patients with eczema against the background of type 2 diabetes to assess the expression of LIM1 lymphatic vessel endothelial marker in the skin epidermis of patients using digital 3D modeling technology. Clinical observations of 3 male patients with eczema and type 2 diabetes were carried out. There were such methods: histological, immunohistochemical staining for LIM1 and 3D modeling of skin samples stained for LIM1 was performed using a nanolith laser holographic confocal microscope. Skin samples were taken from a lateral surface of the torso. And the next slide provides a step-by-step description of the indirect biotin peroxidase method. But I want to say that during all immunohistochemical reactions, negative controls were carried out, replacing incubation with primary antibodies by incubation with non-immune serum of the corresponding animal, rabbit serum. And about our results, all 3 patients who received treatment for eczema, concomitant diseases were type 2 diabetes, fatty generation of the liver. The skin disease proceeded with moderately pronounced clinical manifestations in the skin lesions from the unsharp boundaries were located on the back of the hands, inguinal, folds, and trunk. In the foci, against the conjunctive erythema background, infiltration of skin and ocular vesicles was noted. Moisture was absent in all patients. In the skin of the patients, we revealed an uneven thickness of the epidermis, thinned in places and having moderate acanthosis. Vasodilatation, thickening, and increased permeability of the walls and swelling of the endothelium were noted. There were pictures of edema, both intracellular and intracellular and intercellular, and in this case, the cells were separated, the intercellular connections were strained, and in some cases, torn. Separated patterns of acantholysis were observed. In some areas, spondylitic vesicles were detected in the stratum corneum of the epidermis. We noted edema, dilatation of capillary vessels in the papillary layer of loose fibrosis and connective tissue of the skin. Lymphatic vessels, their back arrows often started from the enlarged blind and and were disrupted in the dermis, forming a network of dilated lymphatic vessels. Cellular infiltrates were detected in the dermis. In the endothelium of the lymphatic capillaries, we detected the expression of LIV1 defined as a pronounced brown color, black rose. But we also detected the positive LIV1 signal in the cells of the basal layer and the spinal layer of the epidermis. There are blue arrows. That gives a reason to consider the intercellular spaces in this layers of the epidermis as pre-lymphatics. We observed intracellular edema, most pronounced between the cells of the spinal layer of the epidermis as a result of the penetration of serous exudative from the dilated vessels of the papillary layer into the epidermis. The phenomenon of spongiosis was noted, in which the cells were separated, the intracellular connections were strained, and in some cases broken. Under these conditions, some epithelial cells developed intracellular edema, which could later lead to the death. 3D modeling of the image of the LIV1 expression in the skin epidermis of patients with eczema and type 2 diabetes revealed clear boundaries of pre-lymphatics and confirmed that intercellular edema is most pronounced in the spinal layer of the epidermis. Using the nanolymph microscope software, we were able to measure the width of the intercellular spaces. The intercellular spaces can be up to 4 micrometer wide. Analyzing our results, the following facts should be taken into account. At first, LIV1 was the first identified as a cell surface receptor in lymphatic vessels. But now, to date, LIV1 expression has already been detected in sinusoidal endothelial cells of the liver, spleen, and lymph nodes in humans. Besides, then, with colleagues, using immunohistochemical and electron microscopic methods, revealed the LIV1 expression in the non-sinusoidal endothelium of many other organs, such as the lungs, heart, and adrenal glands of rodents. And as for LIV1 receptor functions, we can say that LIV1 serves as a receptor for Hyaluronan or dendritic cells, macrophages, T lymphocytes, and ensures their delivery to the lymphatic vessels. But for us, interesting that other function of LIV1. On the other hand, LIV1 as a hyaluronic acid receptor is involved in the sequestration of this molecule. As for hyaluronan, the main ligand for the LIV1 receptor is hyaluronan or hyaluronic acid, which is the main component of the extracellular matrix and is also part of the phycocalyx produced by the endothelium. Most of hyaluronan properties depend on the size of its molecule. There are high molecular weight hyaluronan and low molecular weight hyaluronan. The first molecule exhibits anti-inflammatory and immunosuppressive properties, and low molecular weight hyaluronan is a powerful pro-inflammatory molecule. So we determined the lymph-1 expression in the lymphatic vessel endothelium of both the papillary layer of the skin and the deep layers of the dermis. But we also detected a pronounced lymph-1 positive signal in the cells of the spiny and basal layers of the skin epidermis in patients with eczema and type 2 diabetes. Previously, we found that skin labeling of lymph-1 gives a pronounced positive signal in the cells of the spinal layer of the epidermis in patients with cutaneous mastitis. The results obtained in this study indicate that the cells of the spiny and basal layers of the epidermis may have lymph-1 receptors, due to which they participate in the lymphatic transport of hyaluronan in the skin of patients with skin oxidative processes. Our observation have shown that the intracellular spaces in the spiny and basal layers of the epidermis should be considered as part of the lymphatic bed, the pre-lymphatics. This is indicated by histological changes in intracellular spaces, intercellular edema, and epidermal cells intracellular edema, as well as the lymph-1 expression on the epidermal cell membranes of these layers. The development of such changes in the epidermis is facilitated by stagnation in the blood and lymphatic vessels of the papillary and reticular layers of the skin dermis, and by the formation of perivascular infiltrates. 3D modeling of the image of the lymph-1 expression in the epidermis of the patient's skin revealed clear boundaries of the pre-lymphatics and helped to establish that intercellular edema was observed throughout the entire depth of the skin section and was most pronounced in the spiny and basal layers of the epidermis. In the spiny layer of the epidermis, this led to the loss of communication between epithelial cells and their death in limited areas. So our study allowed a new look at the molecular characteristics of the lymphatic system of the skin of patients suffering from eczema and type 2 diabetes mellitus. It was found that the epidermal layer of the skin, although located at the junction between the external and internal environment, plays a crucial role in maintaining the homeostasis of the skin of patients with eczema and type 2 diabetes. The detected expression of the lymph-1 lymphatic vessel receptor in the cells of the spiny and basal layers of the epidermis, the use of tomographic analysis of preparations and 3D modeling of images of the epidermis and skin prolymphatics indicate that these prolymphatics are involved in the lymphatic transport of hyaluronan in the skin of patients with the presence of oxidative processes. At the same time, the functions of the epidermal layer of the skin are largely coordinated by the work of various parts of the lymphatic region of the skin, blood vessels, and connective tissue compartment of the skin. Thank you for your attention.
Thank you very much. We await some questions. We have time for 1 or 2 questions. I want to ask you about statistics. You present some histology slices, but could you provide statistics and information about, yeah.
I could say that in this article, we only prepare the information about this about these facts. We haven't statistics.
Okay, thank you. Any questions? Okay, in this case, we go to the next presentation. It's presentation of Markov Nikita. Heart rate variability reveals two phenotypes of atrial fibrillation. Let's start, please.
Okay, thank you. One moment. Good evening. I'd like to thank you, the chair of this conference, to provide us with an opportunity to present a report on our study, which the name suggests is a disease classification study. The object of our research is.
Excuse me, could you hide part of Zoom panel from right side of your screen?
Yeah, of course.
Okay, thank you very much.
The object of our research is atrial fibrillation, which is the heart condition that causes irregular, abnormally fast heart rate. It is known to be the most widespread arrhythmia in human population, and generally the attention of healthcare professionals is focused on the paroxysmal form of atrial fibrillation, since proper drug therapy and interventional methods can lead to rhythm restoration for patients. Since atrial fibrillation directly influences the heart rate, we believe it is possible to derive phenotypes of this disease using metrics of heart variability known as HRV. Okay. The aim of the study is to define paroxysmal atrial fibrillation phenotypes based on HRV indices derived from long-term electrocardiogram recordings. For this research, we used the largest open database of auto ECGs with atrial fibrillation episodes called long-term AF database. It consists of 84 recordings ranging from 24 to 25 hours. At the bottom of the slide, you can see snippets of recordings from the database. For each recording in the database, we calculated a vector consisting of 52 HRV indices that includes a comprehensive set of nonlinear measures. These indices indicate the influence of parasympathetic and sympathetic nervous systems on heart rhythm regulation. It should be noted that HRV was calculated on normal sinus rhythm. After calculations for each record, the data analysis pipeline was established. It begins with dimensionality reduction with principal component analysis, which allowed us to explore the component space. Other pipeline steps include outlier detection and hierarchical clusterization of subgroups, and then subgroups were validated with statistical tests. At the first stage, we conducted the dimensionality reduction using principal component analysis. This slide demonstrates a truncated pair plot of the 2D component space projections. On the diagonal, you can see distributions of each component. Can I draw on the slide? Yes. On the diagonal, you can see distributions of each component. Above the diagonal are the scatter plots of 2D projections where each dot represents a feature vector. And below the diagonal are the density plots of 2D projections. As note, this data suggests the existence of two possible attractor points, which led us to assume that feature vectors can be divided into two clusters. Okay. For the next step, we conducted the outlier detection with the method of voting. We utilized 4 most popular algorithms of outlier search, and if a vector was found to be an outlier by at least 3 methods out of 4, they were excluded from the data. On the left, you can see projections with marked outliers. On the right, the filtered outlier-free distributions. Oh, I'm sorry. After dataset filtering, we conducted another dimensionality reduction step, converting the space to 5-dimensional components. Yet again, we can see the attractors on the density plots that suggest 2 subgroups. Next step, we used hierarchical cluster analysis with Ward's criterion, which confirmed the presence of 2 subgroups in the data. Clusterization datagram is presented on the left, and the right figure displays the distribution of clusters on the projections, which is the result of clusterization. With subgroups derived, our next step is physiological interpretation of phenotypes. For this, we examined HRV characteristics of 2 derived subgroups. Using visual and statistical analysis such as Mann, Whitney, U-test, and T-test, we found out that HRV indices differ between clusters. The first subgroup demonstrated a convincing vagatonic profile in heart rhythm regulation, which is confirmed by increasing parasympathetic indices. For example, PNN50 and RMSSD. Can I draw? Yeah. So you can see the increasing. You can see the increase. The growth of measures of parasympathetic activation in the first subgroup correlated with the increase in nonlinear indices. These patterns confirm physiological notions about parasympathetic response that forms in case of background vagatony, which sustains fibrillation paroxysms. The second subgroup was characterized by increasing values of sympathetic indices such as CSI and CSI modified, and low values in the group of linear indices. These are sympathetic indices, and you can see in the second cluster lower values. Okay. As a result, our model verified by the differences in HRV allows the prediction of total atrial fibrillation duration. Median AF duration in records of the first phenotype is 21 hours, when in the second phenotype, median duration is only 4.5 hours. This data wasn't included in the baseline analysis, which confirms the existence of links between HRV indices and duration fibrillation paroxysms. And with this, my report comes to an end. And we can draw following conclusions. First, inclusion of the extended set of nonlinear HRV indices allowed us to extract groups of heart rate asymmetric correlation with branches of sympathetic and parasympathetic activation. And the group of recordings defined by vagatonic heart rate regulation displayed longer duration of atrial fibrillation paroxysms. It should be noted that limitation of our work is that we use ECG from the blind database that lacks clinical data. And with this, it appears to be promising to conduct a research and aim to find correlations between electrophysiological data and clinical data of patients. And that is all. I'd like to thank you for your attention and hear your questions if you have any.
Thank you very much. We await some questions, and I ask Vasily to ask something because he experienced in this topic. Vasily, are you here? Okay. In this case, I ask only one question. Do you try to use LDR, linear discriminant analysis, to this data? Could you separate these 2 classes with linear rule or only with non-linear surface?
Yes, we attempt to confirm the existence of these subgroups with linear discriminant analysis, as well as another method, linear SLC. And at this point, this result will probably be expanded in the subsequent research. And at this point, I would say that results aren't as concluding as I'd like to, but maybe they can be improved.
Okay, thank you. We are waiting for the next speaker, Kirill Slasnikov, review of instrumental methods for detection the respiratory signals. Kirill, are you here? Okay, we finish too early with this presentation. Maybe speaker can't come to us. We will wait sometimes. Okay, I ask again. Review of instrumental methods for detection the respiratory signal. Okay, maybe the next speaker, Lukin Anton, methods for biomedical information, extraction of immunosuppressive cell properties. No answer. Okay, and this time we still wait a little bit more. We have some times for discuss about part of our section about presentation. Maybe some people have opinion about topics and presentations. Okay, Kirill, hello. We are waiting for you. I see that Kirill comes to us. Kirill, are you here? Maybe something not working. Anybody, could you hear me? Something goes wrong from my point of view, because nobody answer me. Maybe we have issue with sound, but I know how to check it. Kirill, are you here?
Yes, hello.
Okay, good. We are waiting for you. Maybe we start with your presentation.
Yeah.
Yeah, very good. Review of instrumental method for detection the respiratory signals. Let's start, please.
Do you see my presentation?
Unfortunately not. We see the black screen. Oh, yeah, okay, good.
Oh, okay, okay.
Run the presentation mode, please.
1 second, please. Do you see my presentation now?
Unfortunately, not now. We see the black screen.
And right now too?
Nothing changes.
Yes, we see.
You see?
It's the presentation, yeah?
Yes, yes.
Okay.
Review of instrumental methods for detecting the respiratory signals.
Maybe you try to rerun your share, because I see nothing, and translation on YouTube also don't show your presentation.
Okay, can I stop my video
Stop, yes, stop
translation?
and try again. Okay, in any case, we have some times because we goes a little bit faster than usual. Kirill, what do you see? Could you start presentation, please?
I'm trying.
Okay.
Oh, I start my presentation. Do you see it?
I don't know about others, but currently I see the slide in PowerPoint, but not in the full screen mode.
Yeah. Okay. Okay.
Okay, maybe you close the Zoom panel from the right side of your screen, and we see presentation from this from PowerPoint, because we spend some time.
How can I close the Zoom panel?
You have a button, small button, which reduce the size of this panel. On the right side of your screen, you have a face and participant of our meetings.
Yeah.
Could you press on the small button on the top of this panel for make it smaller? Great. And let's start, please.
Okay, hello everyone. Today I want to talk about instrumental methods for detecting the respiratory signal. One second, please. Do you hear me now? I have some problems.
Yes, let's start.
Oh, okay. Oh, okay. Due to many respiratory diseases, human respiratory wave detection is a very urgent problem. Especially strongly, this problem is expressed in the frequent occurrence of obstructive apnea. The prevalence of apnea in newborns and preschool children is estimated in range from 1 to 3%. The present report provides an overview of existing methods of detecting a human respiratory wave, namely MRI with the respiratory.
Okay, we have trouble with your sound and with internet from your side, maybe. Kirill, are you here? Could you repeat the last slide, please? Okay, today is day of big technical problem with speaker. Something really goes wrong. We await our guest little bit more. If presentation goes too long, we bring this presentation in the end of our section.
Sorry, I have a problem. I log out from my Windows account.
Okay, let's start your presentation and go faster, little bit faster.
Mm-hmm. Evidence is increasing, suggesting the frequent occurrence of obstructive sleep apnea in children. Apnea is stopping respiratory movements. In particular, it can be observed with the depletion of blood with carbon dioxide caused by excessive ventilation of the lungs. Apnea can also be caused in the experiment by a strong increase in arterial blood pressure, which excites receptors of some vessels. First, I want to tell about the respiratory belt. The respiratory belt is a belt with a defined sensor. As an example, a tensor resistor may be selected. The essence of the strain gauge belt is to change the resistance as the length of the belt increases. The respiratory belt is often used as a supplement to MRI and to improve the quality of the output image. The advantage of this method is the cheapness of manufacture. The disadvantage are low reliability, low sensitivity when determining breathing phase and uncomfortable when using. There are also interesting acoustic methods for estimating respiratory rate. For example, Jovan de Carvalho with his team offer an acoustic method for estimating breathing frequency using a sound recording device. In their method, readings of the breathing rate of cattle were obtained, but it can also be applied in the case of assessing a person's breath, of course. In their experiment, MP3 audio recorders was installed on cow gatekeepers. One observer scored the flank movements of the animal for 15 seconds using binoculars, and the second observer controlled the stopwatch for marks on the number of movement and exact time. The number of flank movements was by 4 to obtain a respiratory rate in the quantity of breaths per minute. The test results showed no important difference between acoustic and visual methods. The results of similarity analysis showed that the more accurate technique is acoustic, since the data were densely scattered along the least squares line. The advantages of this method are easy to use, relatively cheap method, and small dimensions of object. The disadvantages are difficulty removing interface from parallel operating devices, low sensitivity at specific patient positions. One way to access breathing is to use a thermistor or nasal pressure transducer. The method consists of placing a thermistor or a nasal pressure transducer in the area of the nostrils. The principle of operation of the thermistor is to change the resistance from temperature. The nasal pressure transducer consists of a nasal cannula and pressure transducer directly. The sensor can be based on tensor resistors. To compare the thermistor and the nasal pressure transducer, a group of scientists conduct tests. The results suggest that the use of thermistor separate may be incompetent to determine the full scale of sleep-related respiratory disorders and may result in some patients being left untreated as a result of some respiratory cases not being recognized. Also, as a result of research, it can be said that the nasal pressure transducer is more accurate than the thermistor. The advantages of this method are cheap manufacturing, easy to use, small dimensions. The thermistor has high accuracy when working with adult patients. The transducer has increased accuracy when working with newborns. The disadvantage of this method is unstable position on the human body and uncomfortable in use. Using a video signal to recognize a respiratory wave is a new modern way. Various devices can be used as a video signal: RGB camera, depth camera, and thermal camera. One method of brief detecting by the RGB camera is to place the pattern on the body or cloth. The pattern is shown on figure 2. The acquisition of respiratory rate data consists of tracking the change in the average distance between lines or pattern points. The advantages of this method are the cheapness of the cameras, easy to use, high accuracy, and compliance with the rules of use. The disadvantages of this method are the necessary of marking on the patient's body or clothes, the necessary good lighting, and necessary to keep the patient in a certain position. To remove the untreated brief signal from the cloud of depth points, the highest and smallest distance between the subject and the camera was calculated. This average distance varies during the movement of the subject's chest, proportional to respiration. The advantages of this method are high accuracy, easy to use. The disadvantages of this method are high price relative to previous methods, complexity in setting up, and the need to keep the patient in a certain position. The processing of the signal from the thermal camera consists of tracking the temperature of the nostrils. The method is similar to a thermistor and a nasal pressure transducer, but is a non-contact method for evaluating respiratory rate. The advantage of this method is easy to use, but the disadvantages are the highest price relative to all methods considered, and the least accurate method relative to other methods with video signal processing. Currently, new cameras have appeared which include RGB, depth, and IR cameras. With such cameras, we can realize the three-dimensional surface of the patient's torso. This method uses depth information obtained by two RGBD cells at different viewing angles, taking into account the limitation of the intensive care unit. An example of the correct locations of the cameras is shown in figure 3. This method allows to measure the respiratory rate with great accuracy and expand the set of obtained data by adding to it the volume of exhaled air of the patient. The advantages of this method are the highest accuracy relative to all chambers, easy to use, no invulnerable breath detection zones. The disadvantages of this method are high price relative to previous methods and complexity in setting up. In conclusion, therefore, it can be mentioned that the problem of determining the brief is quite large, and methods are now emerging to solve this problem. It's also worth noting that the methods discussed can vary depending on the task. That's all. You can ask the questions for me if you want, and if you have.
Okay, we wait some questions. And one question from me: what is your result in this areas? What are you doing?
In this article, I also make a review of different methods, but in future, I want to take two RGBD cameras and try to create a three-dimensional area. So it's need to get a respiratory rate, and that's it.
Okay, thank you. Thank you very much. The next presentation is Anton Lukin, method for biomedical information extraction of immunosuppressive cell properties. Hello, let's start.
Hello.
Sorry, you disable your microphone.
Oh, hello. Hello, everyone.
Yeah, good.
I hope you see our presentation.
Yep.
Nice. So, hello everyone. The topic of our work is method for biomedical information extraction of immunosuppressive cell properties. In the human body, many cell populations can regulate immunological processes negatively. These properties can be possessed by cells of the immune system and other types of cells that implement their immunoregulatory potential in interaction with the immune system. Data on the immunosuppressive properties of cell populations are presented in dozens of thousands of scientific publications in the form of in vitro experiments, animal model experiments, and clinical studies. Automating the search, generalization, and extraction of information from these articles is a fundamental scientific problem. It is necessary to develop methods for extracting certain types of entities from texts, such as names of cell populations, the process of suppression itself, and to filter statements that are not a description of the immunosuppressive properties of cell populations. It is useful to perform a meta-analysis in that field to build a research landscape of the field and eventually to reveal prospective research direction in the field. So our main goal was to develop a method for identifying descriptions of the immunosuppressive properties of cell populations in biomedical texts. And the main problem is the labeled corpus of limited size that we use. We have also compiled an overview of modern methods of extracting information from texts to determine the approach of extracting. The works reviewed used the following methods: dictionary search, manually composed rules of extraction. I think it's simple. Sequence labeling classifiers. It's algorithmic assignment of a categorical label to each member of a sequence of observed values. Tokens in sentences, in our case. And extracting information using the sequence of interest of such labels. Another method is a convolutional neural network, a subtype of multi-layer neural network that can be used to work with the language constructs, but it's usually used for working with audio data and images. And BioBERT model, a model based on the transformers architecture, pre-trained without a teacher on the open corpus of medical publications from PubMed Central, which allows taking into account the context when extracting information. Today, this model is widely used in the field, but unfortunately, we don't have enough data to fine-tune it. We proposed a hybrid approach that uses dictionaries and a rule-based parser to identify candidates in an entity and then filter them using a pre-trained machine learning model. We consider all available external linguistic resources, such as MeSH, models for generating vectors, vector representation of lexes, word embeddings, pre-trained on biomedical texts, dictionaries of cell names to solve the problem of the insufficient number of labeled texts. After that, syntactic and semantic features are used to identify relationships between entities. The developed method allows one to extract information about the target cells, effector cells, diseases, and descriptions of immune suppression. The method begins with the revealing of linguistical features of the text. At this stage, there is an LP library is used for morphological, syntactical, and relational situational analysis. Dictionaries containing cell names, rules that take into account lexical, morphological, and syntactical features, candidates for the mention of target cells and effector cells are identified with the extracted linguistic information. The dictionaries include more than 200 variants of names of cell populations. Those names can be comprised from several tokens like mesenchymal stem cells. The extraction has the following steps. Using syntactic dependencies, all possible combinations of tokens are built that can constitute a mention of a named entity in the text. Then we found the combination that is the most similar to the names from the dictionaries. The pre-trained fast text model was used to account for synonyms instead of explicit vocabulary matching. The mesh ontology mentions of diseases are identified. A slightly different approach is used to identify suppression descriptions. It is performed with searching for syntax predicate words semantically related to the identified entities. The next step was to select proper sentences that contain all the entities of interest. Such a sentence must have an effector token, a target token, and a suppression description token. A basic information extraction method was used to evaluate the proposed method. We will refer to that method as a baseline. The method uses the dictionary and mesh ontology and doesn't take into account additional linguistic features. Experimental evaluation of the presented method was carried out on a small corpus containing many annotated scientific publications. On this slide, you can see a table that contains the result of the assessment. The results show that the proposed method is significantly superior to the baseline, especially in terms of precision. That means syntactic and semantic features are of great importance in solving this problem. Nevertheless, the relatively low recall indicates that additional expansion is needed for both vocabularies and the labeled training corpus. Using the proposed method, we also analyzed like 48,000 unlabeled annotations of publications from the PubMed research describing the immunosuppressive properties of cell populations. According to the results of processing this unlabeled corpus using the proposed method, references were in approximately 80% of annotations, and the help of the basic one in 100%, which is consistent with the precision estimates obtained on the labeled corpus. Therefore, the approach helps to filter out about 20% of irrelevant publications. And conclusion. The method of information extraction proposed in this work makes it possible to solve the problem of identifying descriptions of the immunosuppressive properties of cell populations in biomedical texts with a sufficiently high quality. A labeled corpus of limited size and many external linguistic resources were used to train the method. As part of further research, we will expand the dictionaries of cell populations names and the labeled corpus of texts and implement filtering of false positive examples using sequence classification method. Thank you for your attention. Questions?
Thank you very much. We await questions for this to speaker. Okay, some questions from me. I see that you present some accuracy, some precisions and F1 scores, but could you present results of your system which they show and how it works, what kind of result you can produce? And also, I'm interested, could you have yeah, okay, it's markup, but could you present a little bit more results and explain how to use this system for people who work in this field?
Actually, we have another image, but I'm not sure I can open and present it right now. But it looks similar. It's that you can see it's a part of this image, and this we use for comparing our results. So this is actual markup, and like this we can present our extraction. We actually can convert our data to broad format to present it in broad utility.
Yeah, may I add one word? I've worked
of course
on that project together with Anton. This method can be used together with biomedical information retrieval systems, online services like PubMed Central. So the researcher can perform some search and obtain a lot of documents, and a lot of them could be not really relevant to their query. I mean, in a specific field like immunosuppression. And this method can help them to filter irrelevant results. After that, the extracted information like types of immunosuppressors and cells can be also used to build associative rules and to extract which is the most frequent and the most rare combinations of those entities in the which have been considered in all biomedical research. And so they can find out some new, I don't know, combinations which should be studied more intensively.
Okay, I allow me some discussion with this. Like you notice, the PubMed is also information retrieval system, and your analysis provide some information for immunosuppressive properties, but it's a lot of work for different types of for different field of physiology and physiological studies, for not only for cell but also for oncology, for some properties of cardiac cells, and for many others.
Yeah, I totally agree.
Yeah, and what's your impact? What you provide new to this? Except, of course, I understand that you provide better algorithms that was proposed before. But I see some troubles here because you provide only F1 score, and markup may be measured with other metrics, which may be not so good in more complex topics. And also, I'm interested how your system overcomes the basic information retrieval. Could you provide some ideas about this?
First of all, we have compared our solution with some baseline implementations with PMC and with additional filters which uses only information from dictionaries. So it significantly overcomes it, and it helps to reduce, for example, output of PMC up to 20%. So a researcher could reduce amount of their work while they're studying this field. Also, I have to say that this method could be generalized in the future to cover our fields, and we see the way how to do that. And after that, also it could be tested in that field, so we could find out benefits of this method in our fields.
Thank you. Thank you very much. And the next, we go to the next presentation. Elena Rimskaya, development of a mobile application for an independent express assessment of pigmented skin lesion. Let's start, please.
Hello, dear colleagues. 1 minute, please.
Okay, we see your PowerPoint. Okay, and now we see your presentation. Let's start, please.
Okay. I want to tell you about my work, development of mobile application for express assessment of pigmented skin lesions. Skin melanoma have been more prevalent in the previous decades and therefore have become a very significant public health problem. Our 90% of the death cases from skin cancers attribute to melanoma. A steady increase of mortality of melanoma is observed all over the world. Often the patient turns to the doctor when the tumor is widespread or is observed for a long time by dermatologists, cosmetologists, and surgeons with an erroneous diagnosis, and accordingly receive erroneous treatment that can stimulate the development of the malignant lesion. Today, considerable experience has been accumulated in the early diagnosis of skin melanoma using visual and instrumental studies of clinical sites of pigmented skin lesions. A person is not able to conduct an expedient examinations and evaluate the pigmented skin lesions present of his skin, since he doesn't have necessary knowledge in this area of medicine, and the areas of examination may not always be available. For example, the back. For this purpose, it is possible to use modern mobile devices with digital cameras for self-examinations of the skin. There are prototypes of such devices, and now mobile applications for smartphones are also available, which helps the user to conduct self-examinations of pigmented skin lesions. For example, Skin Vision, My Skin Pal, Skin More Analysis, and another. The main emphasis of this mobile application is placed on photographic documentation and user-friendliness of the interface, while image analysis is mainly entrusted to the user, who must identify all the changes that occur with the lesion over time. The ability to perform any kind of analysis requires minimal standardization of photographic technical conditions. Therefore, mobile applications either don't have automatic analysis or they work with a limited number of mobile device models. At the same time, the results of automatic analysis, then using mobile devices with a built-in flash, are very experimental, since most of the clinical parameters of pigmented lesions necessary for early diagnosis of signs of tumor malignancy are not measured. Automatically, usually only the features of the border of the skin lesions and the uniformity of this pigmentation can be assessed without methodological support for measuring the parameters of lesion, which is clearly not enough to create mobile application of automated image analysis. The sensitivity and specificity of such application doesn't exceed 70%, which is significantly lower than that an expert, doctor oncodermatologist. Therefore, the task of creating a mobile application for necessary the main parameters of pigment skin lesions important for early primary diagnosis of skin melanoma the accuracy of which exceeds 90%. It's for expert. The capability of modern mobile application, including higher functionality, speed, and growing popularity in everyday life, we used in this work for rapid assessment of the state of human skin and early detection of signs of skin melanoma. The mobile application implements the possibility of registering optical images, assessing the image quality, recognizing and processing a special color standard, recognizing a pigment skin lesions, measuring its parameters, including diameter, area, color, sharpness, shape, as well as calculating the probability of malignancy and formation for a conclusion for contacting a doctor. It then analyzes this data. The probability of malignancy is at least 60%. The structural and functional diagram of a mobility application for self-assessment of the state of human skin is shown on slide. The developed method of segmentation and recognition of skin lesions allows to determine its boundaries with a high accuracy, with a key factor for further analysis of parameters of pigment skin lesions. This mobile application for self-express assessment of pigment skin lesions based on digital processing of optical images was developed for mobile operation system iOS and Android. An image on a pigmented skin lesion is recorded by a smartphone camera with a built-in photo flash simulated with a special color standard at a distance of 10 centimeters, after which the reference and its parameters are searched for. The segmented areas of the circles were used to determine the red, green, and blue components of color coordination in RGB space of each of the 5 areas to correctly reproduce the color characteristics of the formation under various certain conditions, as well as to determine the scale of the image. At the next stage, a pigmented lesion is detected and its measured parameters are determined. Maximum and minimum diameters, area, color, sharpness, shape, and the final stage, the probability of malignancy is calculated. A characteristic feature of the developed application is the use of a special color standard with the user print before examining the lesions. The standard has a simplified version. It is printed on paper after celebration of the printer's color rendering. The standard has the shape of a circle, each with a diameter of 20 millimeters, inside of which 5 multicolor circles with a diameter of 4 millimeters are located at the same distance from each other, which are a size scale for determining the diameter and area of a lesion. And 5 multicolor circles are designed to correctly convey the color characteristics of a lesion. The choice of the colors of the reference is tied to the output color space of a digital camera. To determine the resolution of digital image, the average effective diameter of 5 regions on pixel per millimeters was calculated. The main advantage of this application is color calibration against with a standard which is responsible for bringing all images to a single color rendition. Then the errors in color reproduction when printing a standard on an injector laser color printer is no more 3%, and the error of reproduction of geometric dimensions is not more 1%. The approbation of the mobile application for the rapid assessment of pigment skin lesion was carried out on 520 real images of skin lesions in vivo. The study was conducted with the written consent of each patient. The study included an automated analysis of 3 test groups of optical images of pigment skin lesions. The clinical diagnosis of the disease was confirmed by a histological study method. Group 1 includes 274 images with a confirmed diagnosis of benign skin lesion. Group 2, 235 images of precancerous skin diseases. And group 3, it's 11 images of melanoma. The calculation of the parameters of pigment skin lesion using a mobile application was carried out for each of 520 images of these 3 test groups. The parameter of skin lesions of all images were previously calculated manually using the visual diagnostic method. The result of the study made it possible to calculate the efficiency parameter of the developed diagnostic method for the rapid assessment of pigment skin lesions with a reference diagnostic method. Expert opinion confirmed by historical examination. The sensitivity of the method was 95%, specificity 91%, and accuracy 93%. Thank you. Do you have questions?
Yeah. What's you explain mobile application, but what's new in and what's novelty of your methods for processing?
And you in this method that I calculation all of the parameters of skin lesions, and I develop segmentation method which 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 recognizing and processing with a special color standard. It's a new my new in my applications, and
Okay, thank you very much. We finished our section, and I'm glad you. I want to say that I'm very glad to see you here. It's very good that it was a section with very interesting presentation, very interesting results, and currently we goes to some break until the next part of our conference. Thank you very much.
Раз-два. Василий Ильич. Василий Ильич, вы тут? Сейчас. Okay, hello everyone. Start a new session. That's today, which is called the Computational Modeling in Physiology in Medicine. And so we got the reglement for 15 minutes for the talk. And the first speaker out, and we begin with the second speaker according to the program. That's Balakina Vikulova, Italia, Institute of Immunology and Physiology, Ekaterinburg, Russia. So you're welcome. Everyone, I think, is familiar
Thank you.
with Zoom, and you can switch your screen.
Thank you, Alexander. Good evening. I would like to thank organizing committee for the opportunity to demonstrate the results of our work. One of the areas of research in our lab of mathematical physiology is the study of mechanisms of mechanoelectric feedback in the heart. The influence of myocardial mechanics and calcium and electric events in the cardiomyocyte are important for myocardial self-regulation. Numerous experiments on laboratory animals have shown the presence of cardiac mechanoelectric feedback, but such experiments on the human myocardium are obviously difficult. It is obvious for all us that mathematical models allow us to partially overcome these limitations. They allow us to reproduce experiments in vitro and make assumptions about the possible mechanisms of observed phenomena. The results I'll show is an extension of a large research published in 2020 in the Journal of Physiological Sciences. We developed a mathematical model which combines 10^-panphenol electrophysiological model of human ventricle cardiomyocytes with the model of the cardiac muscle mechanical activity of our original development. The key feature of the model is the description of intercellular mechanism underlying direct and feedback contours between mechanical and electrical activity of cardiomyocyte. The model is a system of nonlinear ordinary differential and algebraic equations. The slide shows principal equations of the model, which describe the link between action potential generation, intercellular calcium dynamics, and force generation. The mechanoelectric feedback is based on the cooperative mechanisms of formation of calcium troponin C complexes, which play an important role in the activation of myofilaments and force generation. We use our model to simulate the consequences of the ventricle contraction phases during cardiac cycle. In the left panel, you see the dependence between left ventricle volume and left ventricle pressure. And cardiac cycle phases are isovolumetric contraction, the phase of ejection, isovolumetric relaxation, and returning to the diastolic level. Plot of the ventricle volume against the pressure developed during the cycle, so-called pressure volume loop, is conventionally used to characterize cardiac contractility. The area of PV loop allows one to estimate mechanical work produced by ventricle during the cycle. Ventricular contraction phases are closely associated with periods of electrical activity of the heart chambers, seen as ECG. Thus, within the framework of the model, we try to answer the question: how will the work of the heart changes if mechanical conditions of its contraction change, either due to a change in preload or afterload? Also, how the work of the heart muscle changes in heart failure. In experiments on isolated myocardium, to evaluate a similar relationship, length force loop is used. The area of LF loop defines the preparation's work. In experiments, a heart muscle preparation placed in the experimental setup and consequently goes through the physiological sequence of loading indicated by letters on the left panel. They are isometric contraction at fixed initial length, isotonic shortening down to end systolic length. C is for isometric relaxation at the fixed end systolic length, and D is the final stretch back to the initial length. In our paper in the Journal of Physiological Sciences, we have already simulated separately isometric and isotonic modes of human cardiac muscle contractions. In the right figures, you can see two relationships that combines the effect of diastolic length, preload and afterload on the contractility of the cardiac muscle and duration of the action potential in cardiomyocytes for different two lengths and for different afterloads. So we combine isometric and afterloaded contraction in physiological mode of muscle contraction and estimate the mechanical work it produces. Among the few experimental data obtained in biomechanical experiments on human heart muscle preparations, we found a record of the force loop cycle in the article of Holubash, which was made in 1998. In the left panel, you see the experimental record of the force developed by left ventricle muscle preparation and its shortening. We repeat the experiment in human cardiomyocyte model and obtained the results consistent with the data reported for the muscle preparations. In addition, we obtained the time courses of the calcium transient and the action potential and they are both proved to be load dependent in the model. The smaller afterload, the bigger the duration of calcium transient and action potential. All phases of physiological mode are noted by numbers. For all work loop twitches, we plotted length force loops, different colors. The dashed line on the envelope of LF loops shows the relationship between the end systolic length and the corresponding level of the afterload. This time reveals isotonic length force relationship. We calculate the area of each left loop. These areas are equal to the useful work performed by the virtual sample at given afterload during the shortening. The dependence of the work on the afterload is shown in the middle panel. In our experiments, we obtained peak value of the work obtained approximately at load equal to 40% of peak isometric force. As we will see further, this result is in good concordance with experimental values. The dependence between action potential duration and afterload is coincided with isotonic one. This shows that the action potential duration is defined by contraction phase, not relaxation. Returning to Holubash's paper, in their work they compared mechanical activity of failing and non-failing samples of left ventricle myocardium. In particular, they compared work performed by them. Heart failure is a pathological state characterized by weakening of the heart contractile function, and molecular and cellular disorders in the heart failure are diverse. Experimental data have shown that the most influential mechanisms of contractile loss and disturbed excitation contraction coupling in human heart failure related to pathological decrease in calcium content in the cell. In the model, such decrease in calcium can be obtained by weakening the capacity of sarcoplasmic reticulum. For that, we reduce maximum velocities of the sarcoplasmic reticulum, calcium pumping and release by 40 and 30% respectively. We have seen that in so-called failing cells, this change leads to a decrease in the peak calcium transient and diastolic concentration in the sarcoplasmic reticulum at each length we take. We simulate experiments with length force loop contractions of non-failing and failing cardiomyocytes at different diastolic lengths. The workload relationship obtained experiments is shown on the right panel. On the left panel, simulation results are shown. The main finding of experiments on isolated human preparation was that the peak of developed isometric force and maximum performed work is different in normal and failing human myocardium at the same initial length. Our simulation qualitatively repeats these findings. For simulation results, we normalized force values on the peak isometric force seen on the axis in the series of simulation. We obtained the maximum of isometric peak force at initial length equal to 95 of L max, and work values are normalized on maximum work value seen at the initial length equal to 90% and under the 40% of afterload, 40% of maximum isometric force. So we see that failed muscle samples always develop smaller force than normal ones for the same initial length. The decrease in maximum work, this decrease, performed by the failing sample compared with normal one is about 32–35% and may be explained by the decrease in the extent of the afterloaded shortening due to the smaller intercellular calcium available for formation of calcium troponin C complexes. The left panel shows end-systolic length and force relationship. For each initial length, the failing virtual sample, shown by solid lines, becomes much steeper than for the normal ones, shown by dashed lines. As already said, if we take a particular afterload, the horizontal line, relative end-systolic length for failing cell is less than for non-failing one. The mechanoelectric feedback and changes in calcium reveals itself in action potential changes in failing myocytes. On the right panel, you see the relation between action potential duration and the level of afterload. So both non-failing dashed lines and failing cells solid lines at different loads. the action potential duration of failing cells is always greater than non-failing ones for the same length at each afterload. So the conclusions are as follow. We use our model of electromechanical model of human ventricle cardiomyocyte to simulate work loop contractions in human cardiac muscle at normal and failing myocardium. Our simulation of the muscle contractions in physiological mode and calculated work developed by non-failing and failed muscle in the model are in good concordance with experimental data. Both calcium and mechanoelectric feedbacks can be clearly seen in human cardiomyocytes in the work loop twitches. The model predicts and explains increase in action potential duration and decrease in calcium in the cells in failing ventricle myocytes. You see the list of members of our lab on the slide. Please visit our lab webpage to find out more about the research carried out by our laboratory. Thank you for your attention.
Okay, thank you, Natalia. So questions, please. We got a couple of minutes. All right, you're welcome for your questions. So actually you can type your questions in the chat. So, okay. So the first talk without questions, it's okay. So, thank you very much. The next speaker is Dokuchaev Arsenii from Institute of Immunology and Physiology, Yekaterinburg, Russia. You're welcome.
Hello, everyone. Do you hear me?
Yes, we do. So we see your presentation.
Okay, thank you. My report will be about the comparison of His package and biventricular pacing in a patient-specific computer models. Cardiac resynchronization therapy is one of the effective treatments for chronic heart failure. Conventional setting of a CRT device suggests the implantation of two pacing electrodes, one into endocardium of a right ventricle and another on epicardium of a left ventricle lateral wall through the coronary sinus. However, about 30% of the patients do not show any clinical improvements and do not respond to this therapy. The reasons for this may be, for example, an extensive infarction scar field, the lack of biventricular and intraventricular synchrony, and non-optimal electrode location. Pacing of a cardiac conduction system here is a promising alternative to conventional biventricular stimulation and can restore physiological activation and reduce desynchrony. This study is an attempt to evaluate the effectiveness of His Packing system pacing and to compare characteristics of ventricular activation and with different types of stimulation. In our research, we used data from 27 patients with related cardiomyopathy and left bundle branch block who underwent the implantation of CRT device. The data was provided by Almazov National Medical Research Center and included computer tomography data and the ACGI data recorded from unipolar leads of the AMICARD system. This slide shows a general modeling pipeline consisting of 5 stages. The first 2 stages are the processing of computer tomography data and semi-automatic segmentation of surfaces of lung, torso, and heart. which is needed to build a volumetric model. Stage 3 was concerned working with only with heart. The important sub-stages here are assignment of myocardial fibers, the fitting of a heart conductivity to personify the model, and the choice of the stimulation protocol, and the calculation of the activation map. Then all of this data was incorporated into the volumetric model of the torso and the potentials on the torso surface, and the corresponding ACG signals then was calculated. So as you know, the electrical excitation of the heart propagates along the fibers faster than across fibers, and the setting this kind of anisotropy is important for solving electrophysiology problem. Since we do not have any data about real direction of the fibers for a particular patients, we used an algorithmic rule-based approach to assign the fibers. To obtain an activation map at each point of the heart, we solved the equanal model. And the information about tissue anisotropy was incorporated in conductivity tensor D in equation. The anisotropy ratio was set as 2 to 1. So solving this equation, equanal equation, we get an activation map. Then at each point of the heart, we assign the pre-calculated action potentials shifted in time depending on the activation time of a particular point. So for points with a larger activation time, action potentials was shifted by a larger period of time. Thus, we received the potentials map on the heart surface and for a certain period of time. And as a model of action potential, we use the cellular model of a human heart ventricle, TNP 2006. So the resulting geometry of the heart with the potential map and fiber field was used in pseudo-B-domain model to obtain a potential map of the torso surface and then to calculate the ECG signals. In order to obtain the personalized model, we varied the global conductance of the heart. We consider the cardiac tissue as anisotropic and homogeneous and solved the optimization problem. We selected the conductivity of the whole heart in such a way that differences between the amicard QRSD complex duration and the calculated model QRS complex duration were minimal. On this personalized model, we compared 3 stimulation protocols. The first one is ventricular stimulation from the clinical lead position obtained from CT data. The second one is ventricular pacing from optimized lead position. In this case, we fix a right ventricle electrode on the endocardium in the apex area, and left ventricle electrode was positioned on the epicardium on the lateral wall, and the position of left ventricle electrode was varied and was not limited by configuration of a coronary bed of the particular patient. And the criteria of optimization of left ventricle electrode was the minimization of the QRS complex duration. For both type of biventricular pacing, we set the 0 time delay between the electrodes. The third stimulation protocol is selective stimulation of his Packendian system. For that, we have identified the surfaces of left ventricle and right ventricle endocardium, and in the basal region, in the middle of the septum of left ventricle, the root node was set, and from this node, his bundle branch was grown. Since the left and right branches of his bundle are isolated from the working myocardium and are connected with working myocardium only at ends of the Purkinje fibers, the excitation of his Purkinje system was modeled separately from the heart. We set conduction velocity about 3 mm per millisecond and calculated the arrival time at each end node of the Purkinje system. And these end nodes with the corresponding time delays were used as a kind of virtual stimulating electrodes for solving Econal equation. This slide shows an example of a calculated activation maps from left to right. The activation map for a clinical lead position. Left ventricle electrode here is on the lateral wall. The activation map for pacing from electrodes with optimized position. Left ventricle electrode here is on the basal part of the anterior wall. And right figure is selective stimulation of his Purkinje system. Blue zones indicate the early activation, and red zones indicate the later activation. Total activation time for clinical electrodes was about 140 milliseconds. For optimized lead location was about 100 milliseconds, and for his stimulation was about 63 milliseconds. For every patient and every type of stimulation, we calculated the following indices of electrical desynchrony. The first indices are total activation time of the heart and the left ventricle. The second is maximal width of the QRS complex in precordial leads. The third are standard deviations of the activation times of the heart and left ventricle. The next one is the biventricular desynchrony index, is the difference between left and right ventricle activation time. Next index is the intraventricular desynchrony index, is the difference between the lateral wall activation time and the intraventricular septum activation time divided by total activation time of left ventricle. And the last one is the integral index, is the integral of difference where the first term is the fraction of the septal volume activated at time t, and the second term is the fraction of intraventricular septum activated at time t. This slide shows the values of indices of desynchrony depending on the type of stimulation. Each plot here is individual index, and each box are for a different type of stimulation. The light blue boxes is for a biventricular stimulation with clinical leads. The boxes in the middle is for stimulation from optimized lead position, and the dark boxes is for his stimulation. For most indices, his stimulation showed superior results to both types of biventricular pacing. For example, the average width of the QRS complex for his stimulation was about 70 milliseconds versus 174 biventricular with clinical leads, and about 154 biventricular pacing with optimal lead position. The indices characterizing the variance of left ventricle and heart activation times also demonstrate the superior results for his stimulation. But at the same time, it's interesting that these indices do not differ or slightly differ for biventricular pacing protocols. The index of desynchrony between ventricles, so the right top here, also has a lower absolute value for his stimulation. And it's interesting too that for both type of biventricular stimulation, this index has approximately the same absolute value, about plus 16 for stimulation from optimized lead position and about minus 16 for stimulation from clinical leads. It means that the optimization of position of left ventricle electrode in terms of shrinking QRS complex does not reduce the desynchrony between ventricles, even if we do not take into account the configuration of coronary bed of a particular patient. Intraventricular desynchrony index also shows the best results for his stimulation. So, in this pilot study, 3 stimulation protocols were compared. and most of the synchrony indices show that the his stimulation is superior to both types of biventricular pacing protocols. Thank you for your attention.
Thank you, Arseny. Okay. Questions are welcome. Please, okay. Keep the presentation. All right, any questions? Okay, got a question about the torso. Pretty simple question about, could you please show the plot of torso electrodes?
This.
The next one, okay, because it's very a good picture to see that the torso. How do you take into account? Very simple question. How do you take into account the fat? So the torso is if you get it personalized.
No, in this study we do not take into account the fat and the bones of a patient. We only use the blood lungs.
Resistance of the fat, and if the model is personalized, so you don't take into account. It's just all the same for all patients.
Yes, we personalized only the heart.
Only the heart, okay. Thank you. Any other questions? Please switch on. So, okay, Alexander Kursanov asked, you used the rule-based algorithm for assigning myocardial fiber orientation implementation. You can read it. Yeah. No. Can you read it in the chat?
No, I
copy for everyone.
I can't see any question in the chat.
For everyone. So I read it, and you use the rule-based algorithm for assigning myocardial fiber orientation implementation.
Yes, it's implementation of our group, and I know that Vladimir Zverev taken part in the implementation of this algorithm too.
Okay, thank you. Okay, no questions anymore. All right, thank you, Arseny, one more time.
Thank you.
The next speaker is Bajutina Anastasia from Ekaterinburg, the Institute of Immunology and Physiology. You're welcome. Please say something. We'll say, do we hear you? Actually, we don't. Anastasia, I don't hear you. Oh yeah. Now it's. Please say something.
Hello everyone, do you hear me?
Yes, we do.
I want to present my study about comparison of the cardiac resynchronization therapy results for personalized model with different accuracy. Cardiac synchronization therapy is procedure of implantation patient electrodes of the heart ventricles. The first patient electrodes is located on the right ventricle apex, and the second electrodes usually located on the epicardial surfaces. Cardiac resynchronization therapy is recommended for patients with heart failure. After this procedure, the ejection fraction increases and ECG duration becomes shorter. But unfortunately, about 30% of patient doesn't respond on this procedure. The reason of non-success cardiac resynchronization therapy is connected with non-optimal pacing electrodes position. We can use personalized geometrical and electrical models for maximization of cardiac resynchronization therapy. But we can build personalized model with different accuracy. We can use all ECG signals, for example, or only QRS duration. So the aim, in our study, we compared cardiac resynchronization therapy results for personalized models with different accuracy. In our study, we used retrospective clinical data of one patient. We had computer tomography data. We had magnetic resonance imaging data. Also, we had information about accuracy, location of cardiac resynchronization of pacing electrodes. Also, we had ECG signals on each point of the torso. This clinical data was provided Almazov National Research Center. On the slide, you can see pipeline for constructing personalized geometrical and electrical models. We got torso, hearts, and lungs geometries from computer imaging data. After that, we built finite elements model. We set fiber orientation in each point of the heart. We used a rule-based algorithm for it. For our patient, we built infarct geometry from magnetic resonance imaging data. Also, we built heart segments. After that, we may calculate activation map, and in the last step, we can compute ECG signals on each point of the torso. We used econal models for calculation activation map. After resolving this equation system, we got local activation time in each point of the heart. For calculation ECG signals, we used pseudo-binomial model. For calculation action potential, we used Tintusher-Panfilov model for human cell electrical activity. After resolve this equation system, we got ECG signals in each point of the torso. In our study, we built two models with different accuracy. In the first model, we set the same local conductivity in each heart segments. This model was fitted by QRS duration. The second model in the second model, we set the different local conductivity in each heart segment. This model was fitted by all ECG signals. In the infarct zone, we set the low local conductivity. For two personalized model, we varied positions of the pacing electrodes. The right ventricle pacing electrodes was located in the right ventricle apex. The pacing electrodes in the left ventricle were located in the center of segments model. We located, we placed left ventricle electrodes on the epicardium and endocardial surfaces. So we calculated activation maps for two models with different accuracy. For each personalized model, we calculated such electrical indexes. It is total activation time, QRS duration, and also we calculated 3 indexes of electrical desynchrony. Optimal pacing electrodes gives the minimum values of these indexes. For comparison, clinical and model ECG signals, we set the universal coordinate system on the torso. The first coordinates is longitudinal. The second coordinates is angular. On this slide, you can see the results of the validation results for two models with different accuracy in the universal coordinates system. The first map shows correlations between clinical ECG and model ECG. This model was fitted by QRS duration. The second map shows correlations between clinical ECG and the model ECG. This model was fitted by all ECG signals. For two personalized model with different accuracy, we got optimal electrodes position. The blue point shows optimal position for model which was fitted by QRS duration. The orange point shows location the optimal electrode position for model which was fitted by all ECG signals. How you can see, optimal electrodes can be located in the opposite segments of the heart. In our study, we built two personalized models with different accuracy. The first model was fitted by QRS duration. The second model was fitted by all ECG signals. For each personalized model, we got optimal positions for pacing electrodes. We showed that personalized model with varying accuracy gave different results for optimal pacing electrodes position. the global conclusion, it is necessary to use all ECG signals for building personalized models and optimization, cardiac resynchronization therapy. Thank you for your attention.
Okay, thank you, Anastasia. Any questions? All right. Could you please show your conclusions one more time? So the question is, if you got the personalized model, if you got the personalized treatment, and for the third conclusion, and that different indices of electrical dyssynchrony gave different positions. And the question is simple. When the electrical dyssynchrony changes for the patient in time, so you need to change the position of your stimulating electrodes, opening the chest. And so how do you think it can be useful for the treatment? Because it's not so positive conclusion that you should change the position of the stimulating electrode if you see the change of the synchrony. Do you understand the question?
If I understand, electrical dyssynchrony indexes is more useful for detection cardiac resynchronization therapy success. And we can use only desynchrony indexes for detection optimal electrodes position.
Okay, but during the treatment, for example, you can change the amplitude, for example, of the stimulus. And according to your conclusion, you should change also the position of the electrode, but you need an operation to change it. All right. Maybe it's a very stupid question. So any other questions? All right, thank you. Thank you very much. And the next speaker is from Penza, Markuleva Marina. You're welcome. Actually, I don't see her in the list. Maybe Mikhail Gerasimenko can answer. Marina, I see you in the list. You're welcome. All right, maybe she's out. Alexander Kursanov, are you ready?
Yes.
Okay, so we changed the program. We just now it's your turn. Maybe she doesn't know that the first talk was out. Okay. You're welcome. Alexander Kursanov, Institute of Immunology and Physiology, Yekaterinburg, Russia. I'm sorry. That's Marina talking, or Mikhail? You'll get. Okay, very, very bad quality. Mikhail, we don't hear you. Very bad quality of your voice of your microphone or connection. Ah, okay. So, okay. Now Marina is speaking, as I guess. Could you say something? As I guess, Mikhail Gerashchenko is speaking. Okay, very bad quality. Maybe. So I type you in the chat. All right, sorry. Okay, please type in the chat who will be speaking. Mikhail Gerashchenko or Merkuleva Marina.
And now? No?
Ah, now Mikhail, we hear you. Mikhail?
Just listen, if you hear me, just give us like around 2 minutes. I will reset some settings. Hold on just 1 second.
Okay, maybe we change now. Alexander is speaking. The next you will be. It's okay. So you begin at.
Okay, I start my presentation.
Okay, Alexander.
Good evening, dear colleagues. I want to present our work, arithmogenesis in calcium overload human cardiomyocyte in isolation and within cardiac tissue simulation study. As we know, what an overload of cardiomyocytes with calcium is one of the main causes of the cardiac arrhythmias arising at the cellular level. Rhythm disturbances are widespread and potentially lethal after effects of many heart disease. We study these processes of arithmogenesis on our electromechanical models, and in this presentation I want to demonstrate to you the results obtained in modeling the overload of human cardiomyocytes with calcium in isolation and within cardiac tissue. In addition, we studied the influence of fibroblasts on this process. Cardiac fibroblasts could significantly affect activity of the heart muscle during aging or pathology. Dynamic fibroblasts cardiomyocyte interaction may modulate electrical excitability, action potential form, or action potential duration, and mechanical performance of the myocytes. So recently we have developed new mathematical model of the human ventricular cardiomyocytes. In this slide, you can see the rheological scheme and ionic currents in the model. My colleague Natalia spoke about it in detail. I will just remind what's the key feature of this model is accounting for the intercellular mechanism underlying direct and reverse effects of mechanical and electrical activity on each other. The model allowed us to reproduce, predict, and explain the wide range of mechanical electrical feedback and mechanical calcium feedback effects in human myocardium. Previously, the model has been used to simulate electrical interaction between cardiomyocytes and fibroblasts via gap junction. during the twitch of myocyte. We have shown what electric fibroblast cardiomyocyte interaction results in action potential shortening and in significant reduction of myocyte contractility. In this study, we want to see how this affect on the calcium overloaded cell. Also, in this study, we used one-dimensional continuum model of the tissue as a sample one by a contribution of both electrical and mechanical intracellular interaction to the studied phenomena. The one-dimensional strand is formed from electrical and mechanical linked cardiomyocytes, each one being represented by human electromechanical model cell. Those we have three model: cardiomyocytes model, a tissue model, and model of the interaction of cardiomyocytes and fibroblasts, which we can combine in a different way. So it remains to describe how we caused calcium overload in our models. Calcium overloading in the single cell or cell within the strand was reached by inhibition of sodium-potassium pump activity by means increasing in the Michaelis constant for sodium current. This diagram shows the complete overload mechanism, how its realization in our model. The following slides shows the result obtained in the described models with calcium overloaded for cardiomyocytes. In a single cardiomyocyte, moderate level of inhibition of sodium-potassium activity causes after depolarization and extrasystolis at the very end of third phase of action potential. You may see it in the second panel. These written disturbances are based in the intense dissociation of calcium troponin complex during the relaxation phase of the cardiomyocyte twitch. This mechanism has been previously studied in detail by us and named cross-bridge induced calcium released from sarcoplasmic reticulum. Extrasystoles in the described simulation manifested themselves both electrically and mechanically as extra action potential and extra contractions. Upon completion of the extrasystolic attack, which lasted several tenths of seconds, cardiomyocyte demonstrates early after depolarization and force alternance. If two fibroblasts are connected to the cardiomyocytes, moderately overload with calcium, we observed effect disappear. Regular cycle of excitation and contraction are observed without delay after depolarization and extrasystoles. Much more significant inhibition of the sodium-potassium pump was found to be necessary for a period of early after depolarization and extracystosis, not only in the single cell, but also in the case its electronic interaction with two fibroblasts. But in this case, we onset of arrhythmia in the fibroblast cardiomyocyte model arose 1 minute later when in the single cell. With delay of the arrhythmia episode increased, we've increased the number of attached fibroblasts or with increase in the conduction of the gap junction. The reason for the observed relative modification of arrhythmia pattern was partial calcium unloading of sarcoplasmic reticulum in the cardiomyocyte when it's electronically coupled with fibroblast. We dedicate anti-arrhythmic effect of electronic interaction can be interpreted as a protective mechanism what's partly prevent from arrhythmogenic effects of coupling cardiomyocytes with two fibroblasts. So, also we study how local overload calcium may suggest in this phenomena. As we know, calcium overloaded of ventricular cardiomyocytes may have a local pattern due to the local distributions in the local area. In particular, way may be a local reduction of sodium-potassium pump activity in a segment of a ventricular cardiomyocyte. The simplest model to simulate with local calcium overload utilized in this study was one-dimension strand formed of two segments. One consists of the normal cardiomyocyte, and the second one consists of pathological cardiomyocyte with depressed sodium-potassium pump activity. Each segment was homogeneous within itself. In this figure shows the result of numerical experiment in which the pathological segment occupies 50% of the tissue. In this case, the pacing was applied to the border cell of pathological segment. Extracellular arrows in the cell of this segment show the red arrows in the middle panel. We spread from to the myocardial strand due to excitability of neighboring cell. As a result, an extrasystolic wave occurred, causing extra contraction, not only of cardiomyocyte overload with calcium, but also of all other cells in the one-dimensional sample. This led to extra contraction of whole sample in the bottom panel. The adding of fibroblasts to the cell in the one-dimensional strand resulted in the middle anti-arrhythmic effect, which similarly to that observed in the single model cardiomyocyte fibroblast interaction. So let me end my speech by this conclusion. The result of the modeling allowed us to suggest what even restricted
locus of the overloaded cell in the cardiac tissue may produce its electrical and mechanical disturbances. Specific pattern of arrhythmia in this case depends substantially on the locus location with respect to the direction of excitation wave. Interplaying electrical and mechanical intracellular interaction in the one-dimensional model modulates the pattern of studied arrhythmia as compared to the single cell, and we show what electronic interaction between cardiomyocyte and fibroblast lowers arrhythmogenicity in the isolated myocytes overload with calcium, and in the case of the one-dimensional strand. So, thank you for your attention.
Thank you, Alexander. Please, questions? Okay. Any questions in the chat? Okay, no questions. So, thank you.
Thank you.
So the next speaker should be Markuleva Marina or Михаил Геращенко.
Да, здравствуйте.
Здравствуйте. Hello.
Hello.
So you are welcome. The next will be Lisara Matasim. Okay, thank you.
All right. Good afternoon, dear conference participants. My name is Mikhail Gerashenko. I'm one of the team members who prepared the material named the brachial artery oscillometry process modeling with the pneuma and hydrocopter technology applying. So it is not the first year when our team of Penza State University has been engaged in the development and implementation of hydrocopter technology for blood pressure evaluation. So the relevance of this topic is due to the systematic increase in the number of people suffering from arterial hypertension. This category of people are forced to carry out home blood pressure monitoring, usually in the morning or in evening time, respectively. Easy to operate and most importantly, accurate blood pressure meters are in a high demand, actually. So in the market of medical equipment, so accurate intravascular pressure recording based on pulse wave analysis allow to improve the cardiovascular system state assessment reliability. So currently the only recommended method for blood pressure measuring, according to the European Society for Arterial Hypertension, is the Nikolai Korotkov's method proposed in 1905 its features are that it is problematic to measure the pressure independently. And it is also necessary to take into account the errors. The errors in the perception of the manometer's arrow fluctuations and the fixation of Korthoff's tones, according to some sources, actually. So the first tone can be heard by just small numbers of people. So taking into account the Korthoff's method features, research and engineering work on a new methods and devices development has been actively conducted recently. Asymmetric and photoplethysmographic methods are actively developing, and just in 1982, actually, Alexander Eman, in his work named Biophysical Fundamentals of Blood Pressure Measurement, showed that despite the widespread use of the oscillometric method, it has its drawbacks that affect the blood pressure registration accuracy. And the drawbacks are the material from which the cuff is made, then the density of its fit to the body, the patient motion activity, also the features of the algorithm for processing the curve of pressure micropulsation in the cuff. So these are some of them. The materials of which work relate to the comparative modeling of the brachial artery oscillometry process with the pneuma and hydrocuff device, as well as the calculation of a pneuma and hydrocuff tonometers characteristics. So the first thing was to present the process of transmitting the pressure generated by the pulse wave from the brachial artery to the sensitive element of the tonometer as a process of propagation of elastic perturbations in the wave channel, the structure of which is shown in figure 1. So it should be noted that the use of a hydrocuff tonometer ensures—
I'm sorry, Mikhail.
Yes.
I'm sorry, we see that you don't switch your slides.
That I'm done. Hold on.
We see only the first.
Oh. Let me fix just one moment.
Сейчас не видно? I'm sorry, сейчас видно?
Yeah, we see materials and methods now.
Next, materials and methods. Let's take.
Maybe you push F5.
Yes, we just tried like several times, but it's, we cannot see our screen either in this case. So just
put your mouse on the slide, all right, and then push page down. No F5. You can change your slides, maybe pushing page down and page up.
Tell me, please, what type of slide do you see right now?
Our materials and methods.
Materials and methods, and now also materials. And
also methods. Materials and methods.
And here,
results.
Results.
Number 8 we see.
Let's take up on slide 6. Hold on one second.
All right.
Here, do you see the?
Okay.
Oh?
Now it's okay.
So let's continue. It should be noted that the use of a hydrocuff tonometer ensures the uniformity of the medium in which the elastic vibrations propagate caused by the pulse wave, which will eliminate the adverse effects that occur at the interface between the two medium, taking into the account the accepted assumptions. The block diagram on figure 1 is converted to the form shown in figure 2. So let's take up on the slide 7. So this is all right. So the study also describes a model of a transmitting channel to obtain the equation of a pulse wave motion in a wave channel with a cross-sectional area by selecting a small section of a channel, volume S. Let's select a small section of a channel with its volume. So in order not to complicate the narrative, right, with the mathematical dependencies presented in this publication, let's move on to the simulation results. Let's take up on slide 8. Yes, here we can say that when modeling the pulse wave signal transmission through hydra and pneumocuff, a new variable called the propagation constant, which is characterized by two coefficients. The first one is alpha, the attenuation coefficient, and the beta, like the phase coefficient, is entered. So the slide shows the dependence of the attenuation coefficient alpha and the phase coefficient beta on the frequency. So it is for two mediums, water and air, actually liquid phase and air. So the wave propagation through the liquid is clearly visible at times higher than through the air. So let's take up on the 9th. So here also in the study, the wave channel transmission coefficients are determined for cases when the cuff and the connected tube of a tonometer are filled with air or liquid. So taking the surface area of the cuff as equal up to 0.03 square meters, right? The cuff thickness, we have about 0.005 meters. The tube cross-sectional area equals to 12 multiplied on 10 in -6 degrees square meter. So the tube length actually is about 1 meter. So the parameters of the cuff and the connecting tube for the cases under consideration are presented in the table 1. So the modeling conditions are described in the materials, and the table shows the two environments characteristics. So let's take up on the 10th and 11th slide. So here, the wave channel amplitude frequency characteristics comparing results are shown in figure 1. So the tonometer wave channel frequency response analysis shows that filling the wave channel with liquid increased the tonometer sensitivity several times. So at this point we can stop for now. So thank you anyway very much for your attention. Please just do one favor, please. In case of any questions, please ask all of your questions in Russian language. Yes, again, thank you very much for your time. Thank you.
Okay, thank you, Mikhail and Marina.
Да, спасибо.
The first question, what do you mean it was misunderstanding because we didn't see a couple of slides? Please show your device and please explain what do you mean the cuff in your device. Thank you.
The cuff is the like
okay, cuff manjeta. Okay,
yes.
We didn't see this slide. Okay,
actually you can see that, right?
Yeah. So it's, for example, the second device, it's on the finger, and you mean that
Здесь на слайде, что здесь за слайд? В общем,
yeah, what do you mean the cuff? Okay. Please show us.
Cuff, it is just like the vessel, right? Vessel where the air or liquid are stored, and actually this one of the main unit in the base of the tonometer. So if we are talking about oscillometric method. I mean, in most cases, like 90% of all of the tonometers which are used, especially in a domestic, right, domestic conditions, are electronic devices, like semi-automatic and automatic devices. So the most of semi-automatic and automatic device just has just as the main unit is like the cuff, and it depends just on the unit, like the air, like the main subject, actually the main object through which can just identify like some of the signals, right, from the artery to the signal detector. So but in our case, we just switch from the air, right, from the air material to the liquid, and the liquid are inside of the cup. So this is the thing.
All right, thank you. Any other questions? Okay, thank you. And the next should be Ali Motasim from Sudan. And I don't see him in the list. Okay. Okay, so I think that Ali is out, and the next turn is mine. Just a moment. All right. Do you hear? All right, my name is Alexander Rivkin, and I'm from Institute of Immunology and Physiology, Yekaterinburg. I will talk about the studying of the ontogenetic changes and electrophysiological properties of mouse cardiac pacemaker cells in silico. Okay, so the introduction. Next slide. Oh, got the same problem. All right. The sinoatrial node is the primary pacemaker of the heart and controls the heart rate through life. Loss or dysfunction of sinoatrial nodal cells result in sick sinus syndrome. A method for generating sinoatrial node pacemaker cells from stem cells could be facilitate the study of human sinoatrial node development and disease and provide a cell source for developing a biological pacemaker. Action potential morphology is the main indicator for subtype differentiation of development stage classification of IPS. And exact electrophysiological characterizations hampered by insufficient knowledge about electrophysiological properties of native sinoatrial node cells from different developmental stages. And the objective is for our colleagues who did the experiments were mapping sinoatrial node area in embryo, newborn, and adult mice heart, evaluation of action potential properties in an intact sinoatrial node cells from the early embryonic and adult stage. In a couple of words, so hearts were isolated from 4 stages of the development: 9 days postcoitum, 12 days postcoitum, newborn, and adult. And you see on the picture the form of the mice heart during and the development during the ontogenesis and after the birth. So the zone of the sinoatrial node was found during the carting, and the microelectrode method was used to study the action potential generation process. And then it was used the PRAM-AP standardized parameterization program to get biomarkers of the action potential, as you can see on the picture, the length of the diastolic depolarization, the length of the repolarization, frequency, and different stages of repolarization. Also, the rate of the action potential increase during the slow diastolic depolarization and fast diastolic depolarization. And the main results are presented on this slide, that embryonic cells have a very long cycle length and a very long slow diastolic depolarization, according with a comparison with the adult and newborn mice. As you can see on the plot, there are huge differences in cycle length and diastolic depolarization duration, and the rate of the fast diastolic depolarization. Okay. And the conclusion of the experimental part, we can say that accelerating action potential generation by sinoatrial node cells is associated with shortening of the slow diastolic duration and the acceleration of its speed. The second part is the theoretical study and modeling. We've used the single cell sinoatrial node taken from my mouse, developed by Sene Harche in 1211. So the main feature of this model that they use many variations of calcium channels, and they pay an attention on the internal calcium clock. It was based on the model of Maltsev and Lakata. They developed the model of the internal calcium clock connected with the sarcoplasmic reticulum and periodic releases of calcium through ryanodine channels. On the first step, we've done the experimental fitting, the model fitting of parameters, and the Carchi model was developed for the isolated adult mouse cell, but the values of the action potential biomarkers in the model did not match the experimental data in the fiber. The action potential obtained within the framework of the base model different from the experiment in a rather low frequency, 5.5 Hertz in the model and 8.3 Hertz in the experiment. So the model is slower than the real adult cell in the fiber because the model is for the isolated cell. And the fitting of parameters is quite hard process because changing the parameters of ion currents we change the frequency of the action potential generation. And we decided to use the PINS probabilistic inference for the noisy time series instrument. It's the package for the Python programming language to vary the conductance parameters of various ion channels involve the formation of action potential by the sinoatrial node cells. Scale factors of various ion channels conductance were optimized to align with the experimental data. On the first step, we fitted the experimental data for the adult cell, And one can see from the table that we've got the change of the role of isoforms of calcium channels, or L-type calcium channels, and also the increase of funny current, which is can be one of the main currents during the slow diastolic depolarization. So we increased the frequency of heart beating of the action potential generation, and on this picture you can see the basic model and our fitted model for the adult cell. On the next step, we decided to take that the adult cell fitted to the experiment is taken as a base, and then we used also the pins package to fit the model and to get the parameters of ion channels for the newborn mouse and for embryons 12 to 13 days postcoitum. And the results is shown in this table. One can see that for the embryons, we've seen the change of calcium transient current and the decrease of the role of internal calcium clock and the increase of the fast potassium current. And the main feature, one of the main features, was the decrease of the funny current involved in the slow diastolic depolarization, involved the authorhythmic activity of the sinoatrial node cell. To check our results, we switched on these effects, changing only the funny current, permeability, the calcium transient current permeability, and the rate of the potassium fast current. As one can see from the picture, the increase of the firing current resulted the increase of the action potential generation frequency, also like the increase of the permeability of calcium transient current, and increasing the fast sodium current, we saw the decrease in the frequency. And in the conclusion, I can say that Carchi model variation was discovered that a decrease in the conductance of calcium transient, funny current, permeability, the intensity of the calcium pump of the cytoplasmic reticulum, so the decreased role of intracellular calcium clock for embryons, and as well as an increase in the conductance of fast potassium current leads to a relative increase in the duration of the interval of the slow diastolic depolarization, and accordingly leads to the reduction in frequency of generation of action potential by sinoatrial node cells for embryons. Okay, thank you. Now it's time for questions. Okay, you can switch on your microphone and ask questions. Okay. Thank you. No more questions. And unfortunately, we didn't see our Africa guest, Alisara Motasim. He didn't write any letters that about the acceptance. And so I'm happy to finish this last session about the cardiac activity, computational modeling in physiology and medicine. And I want to ask Olga Eduardovna to finish this session, to say a couple of words, because it's the last session in this. Olga Eduardovna, can you tell? Can you switch on? Okay.
Unfortunately, no.
Unfortunately, no. It's very pity.
Yes.
Okay, thank you very much, everyone. So I guess that our conference, and I say thank you for Vasily Ilyich Borisov for cooperation, and it was really great to make a doubled conference with IEE, and it's really good, and it's a good practice, and I believe we'll continue this practice, and we'll make our conferences more often. So you are welcome.
Yes, yes. I hope this conference will
Hi guys,
be
excuse me. I was watching. I was watching translation together with our colleagues in the hall. That's why I wasn't on the Zoom. So let me thank everyone participating at our conference, and I would like to thank our co-organizers from Ural Federal University, from Siberian branch of Russian Academy of Science for this great job we've done together. And hopefully this successful story will be continued next years, every year. So I guess that our conference was very successful, very interesting. A lot of interesting works were reported, and we all are happy that that happens. Thank you very much. Maybe Vasily also wants to say some words to us.
Thank you so much. I think all for today, because we say all words, and I think I would like to invite all colleagues next year in next conference. I think it is a really great a really great way to cooperation between Siberian section of IEEE and Euro branch and Siberian branch of Russian Academy of Science. Thank you so much, dear colleagues.
And we wish that the next year pandemic will be over, and we'll have offline to see each other, to talk.
Of course. Thank you.
Thank you very much. Bye, guys.
Goodbye. Коллеги, трансляция.